embed the YouTube channel in the web pages.
Good. That's a good photo of us all, that.
Cool. Make this one public or publicar because the Fems YouTube has turned into Portuguese for reasons I don't understand. Well, I do. We will just make use of Google Translate. Right, okay. Now, we are technically live, although your faces can't be seen exactly yet because I've got a holding slide up. I'm just going to pop us into 2 web pages. And we're nearly ready to go. Embed code, okay. Sweet. And then last one on the International Microorganism Day homepage. Cool. Great, okay. We are live. So
looks good on both pages to me.
Looks good? Okay.
Yeah.
In that sense, I will commence with some ceremony. I'm going to stop the holding slide so it will all appear in one space. Great. Well, welcome to the live stream for International Microorganism Day. We're going to commence now the opening ceremony, and I'll run us through a little bit of an introduction. But I think this year more than any, we've felt a bit of how impactful the microbial world can be on everybody's lives. And while the new pandemic might have caused the cancellation of many wonderful International Microorganism Day events, all challenges present opportunities. And today we've been able to put together a truly international program of speakers from every continent almost in the world. And as the world spins on its axis, we will go through many different locations in the world through this wonderful live stream software and hopefully bring microbe lovers from across the world together and produce a wonderful collection of videos and talks and discussions for everyone to see. So in the call today, at the ceremony, we have 3 wonderful mascots. We have Sac, or Saccharomyces, our lovely yeast. We have Bac, or Bacillus, our slightly threatening bacteria. And then we have Gillis, who is behind Christina here, or represented by Christina, who is another wonderful fungus. All members of the Portuguese Society for Microbiology who've been so instrumental in putting this together. And then, of course, we have Isabel, who I will hand over to in a second, who is one of the founding figures behind International Microorganism Day, and will give us a little bit of an introduction to the history of this day, where we've been, how we've got to the point we are today, and run us through some welcoming slides to this year's edition and run us through a little bit of the previous edition. So, Isabel, can I hand over to you?
Oh my God. Sorry. I'm sorry. I think I closed the slides, International Microorganism Day. I'm sorry, I'm sorry.
Oh, that's okay.
I'm almost there, I'm almost there. I'm almost there. I'm almost there. Here I am. I'm very sorry for this.
That is okay. Well, here we go, Professor.
Cláudia não consegue passar os slides daí, não. É que eu não os consigo apanhar. Devo ter apagado.
So, do you have the slides?
Do you have the slides?
This is what happens, isn't it? Because this worked perfectly 10 minutes ago.
I had the slides here,
Yes.
but now, so here. Okay. Okay. So are you seeing the slides?
Yeah.
Okay,
Yes.
I am very sorry. I'm very sorry, and welcome to this counting down to the International Microorganism Day, the 4th edition of the International Microorganism Day. A very warm welcome and greetings to all of you that are watching us, in particular the students of microbiology, the microbiologists, also the societies of microbiology, and obviously, at last but not least, the microbes, because this is the day of the microorganisms. So why should microbes have their own day? So this is an important question. Well, they should have their own day because it is extremely important to raise awareness among young people, the society in general, for the role, the extremely important role of these invisible organisms that play an extremely important role in your lives. But they are also a vast area of professional activity and career. So it is obviously very important, and this is the mission of the International Microorganism Day. At this time, you know that we are facing a virus pandemic, and more than ever, it is important to understand that they are extremely important from the biotechnological point of view. They are extremely important in our lives, in the environment, in our lives altogether. And they have the day, and the day is the 17th of September. Why this day? This day is a reminder of a letter sent back to the Royal Society of London by Anton van Leeuwenhoek, who was a businessman. It was not a microbiologist, obviously, because microbiology was not a science at that time. And it does not have fortune and no academic degrees. So it is, let's say, a perfect figure for the dissemination of microbiology, because it has an enormous curiosity and ingenious, and obviously the date is the date of the reach of the microscopic life. So this is the idea behind this date. The history of the International Microorganism Day. So the International Microorganism Day started in 2017, so 4 years ago, and it was started in Portugal, and it was supported not only by the the Portuguese Society of Microbiology, but it was supported as well by the Order of Biologists and also Ciência Viva, Living Science Centers in Portugal, and the Society of Ecology, the Portuguese Society of Ecology, and under the auspices of the National Committee, the National Commission of UNESCO. This is important because the idea was just to turn this in a real International Microorganism Day with the support of UNESCO. This was the idea and still is. So it starts in Portugal, it spread to Europe, and in 2019 it spread to outside Europe with an important expression in South America, in particular in Brazil. So the idea is just to have a cohesive image that, let's say, put together under the same umbrella, and not only the same umbrella, but the same philosophy. Everybody commemorating the day all over the world. So it was very important to have these mascots that you have seen, because all the mascots, but also the logo is, let's say, the cohesive image of the day, linking together the community. So the first edition, 2017, was launched in Portugal. It was in Pavilhão do Conhecimento, so it's the Pavilion of Knowledge, so the home of the Contemporary Science Center. That is one of the centers of the living science, let's say, Ciência Viva, in Portugal. And we were able to have open laboratories and a lot of activities with high school teachers, but also with students. And you can take a look of what we have done in this pavilion. And I would like to present you the bastonário of the biology, the Order of Biologists. And here is Cristina and myself. And it's Rogério Terreiro, also from the direction of the Society of Microbiology at that time. It was the General Secretary. And here is Maria Amélia Loução, a colleague from Faculdade de Ciências and also the president of the Ecology Society. And as you can see, those students of Universidade de Lisboa who gave life to the mascots. And they were extremely important, and still are, during all the activities, because it was them who gave life to these activities. So from Portugal to Europe, you can see the flags, and now you can see all the supporters. The Fems enter here supporting us. And all those societies supported, or financially, or spreading the notice, etc. So here you can see 2018. It's the last picture of the day by 10 p.m. So we were just closing the sessions, and during all the day we have a lot of activities at Instituto Superior Técnico, who is my institution from the University of Lisbon, Universidade de Lisboa. And it was important because people come from all the place, supported by grants from Fems, and they work together at Instituto Superior Técnico in Lisbon. But a number of activities were developed in the whole country. We started the Skype calls across Portugal and Skype calls across Europe, so we were in contact with special places in Europe who were commemorating with us. And we have Skype calls also in Portugal. Here is a child in a school because we visited schools and we interact by Skype calls at Instituto Superior Técnico. It was live streamed, the sessions were live streamed as well. And it was a huge success. We have a training school for secondary teachers. This is important because it was the first time they heard about genomics and bioinformatics, and it was also a success. And in 2019, so the third edition, well, Fems entered and provided a lot of help and tools to improve the engagement of the public. So we have here Claudio Godinho, that were working with Joseph, our host, and with Ben Liberton, who is also with us. And they created, well, we entered the social media, and the message was spread wider. And the Fems also launched an art competition. So we were, let's say, improving. Important scientific meetings on microbiology or related topics. We were there presenting the day and advertising the day. And it was, for the first time, also commemorated in South America, Brazil, Cuba, and Paraguay, and also in Europe, and obviously in Portugal. And it was a big day in Portugal. The International Biodeterioration and Biodegradation Society was with us and prepared a number of activities. We also have activities in Romania and in other countries. And here we can see activities in Pernambuco, in Brazil. Here in Paraguay, you can take a look because we see the logo and the mascots, everything just the umbrella of the International Microorganism Day. So we were able to spread it and to join all these communities. And here it's my institution. And this was promoted by the Portuguese Society of Microbiology in Portugal. You are seeing here the mascots making experiments in the garden, because we had the microorganisms under the tree in the garden, and it was a super day. We were able to taste a new beer produced by an yeast developed in our laboratory. All the sessions during all the day presented by PhD and master students talking about their works. And we also have Skype calls in Portugal and also in Europe. So, for example, we contact Joe and Veran during the Fems summer school for microbiology education in Greece. And we also contact Fems office. I don't know if you remember, because you gave us the numbers of access to the website and all that. And well, we were in Portugal, in my institution, but we were together with all the country, with the ITQB, also in the gardens in Oeiras, Universidade do Minho, na Rua Augusta in downtown Lisbon, etc. And now we are coming to the International Microorganism Day. Obviously, due to the pandemic, we are not able of this kind of festivities, just putting together a lot of people. But during this year, we are able to, with the help of Fems, with support, the financial support from Fems, we were able to develop a new website, more professional. I would like to call the attention to the fact that the other website was bilingual, so it was in Portuguese and translated in English, so both languages. And now we have a blog in Portuguese as well. So I invite all the community talking, speaking in Portuguese all over the world, Brazilians, people speaking in Spanish, just to also to contribute to this blog, because this blog is not only in Portuguese, it's not a translation, it's a specific blog produced by us in Portuguese, by the Portuguese Society of Microbiology and colleagues, and you can contribute as well to this blog in Portuguese. So all the materials, all the contents of the old website was here. So discussing the mascot, the mission. We have an international blog in English where Fems is including a lot of new material produced with the Fems support. We have our blog, as you can see, with the flag. Under the flag, we have this information. We have a gallery with the story of the International Microorganism Day. And well, you know that Fems is supporting a new competition on microbial art, and it's also advertised in the site. There are educational resources produced by Fems. And well, you know, we are not able to commemorate it as we wanted and as we did it in previous years. And but the International Microorganism Day, the 4th edition, goes online. So goes online here, and well, this was the picture advertising the day in Portugal, so it's written in Portuguese. And you know that it's 24 hours of microbiology, web seminars, videos, discussions. Please stay with us, send your questions and comments, use our social media just to stay with us. And before finishing, I would like to ask you, let's say, to spread today, but not only today, every day, to be able to share and spread knowledge, microbiological knowledge, and just try to build communities based on microbiology, not only in this International Microorganism Day, but well, during the whole year. Thank you very much.
Thanks, Elizabeth. I will now show you a few stats behind the new website, which you were kind enough to point out. So let me just put some slides up. First, I have just the wonderful mascots in their primary colored glory. But yeah, as you said, we have a new website, which has only been out for about 3 months. But that's still enough time to have got great new blogs on there and for several things to become apparent. So this is just the tracking of the International Microorganism Day hashtag, which we have been doing since the end or beginning of July. And as of now, the night of the 16th, we have nearly 3.5 million impressions, which is, you know, 3.5 million times something with International Microorganism Day has passed across somebody's screen. It's reached over 1 million people, and we've had, yeah, hundreds of users and thousands of posts and tens of thousands of engagements in this time leading up to the day. We haven't even got to International Microorganism Day yet, or at least in Europe or in Central European time zone, we've just had 43 minutes of IMD. If you're still in Lisbon or the UK, you've got 17 minutes to go. Yeah, with our new website, we've kind of just started to reach the tens of thousands of page views and thousands of visitors. And it's really started to pick up in the last month or so. And for me, this is the most interesting slide. We can see that we've picked up people from all over the world in the last 3 months who've come to the website. Every continent's represented here and almost every country. There are a few that we can fill in, but we've got time over today and the next year to try and get every country in the world. And the real big players for us at the moment have been Japan, the United States, Portugal, as you'd expect and hope, the UK, Netherlands, Brazil, and India. So hopefully we can start to build on these centers as being where IMD is being shared and explored, and hopefully spread it to as far as we can in the world. And that's it from me. So I'm going to hand over to Hilary, who's the president of Fems, to say a few words for us before, yeah, the countdown is over. Thanks a lot.
Oh, thank you very, very much. Good evening, everybody. My name is Professor Hilary Lappin-Scott, and here in the UK, it's close to midnight, and we're getting ready to launch International Microorganisms Day in a few minutes' time on September the 17th. So as the elected president of the Federation of European Microbiology Societies, Fems, it's actually a federation of over 50 microbiology societies from all across Europe with over 30,000 members. I'm honoured to be part of this global event with you all, and I'm happy and delighted to warmly welcome you all to International Microorganism Day for 2020. So the idea of celebrating microbiology and microorganisms via its own day was cleverly started by the Portuguese Society of Microbiology, and we give them full respect for this, for starting it. So just a few years after it started, we're in a position now where many societies are joining in the celebration. So we're holding hands virtually across the globe to celebrate the achievements in our science and share these with everyone. So this year, 2020, it's been a really tough year for all of us, but the challenges of living with a novel coronavirus, SARS-CoV-2, it also unites us all, just in the same way as International Microorganism Day does as well. So for 2020, unlike the last few years, we can't celebrate in-person events in many different locations with the public like last year. But you know, in my opinion, it's more important than ever before that we share all over the world the importance of microbiology, all of the good things that microbial life forms do for the planet Earth. And as Isabel said a few minutes ago, let's use this to really spread the knowledge globally of microbiology. So as Fems president, I'd like to say thank you all for being part of this. I'm looking forward to working with microbiologists from all over the world to celebrate International Microorganisms Day with you. So on behalf of Fems, thank you to all the contributors, and I hope that you all enjoy the 24 hours of live streaming events. Thanks. Over to you, Joe. Thanks
very much. So for the final few minutes before we commence with the talks and discussions, I'm going to hand over to our three beautiful mascots, who are each going to deliver for us, I think, some poetry, some microbial themed poetry as well. So I guess we will start with Saka. It is your turn to begin.
Hello, I'm Saka. I'm the most famous yeast. I produce bread, beer, and wine that are present in every feast. You can find me everywhere, from soil to water, from fruits to skin. I can be on the open air, or even where your digestion spin. I'm part of the kingdom fungi, like mushrooms and mold. Be careful with the first ones, or you may not get old. But all nut yeasts are very friendly, like Candida, my sister. She causes human infections, and she's an antifungal resistor. Learn more about microbes on our 24-hour live stream. Check our detailed program and choose your favorite team.
Thank you very much, Saka. A wonderful introduction. So next up, and actually I'll just, hang on 2 seconds. Just going to just say that Saka is played by Dr. Margarida Palma, who is a postdoc at the Institute Superior Tecno at the University of Lisbon, and is a postdoc in Isabel Sacaria's group. So now we're going to hand over to Back, who is our second and more mischievous mascot, who also has some words for us.
Yeah. Hello, I'm back. I'm a famous bacteria inspired in bacillus. I can be your friend or your foe, so handle your hygiene with criteria. I represent the smallest cells, but don't take my size for granted. I treat me with antibiotics, and I show you ways to be adapted. Also, I have this angry look. Check your microbiology book. Not all bacteria are mean, but for some you really need a vaccine. I have been living in Earth for many, many years. I'm present in labs and dirt, and I help Saka with to do some beers. Come with us, see us shine. Just go with the flow, check our program online, and enjoy the show. Enjoy it.
Awesome. Thanks very much. It's a shame we can't have applause in the same way because there just isn't this microphones in every single house out there. But thank you very much. Just wanted to say as well that back is played by Lewis Martins, who is a PhD student again in Isabel's lab at the Institute Superior Tecno, University of Lisbon, who's actually in the lab for us today. So thank you very much.
Yeah. Thank you.
For our final, final representative, who represents our third mascot, Gillis, I'm going to hand over to Professor Christina Cruz, who will deliver some final words.
Hi, I am Gillis. I'm Saka and Bax's good friend. I take my name after Aspergillus, but I am nice in the end. We cross almost every day. Since my home is widespread, I can be in the vegetation in decay or even in your bread. It is true some fungi can kill you, but I will not be that guy. You will pass me through in a blink of an eye. But if your immune system is low, I will make your lungs sick. You will feel as cold as snow, and the recovery will not be quick. Follow IMD, do not be a fool. You just need good internet because microbes around are a powerful tool to make a friend out of a threat.
Awesome. Thank you very much. You can hear some applause from my microphone. Okay, fantastic. So this is it from our mascots. They have set us off on a wonderful start. I just want to check, Isabel, do you want to say any more final words before we move on to the next section?
Well, just perhaps to introduce Cláudia Godinho and Nuno Melo. Cláudia prepared these poems with, I don't know exactly if Margarida gave a hand as well, and Cláudia has been extremely important for us during all this year and in 2019 as well, because she was working with the Fems office, not only for the website, maintaining the website, updating the website, but for the advertisement of all the materials that we have been producing. So it is, let's say, an extremely important figure here for the day. And Nuno is a PhD student. Claudia is a young PhD, but Nuno is a PhD student starting his PhD, and he has also been very important because he produced all the very interesting and nice materials that we have been using while rewriting or refining the mascots and using them for the dissemination, disseminate the day and the activities of microbiology. So, well, I would like to thank them, and obviously the mascots that are here. And it's not easy to to put on the mascots, and they are giving life to them. Thank you. And Christina, who is the alma mater of the mascots and the logo and all that. So the start of this adventure and this big activity that now is spread worldwide is also due to Christina, because, well, the image of the day, we cannot never forget her, and obviously the artists that make the design and everything, and all who were the mascots and gave them life, because this is not easy, especially for Bach, because the young children were always beating them because they didn't like this mascot. This was the bad mascot. Just and well, thank you especially to Bach that he is in the laboratory.
Yeah, but I'm sweet.
I'm
very sweet.
Awesome. Well, that brings us to the end of our little opening ceremony. And speaking of the lovely logo, we're just going to put the logo up for a few minutes while we get the first of our talks ready to go on the dot of midnight in Lisbon time. I just want to say thank you to all our guests for joining us and helping us to get this off to a good start. And I hope that, yeah, for the next 24 hours, we run through a huge plethora of wonderful talks by an amazing group of speakers. And yeah, get to learn a lot and spread microbiology around the world. So thank you to everybody for joining.
Thank you very much from me as well. And passing on very, very best wishes for the live streams. Thank you, Claudia, Christina, Nuna, and the three mascots, and Isabel for your leadership as well. Thank you, everyone. Bye.
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 Bye, everyone.
Bye, everyone.
Bye.
We'll see some of you again.
Leave meeting.
And then there were two. So we have T-minus 3 minutes before our first video, our first talk. Just to give a bit of introduction while we ready ourselves, the first speaker, the very first speaker for International Microorganism Day 2020 is Maria Pinto, who is part of the University of Vienna, the Functional and Evolutionary Ecology Department. And her talk will be on bacteria living on plastic debris in the ocean, which is a fantastic way to start off International Microorganism Day in terms of an interesting and very, very relevant topic. So I will stop sharing my video for the meantime to avoid distracting anybody. And we will count down the remaining 2 minutes until we begin. And I'm pleased to say that we're going to be beginning on time, which is already a good thing to be doing. So that all's gone to plan. Right.
It's good, especially at the beginning, to be on time.
Exactly. We'll have to see over 24 hours if we do slip behind time, but I'm hoping that we can keep everything to a nice rigid order. But yeah, here we go.
Yeah, and if we do manage to keep everything in order, you'll be able to look at the program on the International Microorganism Day website, and you'll be able to see exactly what we're up to at any point during the live stream. So you can dip in and out and join whenever you find something that looks interesting to you.
And you can find the program. Ooh, hang on. Yeah, and you can find the program linked under each of the live streams on any of the social media accounts that you find them. So it should be fairly easy to follow along and see which talks you want to follow and watch. if you have a question for the speakers, please leave any of these questions as comments or as replies underneath the video, and we'll do our best to hand them on to the speakers, either during their talk if they're a live speaker, or if it's a pre-recorded speaker, then they can, when they wake up and check the live stream, they can see any questions left behind them. Just please make sure you name them. Right, okay. I'm going to stop sharing my screen because we're about to begin the very first talk for International Microorganism Day 2020.
Hello, everyone. First of all, I want to thank everyone for being here and for watching my presentation. I want to thank the International Microorganism Day for allowing me to show you this presentation, even though I cannot do it personally. If you have any questions after the presentation, please feel free to send me an email. I will answer them as soon as I am online. My name is Maria Pinto. I am a PhD student at the University of Vienna. My supervisor is Professor Gerhard Handel, and I work specifically in the Microbial Oceanography group at the Department of Functional and Evolutionary Ecology. And today I'm going to talk to you a little bit about the world of bacteria that live associated to plastic debris in the ocean, which was also the majority of my PhD project. First of all, what is plastic? Plastic is pretty much any synthetic or semi-synthetic organic compound that can be molded and shaped into solid objects. Here you see a wide variety of plastics or things that are made of plastic that we use in our everyday lives. There are several different types of plastic. You have probably seen these symbols in some of the plastic materials you use in your everyday. And this simply means that they have different chemical structures. So the main polymer has a different molecular structure, for, as you can see here. These are the most common plastics used and produced worldwide. The most common being polypropylene and low density and high density polyethylene. These are the most produced plastics. They are usually the ones that are found mostly in polluted areas, and they are also the ones that are mostly found at the surface of the ocean. Not only because they are the most produced and therefore the ones that are usually more wasted, but they are also the plastics that are the most buoyant. Other plastics are, some of them are denser, so they end up sinking to deeper layers of the ocean, and some of them even reach the sediments of the deep ocean. Because of their durability and their ease of manufacture, low prices, plastic production has seen a very steep and almost exponential increase since the 1950s. Continue on until 2020. So it is not unreasonable to assume that the amount of plastic entering the ocean is also increasing year by year. In 2015, John Beck et al. Tried to estimate how much plastic had entered the ocean in the year of 2010, and they estimated that out of the 270 million metric tons of produced plastic in that year, 8 million entered the ocean. And they also estimated that around between 6,350 and 245,000 metric tons of plastic were already in the ocean by that year. These estimates, as you see, this is a very large, big range, and these estimates are very difficult, and therefore, and some even suggest that these estimates in this study are conservative. So it could be that there's even more plastic than it's said here, but nevertheless, it is quite a large amount already. So what are the impact of plastics in the ocean? Well, usually the first thing we think about, and the things that most people know or think about when we talk about the impact of plastic in marine ecosystems, is the direct physical impact on organisms. However, there are other more invisible potential threats or impacts that we are not so much aware about. For example, the release of additives into the seawater. Additives are compounds that are added during the manufacturing process to the plastics to increase their performance, their durability, to be more resistant to biodegradation. And we know that they can be leached from the plastic to the surrounding environment, including seawater. And these, for example, here are just some examples of types of additives that can be added to the plastics. We also know that plastics can absorb persistent organic pollutants, for example, fertilizers that are already in the seawater. And because plastic is so durable and they might transport these POPs across long distances. And the main focus of today's presentation, we also know that they are a surface for bacteria and other organisms to colonize. Of course, anything that is on the plastic and that is consumed by organisms at any point can be then transferred throughout the food webs. But okay, so let's now talk about the main focus of this presentation, which is the microorganisms that live attached to plastic. So what this is called a biofilm, and a biofilm is this kind of structure that forms on surface. Bacteria and other microorganisms create this sort of layer on top of surfaces. Here, for example, this is a photo I took with a scan electron microscope from a piece of plastic that I collected in the Northern Adriatic. Here you can still see a bit of the plastic, and all this fluffy structure is the biofilm, and you can even see some diatoms, so some cells embedded in this kind of mesh. And a biofilm is basically an assemblage of microorganisms that attach to each other and via something called EPS, which is an extracellular polymeric matrix, And it's a kind of a glue that allows them to attach to each other and attach to surfaces. And we do know that they attach to plastics in the ocean. Just to give you an idea how this works. So there are around 1 million bacteria per milliliter of surface seawater around the world. This is an average estimate. And some of these bacteria, like when they encounter surfaces, plastic, for example, they like to attach to the surface. Being attached to a surface and creating a biofilm can provide a lot of advantages to the microorganisms. They can have easier nutrient availability, protection against harmful environment, easier to exchange favorable genes between them, and there's other advantages. So biofilms are, for many organisms, very advantageous. Living in a biofilm is, for many organisms, an advantageous lifestyle. And so some of them, from these 1 million bacteria per milliliter, like to attach to the surface. Then they create this EPS that I was talking to you about before, which is this sort of glue that allows them to attach to each other and to the surface. They continue dividing. Then there's new players coming in, predators, viruses, organic matter, and the biofilm continues developing. Sometimes there's some organisms that leave, sometimes there's whole chunks of the biofilm that detach. There's other organisms that come to the biofilm. So it's while it is relatively stable in comparison to the surrounding seawater, it is still a habitat that is developing and kind of changes over time, depending on which stage of the development we are, or the biofilm is. In 2013, Zettler et al. Published the first study that used molecular tools to determine the community composition of the biofilm on plastics from the North Atlantic that they collected from the surface of North Atlantic waters. And they called this biofilm the plastosphere. And the main things they saw in this study was, first of all, that the plastosphere is composed by a diverse community of prokaryotes and eukaryotes, that the prokaryotic community of the plastosphere was different from the one in the seawater, which is not very surprising because as I said before, only some organisms like to attach and live in biofilms. So not all organisms that are in the seawater will participate in the creation of the biofilm, or just are not components of the biofilm. So this is not surprising. It's common for any biofilm. The community is usually different than the one in the seawater. And furthermore, something which was interesting is that there were some observable holes on the surface of the plastic. It's really hard to sometimes tell whether this is the plastic surface or just another part of the biofilm, but it was very interesting. You could still see these holes that seem to have the shape of the organisms that were in them. So there's not much you can conclude from that, but still interesting to keep in mind. And since this study, there have been many studies trying to understand how this plastosphere affects the fate of plastics in marine environments. And part of my PhD is also was trying to look at this. And one of the first things we need to understand before reaching a conclusion on how the plastosphere is affecting the plastic in the ocean is understand what are the factors that influence this community, its development and its structure. And it seems like it's not that simple. It's not like one or two factors that just affect the communities everywhere the same. Usually, like any other community, it's affected by a complex network of factors. These are just some of the suggested ones. We know, for example, that geography is an important one. By this, I mean plastics collected from different water masses have biofilms or have plastospheres with different communities. This is also not very surprising because different water masses have different properties, different characteristics, different pH, temperature, nutrient availability, chemical composition, and so on, that usually affect any community. So not only the ones on the biofilm, but also in the seawater. So it's not very surprising that there is different plastospheres from different locations have different communities. But we, for example, in one of the projects of my PhD was trying to see if this was true. I had plastic samples from these stations in the North Atlantic, North Pacific Ocean, Northern Adriatic, here off the coast of Rovin. And we did see that, for example, the plastosphere of plastics collected in these two stations were relatively similar to each other, but then different from these two, and these two were relatively similar to each other. And that's because here you have one water mass and here you have another water mass with different characteristics. And the same for here in the North Atlantic. This one, which was coastal, had a very different plastosphere than the ones that were not so coastal anymore. And you could see a gradual change of plastosphere with further off from the coast you were. So that even though it's not surprising, it is still something that now we know, and that has been shown several times with other studies as well. The next thing, which I also try to focus a little bit on my PhD, is how does the plastic itself influence the plastosphere? Because geography and every parameter that varies depending on geography and water mass is known to also influence other biofilms and other microbial communities. But is plastic different? Is the plastosphere different from other biofilms? And is the plastosphere between different plastic types different, or do all plastics have the same community? Are plastic additives somehow influencing the plastosphere, and are the plastics degradable? These are all questions that we have been trying to answer, and that I also delved into a little bit in my PhD. And so I just want to plug in a part of my project that is related to that, in which we try to understand if different plastic types that were incubated for the same amount of time in the same location developed different plastispheres, or if they were all the same. We also then had a glass control where to see if the glass and the plastics actually had the same communities. So I just want to show you here a short clip to show you this experiment. This was off the coast of Rovin in the Northern Adriatic. We had all these different plastics. This is me and my colleague Teresa, and we were, after 1 week, 1 month, and 2 months of incubation, we would go out here and collect the plastics, and then via molecular tools, we tried to characterize the different communities of the different plastic types that were living attached to the plastic types. I can tell you that immediately after 1 week, there was already a visible biofilm created. So the biofilm establishes quite fast. And the different plastics we studied were these ones here. So high and low density polyethylene, polypropylene, and then 2 PVCs, that's polyvinyl chloride, one which had the additive of DEHP and one which it didn't have DHP, instead had DINP. And then also the glass control to see if it had different communities from the plastics. So the results of this study are already published in PLOS ONE. And the main findings were, which are the most interesting findings from the study, is that the communities of the PVCs seem to be relatively similar to each other, but different from these other 4 surfaces. So the glass and the polyethylenes and polypropylenes. And on the other hand, these 4 seem to be very similar to each other. Now, it's very important and worth noting that PVCs are the plastics that usually have the highest amount of additives. For example, the ones that we used in our study had up to 40% of their weight was additive. So in the PVC DEHP, up to 40% of the plastic was DEHP. And while we cannot say for sure that that was the reason for why the community on these plastics was so different from the ones on these plastics, it's still an indication, and it's still something worth looking for in the future. Even though these were similar and these were similar, there were always certain species that preferred apparently to attach to certain plastic types over the entirety of our incubation. And the highest difference between plastic, or between communities, between plastispheres of the different surfaces, was in the beginning, so after 1 week of incubation. And that is not necessarily surprising either, because in the beginning is when the organisms that are attaching are in direct physical contact with the surface. Therefore, if the surface is somehow influencing the community attachment, then this influence will be stronger earlier on than later on, when the biofilm is thicker and the organisms that are newcomers are not so much in direct contact with the surface. And so this is very interesting, and it answers the question of whether or not certain different plastics can have different plastispheres. We see that yes, in this specific case at least, because there are other studies performed in other parts in other water masses with other plastics in which they don't see the same pattern. So it appears that the plastisphere is only specific for certain plastic types under certain conditions. And so this is something that we still have to research further on to understand, because apparently the plastisphere is only specific for certain plastic types, sometimes under certain conditions. And we are still trying to figure out when and why. Something very important that I did forget to mention is that in this study, we only looked at the prokaryotic component of the plastisphere, which means the archaea and bacteria, and we didn't really look at the eukaryotes. So this could change. The results that we obtained for the prokaryotic community could change if we would look at the eukaryotic community. And it has been shown in other studies that they do not necessarily, that sometimes the prokaryotic communities are very similar, but the eukaryotic communities are different between different plastic types. So I just wanted to leave you here with, end this presentation with this figure, which really shows the complexity of this topic. I just talked to you a little about a very small part of the interaction between microorganisms and plastics, but this is something that we and people who are studying this are really trying to figure out how plastics and their attached communities interact with the environment and the fauna and flora that lives around them. So I hope that you enjoyed a little bit of diving a bit into the world of plastic pollution in the ocean and microorganisms. I want to thank, of course, my group. And again, I want to thank the International Microorganism Day for inviting me and allowing me to do this presentation and showing you all this that I just showed you. And I want to thank my funding agencies, the university, and the Institute of Hudar Bošković in Croatia for being our partner. And I want to thank you for watching this. Please feel free to send me an email if you have any questions. And thank you very much for watching.
Well, what a great way to kick off International Microorganism Day. We've had a few comments already on social media about questions, and so we have some pre-recorded talks, and obviously the speakers won't be able to answer questions live, but if you post them on social media, then we'll make sure that that they have a look and try and get back to you. And you see that some of the speakers also post their contact details in the presentation so that you can contact them directly if you want. So in the name of sticking to the schedule, we're going to move right on to the next pre-recorded talk, which is from the Micro-Eco-Evo team of the Biological Science Research Group from the Instituto Superior Técnico at the University of Lisbon. So I will...
Evolution team is a branch of the Biological Science Research Group at Instituto Superior Técnico in Lisbon. Our mission is to advance knowledge of the function and diversity of microorganisms in nature. Here we give emphasis on the environmental and host-associated microbiomes in marine settings, their implication to host and ecosystem health, climate regulation, and potential use as renewable resources of novel biotechnological appliances. Our primary mammal hosts are marine sponges and soft corals from the Atlantic Ocean, particularly Portugal, but we also have projects on algae and in the aquaculture sector. Corals and sponges, they rely on really tiny microbial symbionts living inside them for survival. Those symbionts, they provide sugars and other goodies, they help in waste recycling, and they even protect them from pathogens and predators through the production of chemicals. I think this is really fascinating.
The symbionts of sponges and corals possess a really interesting secondary metabolism, which we can look for new activities and see if they can enter the drug discovery pipeline. We look into the genomes and metagenomes of these marine microbiomes to search for new interesting biosynthetic gene clusters.
My PhD project is about the discovery and isolation of novel chitonases from marine sponges and coral bacterial symbionts. I think that this project is very interesting because it will allow us to isolate chitinases from food waste resources like shred shells, reducing the environmental impact and the costs for the industries. Chitinases are enzymes that can degrade chitin, the most abundant polymer of the ocean and the second most abundant in the planet. Chitin fragments obtained from the action of chitinases can be used in the production of bioplastics, and those chitinases can also help to degrade them after all.
I'm a PhD student, and the study of the Arctic ice microbiome is part of my integrative PhD study.
I like my PhD project because it's the discovery of the unknown and integrates different areas of expertise. The study of the Arctic ice microbiome is key to different areas such as astrobiology, biomedicine, and of course environmental science, because microbes can be signatures to climate change.
One of the emerging research topics addressed by our team concerns the fish microbiome and the extent to which it helps the host organism to combat infectious diseases. This is of relevance in aquaculture, which is more than ever becoming an important sector of today's economy, where fish densities can be quite high and disease proliferation may be favored. Our project, Microcontrol, evaluates the use of fish probiotics in controlling bacterial pathogenesis in aquaculture. The idea that symbiotic microorganisms can serve as a reliable and environmentally friendly alternative to disease prevention is what excites me the most about this project.
Great. And so again, please keep asking questions. We're on live now on Facebook, on Twitter, and on YouTube. And we'll go straight into the next pre-recorded talk now from Paulo Durão from IGC in Portugal. And he will be talking about the role of evolution in the marine. I've just lost the slide there. The role of evolution in the maintenance of antibiotic resistance.
Bacteria are the oldest inhabitants of Earth, and you can find them literally everywhere. Most are beneficial or harmless, but some can cause infections. Fortunately, antibiotics allowed us to control harmful bacteria, making surgical interventions possible and healing previously dangerous diseases. However, antibiotics are becoming less effective lately due to the rise of antibiotic resistance. Bacterial success as colonizers relies on their incredible adaptability and their huge numbers. Mutations, which are changes in the DNA code, occur spontaneously at a very low frequency, but large bacterial populations likely include individuals carrying mutations. Most mutations are deleterious or neutral for the bacterium, but some might be beneficial in certain conditions. In antibiotic-rich environments, such as our societies, mutations conferring resistance provide a big advantage. Moreover, mutation is not the only way to acquire resistance. Bacteria themselves produce antibiotics and thus often carry genes encoding resistance proteins to protect them from their own antibiotics. Furthermore, resistance can be transferred from bacterium to bacterium favoring the spread of resistance. Antibiotics target proteins involved in processes essential for bacterial physiology, and resistance mutations often land in the genes encoding these proteins, disturbing their normal function, therefore causing problems to the bacterial cell. Thus, when the antibiotic is absent, most resistance mutations actually slow down the growth of resistant bacteria. This negative effect is called the fitness cost of resistance and is important to us because if, for instance, a few susceptible bacteria survive an antibiotic treatment, the cost allows them to outcompete resistant bacteria once the treatment is over. Lowering antibiotic usage would decrease the advantage of resistant bacteria. Unfortunately, it does not completely prevent the spread of resistance because of an evolutionary phenomenon called compensation. Compensation occurs when the resistant bacteria acquires additional mutations that alleviate the cost of resistance, allowing resistant bacteria to grow efficiently both in the presence and absence of antibiotic. Moreover, there are interactions between resistance mutations where one compensates for the cost of the other. Talk about bad luck. Scientists studying antibiotic resistance know, however, that the cost and the compensation of resistance depend on the environment where bacteria grow. Traditionally, the costs have been measured mostly in laboratory conditions, but scientists are now starting to measure them in animal models, more similar to clinically relevant environments, to quantify the real costs of each resistance. Antibiotic resistance is a very serious problem, but don't get pessimistic. Research and health institutions, such as WHO, ERC, NIH, and others, have made a top priority reducing antibiotic resistance. At this moment, thousands of scientists around the world are working hard to stop resistance dissemination by investigating solutions and alternatives to antibiotics, such as phage therapy, antivirulence drugs, novel vaccines, and more. Additionally, novel policies to limit the misuse of antibiotics are being implemented. Here are some things you can do to help. First, it's important to use antibiotics correctly. Antibiotics should be a last resort, reserved only for bacterial infections that our organism is not capable of fighting on its own, and never for viral infections, because antibiotics only kill bacteria, not viruses. Second, if you really need antibiotics, make sure you take them exactly as the doctor prescribed, at the dose, and for the number of days recommended. And of course, washing your hands, using soap, and getting vaccinated helps a lot. By preventing infections, you avoid the need for antibiotics in the first place. Let's resist resistance together.
Another great talk for International Microorganism Day. If you're watching this on any of the social media channels, we're broadcasting live right now. Please don't be afraid to say hello, ask questions. We'd love to hear from you, and we'll get back to you if we can. We'll get the speakers to try and engage you there as well. I'm going to play another 2 videos right now. The first one is going to be a research talk from Maria Miragaya from ITQB Nova in Portugal, and she's going to be talking about Staphylococcus epidermidis and skin protection. And then just after that, we're going to play a short video from Divulga Micro, and they're going to be talking about the SciComm activities that they have going on their Twitter and Instagram accounts. So I'm going to play the Divulga Micro video first.
I am Maria Miragaia. I'm the group leader of the Laboratory of Bacterial Evolution and Molecular Epidemiology. In our lab, we focus our research in a particular group of bacteria, which are coagulase-negative Staphylococci.
In the case of Staphylococcus epidermidis in particular, this is a species that is one of the first bacterial species that colonizes humans when they are born. I think it's also important not only at emotional level, but also at the microbiota level, that babies have contact with the mother's skin. Because the Staphylococcus epidermidis is one of the main colonizers of the skin. And so when baby gets contact with the mother, it's going to take on the Staphylococcus epidermidis, but also other bacteria that are in the skin of the mother. Besides the role as stimulating the immune system, it has also a role on fighting against pathogens. So there are several examples also of these Staphylococcus epidermidis, reducing, for example, bacteriocins that inhibit pathogens like Staphylococcus aureus, which is a human pathogen, a non-human pathogen. This awareness of the good bacteria is a more recent thing. And so nobody cared much about Staphylococcus epidermidis. But now there is good bacteria and probiotics in general are gaining more and more importance because bacteria, people are understanding that if we have a good balance of our microbiota, then we are healthy. And if there's something that breaks this balance, then disease cannot occur. So they are bacteria or virals that live on us, and usually they are good. But in certain occasions, they can become opportunistic pathogens. The kind of infections that are caused by these bacteria are infections that are associated to medical devices. And so what happens is that, for example, when a person goes to the hospital and gets a catheter into their veins, the model that exists of this infection is that the bacteria that are living on our skin are those that are going to have access to blood, and they are going to create a biofilm all around the catheter, and this is going to cause the infection. So I was interested in this conversion from a good bacteria to a bad bacteria, and what were the factors that were making this conversion possible. Our first studies showed that this bacteria that we thought that was kind of endogenous and that each person has its own Staphylococcus epidermidis, we got to the conclusion that it's not the case and that this bacteria can be transmitted from person to person and can even be transmitted from country to country. And we understood this because we compared them genotypically, and we reached the conclusion that they were very similar strains that were causing infection, for example, in Portuguese hospitals with those in Denmark. And so this was one of the first studies that we did. And so we understood that actually Staphylococcus epidermidis infections, when they are opportunistic pathogens, are not caused by their own flora, but they can be caused by pandemic flows that are disseminated everywhere. In this case, we got to the conclusion, because we could do a deeper analysis, that we had two clones of Staphylococcus epidermidis mainly. And so we got curious to understand, okay, so we have two clones. Are they exactly performing the same function in humans, or are they performing different functions? And actually, we got also to the conclusion that there was one clone in particular that appears to be more pathogenic than the other. This means that if they are present in the skin, then they are more probably going to cause disease if opportunity comes than the other one. So what happens when a bacteria goes from the skin to the blood is that to cause infection has to have the ability to survive in both environments. So we think that the more pathogenic clone is more successful because it's able to live in both environments, while the other clone is more adapted to the skin environment only. So if we know what is the kind of clone that we have on our skin, if we can predict if a bacteria is more pathogenic or not, or if the risk of infection is higher, and that's one of the purposes of our studies is also to try to help in the clinical setting for doctors to have a better idea how to deal with this kind of infections. One of the questions that remains is so if these two clones, how they interact with the human hosts, if they interact in the same way or in a different way. Another question that also we would like to understand is how this bacteria can be so recombinogenic. Because we think if we can control this characteristic, we can somehow control the pathogenicity of this material. And so that's another line of research that we would like to follow.
And now we're going to move directly into the next talk, which is a slightly long one, from David Clark from the University of Essex in the UK, who's going to give his presentation, a pre-recorded presentation, live in a pinch of salt, the extreme world of halophilic microorganisms.
Right, hello everyone. My name's Dave Clark, and it's my pleasure to join you on International Microorganism Day 2020. I hope wherever in the world you're watching this from, you're keeping safe and well. So my name's Dave Clark, as I mentioned. I'm a research scientist at the University of Essex in the east of England. I'm a microbial ecologist, which means that most of my research revolves around the two broad questions of why do microorganisms live where they do, and what are they doing there? What benefits or what processes are they contributing to in the environment? And to answer these questions, I get to play some pretty cool toys. So I get to use DNA sequences and cluster computers to perform experiments and analyses that even as little as 10 to 15 years ago weren't really possible. This is a pre-recorded talk, so unfortunately there's no time for sort of live questions at the end, I'm afraid, but I am on Twitter at Microbicology, and I'd love to hear from anyone with questions, so please feel free to reach out and get in touch if you have any questions. So obviously microorganisms have been getting a bit of a bad press recently. The current COVID pandemic, of course, has completely changed all of our lives and the way we go about our daily lives. But I wanted to talk about a group of microorganisms that I think are really, really cool. And the reasons why I think they're such an exciting group of organisms to study. So if you're watching some of the other talks on International Microorganism Day, then you probably already have a bit of an idea about what microorganisms actually are. But for those of you who might not have seen the talks, then I'll present a sort of brief summary. So obviously the first thing about all microorganisms is that they're very small. So that means they're mostly single-celled organisms or unicellular. As opposed to humans, we have several trillion cells that make up our bodies. They're also incredibly abundant. So the human population on Earth at the moment is about 7.8 billion people. If you go into your garden or into your local park or wherever and pick up a handful of healthy soil, there'll probably be more microorganisms in that handful of soil than there have been people that have ever lived. I think that's pretty amazing. And most life on Earth is microbial, so we estimate that the number of microbial species is actually greater than the number of stars in our galaxy. But what actually are microorganisms? Well, they're sort of divided into three main groups. So we have the bacteria that most people have heard of. These include things like E. coli. Then we have the archaea. And then we have the eukaryotes. And of course, we as animals are eukaryotes, like all other animals and all other plants. So most of the living things you see in your daily lives are eukaryotes, but there are also many microbial eukaryotes, so things like diatoms and single-celled algae. But the one thing that really unites microorganisms is that they are absolutely everywhere. So as you're watching this, the surface of your skin and the inside of your body and your gut are absolutely crawling with microorganisms. In fact, probably about 50% of the cells in your body are actually non-human, actually microorganisms. Similarly, the waters around you, the seas, the lakes, the rivers, surfaces and insides of plants, even the very air you breathe is home to microorganisms. And when I say they live everywhere, they really are everywhere. So even in very extreme environments, such as those that are very salty. And we call these environments hypersaline. And these can be a real variety of habitats. So these can be as large as the Dead Sea or as small as an evaporating rock pool on your local beach. They can be warm, like the Mediterranean, or they can be cold. So there's even hypersaline environments in the Arctic and Antarctic regions. And they can be natural, or they can be human-made, like salt pans where we gather sea salt. And these hypersaline environments are all colonized by microorganisms. And we call these microorganisms halophiles, which means they love salt. So what actually are halophiles? Well, most of them are actually archaea. So most of the species and most of the cells that we find in these environments are archaea. We also find bacteria to a lesser extent, and more rarely eukarya. So most of the eukarya we find belong to a single type of algae called Dunaliella. In my opinion, the most amazing of these organisms is this organism. This is called Haloquadratum Wolfsbii, and this is a type of archaea that is absolutely unique because it has square cells. And as these cells reproduce, they form sheets that look like little window panes. This is absolutely unique in nature. You can also see halophiles from space. So this is a satellite image of Lake Tyrrell in southeast of Australia. And like many salt lakes throughout the world, you can see it has this pinkish and even these dark red hues. And this is caused by the dense blooms of halophiles that live in these waters. And the reason they're these colours is because many halophiles make pigments, so often pinks and reds and greens, and that acts like a sunscreen which protects them from the high UV radiation that they experience in these very exposed environments. Halophiles, of course, in that they love salt, can occupy environments that are much, much saltier than seawater. So seawater is approximately 3.5% salt. Halophiles will happily live in environments up to about 40% salt, so about 10 times saltier than seawater. So how do they do this? Well, let's first of all look at what happens if we placed a normal microorganism in a hypersaline environment. So I've represented here a microbial cell, say an E. coli cell. as this pink rectangle. And if we placed it in a hypersaline environment, what would happen is that because the water inside the cell is far less salty than the water outside the cell, there would be a net movement of water from inside the cell to outside. That would cause the cell to shrivel up, and pretty rapidly it would die. Halophiles actually have two main strategies of coping with the high salt concentration in their environment. And these are compatible solutes, or what we call the salt-in strategy. So most moderate halophiles actually use this compatible solute strategy. So here the cell actually makes chemicals inside it that help balance the salt concentration inside and outside of the cell and retains water that way. But this is very costly for the cell. The cell has to spend energy and resources making these chemicals. So you'll find that most extreme halophiles, the ones that live in the saltiest environments, actually prefer this salt-in strategy. So rather than synthesizing these chemicals inside the cell, these halophiles actually pump salt into their cells. This has the effect of equalizing the salt concentration inside and outside of the cell, and thus keeps water inside. The disadvantage is that these cells have to have proteins and cellular machinery that make the cell work that are very well adapted to high salt. And as such, it means they can't really live anywhere else. So one of the things about hypersaline environments is that they often experience really high evaporation. They're often in very hot places and very exposed places, and as such the briny waters tend to evaporate very quickly. Now, as the brine evaporates, the remaining brine becomes what we call chaotropic. So that means that the salts that are left in the brine as it begins to evaporate are very damaging to the cell. They can disrupt the cell membrane and cause the cell to die. So how do microorganisms cope with this? Well, I think the coolest strategy is that many microorganisms can actually entomb themselves within forming salt crystals. So as the brine becomes more and more concentrated as it evaporates, salt crystals begin to form. And inside these crystals, there can be little pockets of fluid. And in these pockets of fluid, microorganisms can sort of hide themselves away inside the crystal and wait for either rains or tides to come and refresh the brine, in which case the salt crystal dissolves and the microorganism can carry on as normal. So to give you a sense of scale, a microorganism in one of these brine pockets inside a salt crystal is approximately the same as one of us standing in an Olympic-sized swimming pool. So here's an image one of the PhD students in our group has generated. So she's taken some brine from a natural environment and left it to evaporate, and it's formed these salt crystals. And as you can see, these crystals have the characteristic sort of pink hues inside them that tell us that there are lots and lots of halophiles entombed inside them. It's a really cool image. And this is something that we at Essex have been very interested in, particularly the question of, can all halophiles survive in salt crystals, or is it just a small proportion of the community and species? So to answer this question, a former master's student, Tom Hubie, conducted a really cool little experiment. So he took brines from this salt pan in Sicily, and he brought them back to the lab and let them evaporate in Petri dishes. And after 1 week, 3 weeks, 8 weeks, and 21 weeks, he would take samples of the salt crystals and he would dissolve these and look at the microbial community that had been entombed within inside these crystals. And what he found was really interesting. So actually, a very high proportion of the community that's in these initial brines was also there after 21 weeks. And the number of cells that were in these brines was practically identical after 21 weeks. So it tells us that actually a very high proportion of the community in these initial brines is capable of surviving inside the crystals, at least over the duration of a few months. But what happens if these brines don't get refreshed? What happens if the rains never redissolve these salt crystals, or if the tides don't return? Well, one of the things that can happen is that these salt crystals eventually become buried and fossilized. And they can remain fossilized for millions and millions of years. And probably the most remarkable thing about microorganisms, and particularly halophiles, is that some of them can actually survive inside salt crystals buried under the ground for millions and millions of years. And we actually have reasonably strong evidence now of halophiles being revived from ancient salts as old as 260 million years. So just stop and think about that for a moment. That means there are microorganisms buried deep within the earth that have been buried inside salt crystals since before the time of the dinosaurs. I think that's just mind-blowing personally. And it also means that they're probably the oldest organisms on Earth. We know of some trees that can reach thousands of years old, but definitely nothing as old as the halophiles. The other interesting thing about this ability to entomb themselves within salt crystals and survive over millions of years is that we know that other planets and stars in our solar system have large deposits of evaporite minerals. So, for example, salt is an evaporite mineral. It's formed by the evaporation of water. We know that planets like Mars have large deposits of other minerals such as gypsum, which is also formed from evaporation. So could these gypsum deposits preserve alien microorganisms from a time when Mars had large expanses of water on its surface? It's a really interesting question, and it's one that current Mars expeditions are trying to answer. There's a number of expeditions to try and find signatures of life on Mars. So I hope that was interesting. It was a brief snapshot into why I love microorganisms so much, and particularly halophiles. I want to thank in particular Professor Terry McGennity, who's also giving a talk on Microorganism Day, so please check out his talk. As well as Miriam Magula and Tom Hubie for their work in the lab in our group. As I said at the beginning, please feel free to send me any questions via Twitter. I'm more than happy to receive questions. And if you'd like to find out a little bit more about my research, I've put a link to my personal website here, so feel free to check it out. Other than that, please have a fantastic International Microorganism Day. Keep safe, and I hope you find all the other talks really interesting. Thank you very much.
Okay, so it's my pleasure now to introduce our next live guest of the evening, and that is, well, evening where I am at least, but wherever you are, whatever time of day it is, it's my pleasure to introduce Erin Boyby, who is here with us on the call now. Erin is a PhD candidate at the University of Rhode Island in the US, and she's going to talk to us about how to crochet your microbes. Hi Erin, how are you?
Good, you?
I'm good, thank you. Thank you very much for for being here. I'm just going to change into the gallery view so we can see you there. We've got a video to show because you've done a really cool, really interesting project for International Microorganism Day, and I can see some of your handiwork there to the side. Would you mind showing it to us?
Yeah, so I just figured out patterns on how to crochet the different mascots for International Micro Day. So the 3 different mascots, Back, Gillis, and Saka. And the patterns are up on Twitter and my website.
Cool. So we'll definitely share those. And you made a video for us for how to actually crochet one of those mascots. So I'm going to play the video for everybody now, and then we can maybe have a little bit of a chat afterwards, and we can see if anyone has any questions from social media. Does that sound okay?
Yeah.
Fantastic. Okay. So here's the video coming up.
Hi guys, so in this video we're going to be going through how to crochet one of the International Microorganism Day mascots back, seen in the image here. And also this is what the final product should hopefully look like. Should take maybe a few hours to make total. And before we get started, we're going to get all our materials together. These are the yarn colors we're going to be using, and then you'll also need a crochet hook, tapestry needle, fiberfill stuffing, scissors, and maybe some measuring tape. This pattern starts off with 6 single crochets into a magic circle. Once you have your 6 single crochets, you're going to pull the end of your yarn tight, and that'll pull those stitches into a circle, as seen here. And then you're going to close that circle or join the ends using a slip stitch. The first round of the body is going to start with chain 1, followed by 2 single crochets in each stitch all the way around. This should end with 12 stitches around the edge. The second round is going to be a single crochet in the first stitch, followed by 2 single crochets in the second stitch, repeated 6 times in the round for a total of 18 stitches at the end. The third round of increases is going to be 1 single crochet in the first 2 stitches, followed by 2 single crochets in the third stitch, repeated 6 times all the way around, and you should finish with 24 stitches around your edge. The next round is just going to be single crochets in every stitch. After that, we're going to do another round of increases, which is going to be 1 single crochet in the first 3 stitches, followed by 2 single crochets in the 4th stitch, repeated all the way around for a total of 30 stitches around your edge at the end. Next will be another round of single crochets in every stitch. Now we have one last round of increases, which will be 1 single crochet in the first 4 stitches, followed by 2 single crochets in the 5th stitch, repeated 6 times around for a stitch total of 36 at the end. And now we're going to lengthen the body with 1 single crochet in every stitch for about 20 rounds. And once we have that, we need to stuff it before starting our decreases. So now we start our decreases, which will mirror how we increased from before. And this first round is going to be 1 single crochet into the first 4 stitches, followed by single crochet 2 together in the 5th and 6th stitch. Repeated 6 times around, and you should finish with 30 stitches around your edge. The next round will just be single crochets in every stitch. The next round of decreases will be single crochets in the first 3 stitches, followed by single crochet 2 together in the 4th and 5th stitch. Repeated 6 times until you end with 24 stitches in the round. And now we have another round of single crochets in every stitch. Our next round of decreases is going to be 1 single crochet in the first 2 stitches, followed by single crochet 2 together in the 3rd and 4th stitch, repeated 6 times so that you end with 18 stitches. The next round will decrease again, this time with 1 single crochet in the first stitch and single crochet 2 together in the 2nd and 3rd stitch, repeated 6 times so that you end with 12 stitches. Our final round will be single crochet 2 together 6 times so that you end with 6 stitches. Next, to make the pili, we're going to start by picking up 6 single crochets in a round on the surface of the body we just finished. Once you have your round, you're just going to work 1 single crochet in each stitch and continue until the pillows is the height that you want. And then we'll repeat that all around the body until we have the number of pili we want. Once we're done with pili, we can move on to the face. For the eyes, we're going to start with 3 single crochets in a magic circle, and then instead of working in rounds, we're going to just work 2 rows of increases, just as we did with the body. The irises will just be 6 single crochets in a magic circle, and then we're going to repeat all of these steps to make 2, and then we'll thread a pupil and a white spot onto these eyes and sew them together. Next, we'll add a green detail with slip stitches along the top of the eye, and then we'll sew the eyes to the body. Then we'll add eyebrows using slip stitches just above the eye on the body. To start the mouth, we're going to chain 9. Now working back across this chain, we're going to do 1 single crochet in the first 7 stitches, followed by 2 single crochets in the 8th stitch. Now, working back across the top of this chain, we're going to do 2 single crochets in the first stitch, 1 single crochet in the next 6, and then 2 single crochets in the 8th stitch. And we'll finish by closing it with a slip stitch. To start the teeth, we're going to chain 26. And we'll join the ends of the chain with a slip stitch. Now, starting in one of the stitches from that circle you just made, you're going to chain 3. Now we'll reattach that chain to the circle 2 stitches over from where we began. And we'll repeat that 4 more times until we have 5 teeth. And we sew the teeth over the black piece and the mouth onto the body. So now we can use our tapestry needle to add any final details we want. And hopefully the final product should look like what we had in the beginning. And the pattern for these guys and the other mascots will be posted online.
Wow. So thanks very much for the video, Aaron. Let me bring you back in so we can see you there now. Yeah, can you hear me okay?
Yeah.
Yeah, cool. Yeah, I've got so many questions. I mean, I've never crocheted before in my life, but I've got to admit that I'm kind of inspired to start by this now. Do you have any tips or advice for me kind of getting started?
I've been told by friends, I actually started with knitting when I was probably like 6 years old. And I've been told by friends that crocheting is much easier than knitting because you only have to deal with one needle.
Okay.
But I just spent a lot of time on YouTube. I taught myself maybe 5 or 6 years ago.
Okay, right. So relatively recently compared to knitting then?
Yeah.
Yeah. Cool.
But crochet is definitely easier to make different figures with.
And is this the project I should be starting with one of the giant microbes, or maybe something a bit simpler?
So I've taught some friends how to knit, and most of the time I start with basic shapes, and like at least with knitting we start with scarves because they're just long rectangles and you don't have to worry about different stitches. The patterns I've written are all pretty basic crochet stitches. There's nothing too fancy, so I'm not sure. It depends, I think, on how the type of learner somebody is.
But I suppose there's plenty of material on YouTube to try different things and get started.
That's kind of how I started, and then I got to grad school and started coding and realized that knitting and crocheting are just different types of codes that you just have to interpret. And once you know what you're reading, it's kind of just second nature. And now I don't really think about what I'm doing.
That's interesting. I don't know why. Maybe it's when you say coding there, it kind of makes sense when I was kind of watching the video and kind of see that the way that you kind of annotated the patterns, it does feel a bit like coding. But that's very interesting that you draw the parallel there.
Yeah, I saw there was an article recently about a lab that studies the physics of knitting, and they related it to coding, and it made a lot more sense all of a sudden that, yeah, it's just
really?
a different language.
Cool. And then you come up with all these patterns yourself. How do you do that? Is it a case of trial and error, or can you kind of look at a shape and more or less know the pattern or how it's going to be formed?
I just started writing patterns when the pandemic started, because there was a Twitter thread started by Zymo Research asking people to show how they were social distancing. And I posted, I think, some pipettes, and people started asking me for the pattern. And I realized that I was good at looking at shapes and making them, knowing how to manipulate the yarn to make that shape. But then I also realized that it's no different from lab work, where if you're trying to figure out a new method, you're just constantly writing down what you're doing, and that's what a lab notebook is. So then I realized that I can just go backwards, and once I figured out the shape, I can go through and write down what I did as I went. And they're definitely not perfect patterns, but it seems to have gotten positive responses from people, especially with everybody trapped in different places right now.
Yeah, I'm not surprised. I mean, I was definitely taken by them, not only because the patterns looked cool when you share them. I mean, that's one thing, but the fact that you'd actually taken the effort to write it up was amazing, because even for someone who's never done it, it felt somehow accessible, because you need some level of instruction as a beginner to actually get into it. So it's a huge asset. You mentioned, sorry, go on.
I got a lot of really cool emails too at the beginning of the pandemic, of messages from people asking if I could email a PDF, because sometimes the images aren't clear for people to read. And I got one really nice email from a woman who was working as a medical examiner and was isolated from her family, and crocheting was apparently her coping mechanism at the beginning of all of this, and that she really appreciated having patterns to work off of. So that was like one of the first patterns I posted, and then that kind of motivated me to keep trying new things and making more patterns to share.
That's amazing. I mean, that must be hugely inspiring. I mean, that's great. Before we wrap up, you mentioned that you kind of saw the parallel with your coding and your work, your graduate study work. What are you actually working on?
So I study the biodiversity and biogeography of protus communities in coastal ecosystems across Indonesia. So we just use metabarcoding data sets, filtering seawater, and trying to fill in sampling gaps where projects like Terra Oceans and other research groups haven't been able to get the permitting to do that research. So hopefully getting some stuff out soon.
Great. And so where can people find out about this, your research, and find out and see more of your patterns?
I just published my website this past weekend. So I'm going to start uploading my patterns there so people can download the PDFs. And I have a research page describing different projects I've been involved in since college.
Cool. What's your website? Do you want to give us the address, or will people find it through your social media?
It's linked to my Twitter, and it's probably not findable on Google right now. So.
Okay, so we go and find you through Twitter, and we find your website there. Great. Well, yeah, thank you very much for sharing that with us and for inspiring so many people to pick up their crochet hooks. Are they called hooks or needles, or?
I think it's hooks, but I call it a needle.
Okay.
So.
Cool. Then thank you very much, and yeah, I look forward to seeing more patterns and more research from you in the future.
Thank you.
Okay, bye. Okay, definitely inspired. Getting ready for our next talk now. Hello, Benjamin, can you hear me? You're on mute at the moment. Can you unmute yourself? It's in the bottom left-hand corner.
Yeah. Yes. Can you hear me?
I can hear you. Fantastic. It's a huge pleasure to have you with us, Professor Benjamin Ortega Morales. Without further ado, because we want to hear more from you, and in the interest of time, I'd love to just hand over to you to tell us about your fascinating research. Yeah, over to you. Thank you very much.
Well, thank you very much. Okay, so I'm pleased to be here and giving this talk, and I'm very happy to be part of this celebration of the International Microorganisms Day. The title of my talk will be Living a Toxic Relationship, Biofilms and Nanomaterials. First of all, I like to describe what a biofilm is. A biofilm is a layer of microorganisms which normally develop over surfaces, and those organisms can develop over different types of surfaces. So in this slide, you can see that some of those stained surfaces, which appear as darkened surfaces, are in fact mostly comprised and made up by living organisms, bacteria and other organisms. In the lower right of the image, you can see myself. I'm doing some kind of microclimatic monitoring of Tikal, which is a Mayan site in Guatemala, in which you see luxurious development of microorganisms living over these Mayan the artifacts, and which under certain circumstances promote degradation. But not only on ancient cities or ancient monuments we see the development of such layers of microorganisms. We can also see them in our own households. So when you see these images, I took them this very morning, and in Mexico, we have a difference of 7 hours to Central Europe. And those stains that you see are made up mostly by microorganisms. Some of them produce some kind of pigments, some kind of dyes, which protect them from the aggression of the environment, mainly the high levels of solar irradiation. So you can see them indoors, but also outdoors. If we have to talk about biofilms, and I'm very pleased to have heard some of the previous presentations, these biofilms can be considered to be cities of microorganisms. So on the left side of the slide, you can see a very crowded building in which you see apartments of people living there. So if we have to compare ourselves to living cells of microorganisms, we could be seeing at three-dimensional levels the development of the organisms inside of houses. And outside, we have the walls and the ceilings. Well, that's part of the biofilm, and it's made up mostly of exopolymeric substances, which are some kind of glues which are produced by the organisms, which help them to attach to surfaces, but also to protect them as a buffer to the atmosphere. But in fact, this image that I'm showing you, you would be seeing like a city. In fact, this self-organized group of cells can develop at different levels, and you see organisms living there, but also at the same time, they are perpetuating themselves to cover some surfaces. Which is interesting. Another concept is when we talk about nanomaterial. What is a nanomaterial? A nanomaterial is a type of material which is very, very small in size. But when we saw this material at this size, the properties, the physical and the chemical properties change a lot. And these nanomaterials can be derived from natural resources, but also from man-made anthropogenic sources. For example, in a urban atmosphere, of the exhaust of diesel and gasoline vehicles, or also as a source provided from the stationary combustion. So, which is very important about these nanoparticles, some of them exhibit some kind of biological activity. Nanomaterials have been used recently also to protect cultural heritage. So nanomaterials can be used for the photocatalytic and their catalytic property, or also as consodilants, which is some kind of glues which bring back deteriorated, degraded material. And in that way, they provide some protection to degraded materials. In our region, in the state of Campeche, which is located in southern Gulf of Mexico, southern Mexico, next to Central America, we have one of the most important Mayan sites. And there's been some use of nanomaterials to provide this consolidation in order to protect the materials. But some of these nanomaterials have also some antimicrobial properties. So recently we have been studying the potential of fungi and other microorganisms in the bioweathering, in the biodegradation of the surface of limestone, which is the main building material used with the Mayan used to build this magnificent culture. And just to give you a perspective, we still have about 800,000 people whose first language is Mayan, and the second is Spanish. So this is just to give you an idea of the importance still of the Mayan culture. So when these biofilms grow over the surfaces, under certain conditions, they degrade limestone and other materials. And it is very interesting to say that there is a concept which is called bioreceptivity. Bioreceptivity is the ability of a material to be colonized by living organisms. And this is a very important concept because we can use it for cultural heritage protection, but also in our own houses, and also in other types of materials which are prone to biological degradation. So in our case, for us, it was important to understand and to use a different approach in order to control this biological colonization. So nanoparticles have been used recently because they have some kind of biological activity. They have antimicrobial activity. I think that most of us are aware with the current pandemic situation we're living in the world, which is caused by a virus. And we have heard that it's important that we wash our hands and that we sanitize surfaces. Well, nanomaterials and antimicrobial nanomaterials possess certain properties which are important to control microbial colonization and also antiviral properties to sanitize surfaces. So in these antimicrobial properties have been studied for several types of metal-bound nanomaterials, which is some kind of metal which is linked to a nanoparticle very tiny particles which, under certain circumstances, they interact with biological material. They can degrade, they can break, they can bind different molecules, and in doing so, they inactivate microorganisms. And also, they can produce some kind of chemical species like the reactive oxygen species. And it is very interesting because this reactive oxygen species is some kind of the same procedure which is normally important for aging in higher organisms like ourselves. So in this slide, I'm just showing you that this is very important that we consider the shape and the size of the nanoparticles and the type of metal which is used to conduct cell death. So there is use of copper, of silver, and also titanium dioxide, which is very, very important in this case, and to provide this kind of antimicrobial activities. In this slide, I'm showing you that these biofilms, under certain circumstances, it is very important that we control them because they, at the first sight, they produce some kind of aesthetic deterioration. But under certain conditions, you can see this slide. It's interesting. This is a location in Japan. So an artist considered the importance of this biofilm, and he just scraped around the wall, and he left the shape of an apple. And to the right most side of the slide, you can see myself, I'm in Germany, I'm seeing this slab of sandstone under this item of an alloy of metals. You're seeing there is some lixiviation of ions, and it is very interesting because the grayish and greenish coverage over the slab is mostly biofilm, but under the lixiviation of the runoff of the materials, you are seeing mostly the sandstone, which is in a pink appearance, which is in this case is producing a natural control of the biofilm. So as you can imagine, in science, there are many different types of tests or different types of conditions when we test to see if a new nanomaterial is important and is relevant for control of different types of organisms and different types of methods to determine if they are efficient. And just to show you in these slides, some of the previous studies that have been published in the specialist literature, that some of these greenish or blackish surfaces over the walls or over the ceilings, mostly on outdoor surfaces, are very important. And under certain conditions, we see this efficiency, which is important to control those organisms. Just to give you a perspective, in Mexico, for example, in southern Mexico, we have in Yucatan Peninsula, we have temperatures which reach perhaps over the surface 55 Celsius degrees. And when you have the coverage of biofilms and you don't have them and you just eradicate them, there is a difference of between 10 and 12 Celsius degrees. So you can imagine what is the biophysical impact of biofilms growing over a sill, over the roof of a house, on the gain of heat, and how much it costs for a household to pay for energy using air conditioning. So this is a way which is very important to understand the impact of these biological entities in our living and daily conditions. So I was explaining to you there is a different ways of making those assessments to see if those organisms can be important, those nanomaterials, sorry, are important or are efficient to control the colonization and the viability of organisms. This is just to show you these very dark surfaces, which are mostly associated to outdoor environments on this religious building in my home city of Campeche. And you see that mostly in those areas, when you see the runoff of water, you see mostly this association of biofilms. And these kind of pigments mostly are produced by cyanobacteria, which are some kind of phototrophic organisms, which are lower plants like. And those organisms are producing those pigments because they need to protect themselves from these desiccation conditions, high UV, high radiation. And these pigments are, and some of them are very similar to the melanin that we have in our cells on our skin, because this is another biological adaptation to protect ourselves to excessive irradiation that we have in these locations. So these black biofilms, which colonize limestone, are mostly dominated by cyanobacteria. I'm showing you here those areas. In some people have asked me in some conferences if the building will fall apart because the biological colonization no, of course not. But you don't really need to have excessive or colonization of microorganisms to see or to lose the integrity of a building. In here, in yellow, you can see there is a warrior carved over the surface. So if you have biological colonization and you are not able to see the iconic representation of the warrior, you're losing cultural heritage. And cultural heritage is not only related to tourism, it's also related to our own identity as people in different languages. So another aspect of our research deals also with the ecotoxicological consequences of releasing nanoparticles in the environment. So we can use some kind of treatment to control the biofilms, but we also need to understand that if those nanoparticles are active, is because they can also have a potential impact over other non-target organisms. And this is very important for us. So in order to meet the time, I'm just showing you, this is my team, our group, we will work in one of these areas, and just want to emphasize, I'm just very, very happy to have been here and be part of this very important initiative. We need to increase literacy of microbiological sciences among the public, among the men at the street level. Thank you very much for this opportunity.
Thank you so much for a very interesting talk from a very, very important angle. I mean, discussing microbiology, microbial degradation in the context of protecting cultural heritage is something I haven't thought about before, and really great to hear your perspective on it and to hear the important work that you're doing. So I don't have time to ask you all the amazing questions I want to get answers to, but I have one question from Facebook, and I wonder if you could share with me where people can follow up with you to ask you more questions on social media. Does that sound okay?
No, I would be glad and an honor to do so. I'm also very enthusiastic scientist. I thought 3 years ago that scientists should spend more time promoting and communicating our results. Now I'm more convinced that we really need to talk more about our research to increase our public awareness. I do believe in the importance of social networks, so I would be happy to do that. I have a Twitter account that I can provide, and I would be and I'm very happy to reply and to answer the questions if there is any. Thank you very much.
Okay, thank you. And so I've seen your Twitter account. We'll share that through International Microorganism Day. And then if I could just ask you this one question. So I've got some compliments about your talk on Facebook. And also have a question from Mohit. who says, can we use these nanoparticles against viruses to inactivate them as well?
Well, I think that if we look at the Environmental Protection Agency webpage, I just did that a couple of weeks ago, and I found over 60 pages of different antimicrobial agents and also antiviral agents, which are or could be used for the to control COVID. So I think that antimicrobial, but also considering different types of nanomaterials. And I think that the possible use is also at different levels. So what we need to understand is that the widest spectrum, the better for a new material to be used and to be developed and potentially commercialized.
Perfect. Thank you so much for your time, and I'll be following your research and we'll be in touch on social media. Thank you very much.
Thank you very much.
Okay, we're going to move swiftly into our next talk now, which is a pre-recorded talk from Daniel Tamarit, who is in the Netherlands. And his talk is on the growth of the microbial tree of life and the evolution of cellular complexity. So without further ado, over to Daniel.
Hello, everybody. My name is Daniel Tamarit, and I would like to start by saying that I'm extremely happy to be able to contribute to this event. So thanks a lot to Valerie, Deanda, and to Joe Shuttleworth for the invitation. So yes, I'm a postdoc at the laboratory of Thijs Ettema in Wageningen in the Netherlands. And I'm going to talk about the incredible growth that has occurred in the microbial tree of life and how this has impacted our understanding of the evolution of cellular complexity. So in order to visually introduce the tree of life, I'm going to start by quoting Carl Sagan. In his show Cosmos, he said that there's an unbroken thread that stretches from the first cells to us. And I think this is a very powerful metaphor to see how we directly descend from some of the primordial organisms in our planet. And it will also be very helpful to visualize how all species on Earth are actually related. So in order to see this, here we have a stick person. So this person represents the whole of humanity and the entire human species. And this person is holding the end of a thread. So this thread will go back in time and we're going to follow it. So very soon, of course, we would find the last common ancestor of all humans. But then if we keep on going generations after generations, some millions of years ago, we would find the last common ancestor of all primates. But we can keep on going, and then we would find at some point the last common ancestor of all mammals, and then of all animals. So we are already some 500 or so million years back in time. But we can keep on going, and around 2 billion years in the past, we would find the last common ancestor of all eukaryotes. So eukaryotes are organisms that have cells with a nucleus and mitochondria, and they are going to come up again quite centrally in this talk. But yes, we can still keep on going, and some 4 billion years in the past, we would find the last common ancestor of all living beings and the first cells related to our lineage. So as you can see, this was a very, very long journey of around 4 billion years. And this really comes to show how a currently living species directly descends from one of the original organisms in our planet. But of course, this is an exercise that we could have done with any other currently living species. And if we would do that, we would see that each one of those species would hold the end of a thread, but the other end would always be the same. So these threads would interconnect and interweave, and together they would form what we call the tree of life. And the tree of life, of course, contains all species that live and that have lived. And we can easily find some that we are very familiar with, Like for example, here in green, you can see plants and algae. In yellow, you can see fungi. And then in orange, you see all animals. And these three groups contain basically everything that we can see with the naked eye, all organisms that we can see with the naked eye. Everything else is majorly microbial. So you can really see that the tree of life is mostly a microbial tree. And then if we look at it with a global perspective, something that we currently do in order to classify all current life is to divide into three different categories. So here in pink, you can see the eukaryotes, cells with nucleus and mitochondria. And then the rest of the organisms would be divided into two different groups. So we have on one hand the bacteria here in blue, and then the archaea here in yellow. And the tree of life, of course, is extremely diverse. And you can see that the bacterial and archaeal side of the tree are hugely diverse as well. But this is the diversity existing in this tree is something that we have only started to really glimpse in the last decade or perhaps two. And before this, we had a very, very reduced knowledge of the lineages that exist in our planet. And it's advances in massive sequencing that have really turned this upside down. So one already classic example of this is this figure published in 2016 from Laura Haag et al. In a paper in Nature Microbiology, where the authors showed the diversity known at the time. And here you can see the lower branches here in green, eukaryotes, and then the rest of the lower branches would be archaea. Then everything else in the upper part of the tree would be bacteria. So here you can see this purple group that the authors named the candidate phyla radiation. And this included a lot of newly discovered lineages that we simply had no idea existed. And you can see by comparison with the rest of bacteria, this represents a huge chunk of the tree of life. And of course, additional lineages within bacteria have been discovered in the last decade or so. And this is also true for the tree of archaea and within eukaryotes as well. So in order to describe this in archaea a little bit more, you can see this figure from a review from 2017, where the authors described how the archaeal side of the tree of life developed in the last decades. So yeah, in the 1990s and early 2000s, we knew of only two archaeal groups, the Euryarchaea and the Crenarchaea. But then the following years were very informative in showing that really we didn't really know much about archaeal diversity yet. And in this coincided with the years that I started to study biology. So for the first time, I understood what archaea were. And then every year of my studies, one archaeal lineage, one phylum or equivalent group was described for the first time. So that was really mind-boggling to me, because it seemed that very late in biology, we were starting to understand diversity on Earth. But indeed, it was only the beginning. And here you can see in the bottom half how the tree of archaea looked like by 2017. And there's many additional lineages that we simply didn't know before this time. So for example, here you have the Deepan archaea that before we only knew one lineage, the Nanoarchaea, and yeah, everything else was unknown, and it has happened to be a very interesting and diverse group of archaea. And here in pink in the lower right side, you have the Asgard archaea. So Asgard archaea are a fascinating group. I'm going to focus more on them. And yeah, you will see how their discovery has also not only revealed that we knew very little about the diversity of archaea, but also helped shape the tree of life a little bit better. So in order to talk about Asgard archaea, I'm going to present the efforts that led to their discovery. And it all started with a group of researchers choosing to explore the microbial communities existing in the bottom of the ocean. And they sampled the ocean floor in the North Sea, close to a structure called Loki's Castle. And of course, they found a lot of interesting microbes, but one that really stood out was what was called low-key archaea. And low-key archaea were fascinating in several regards, but of course their most salient feature was that they seemed to be the closest relative to eukaryotes that was known by the time. So the origin of eukaryotes is a very complex field that has suffered a lot of controversies and has focused a lot of attention of researchers. And something that was established by the time was the fact that the ancestor of eukaryotes originated from two different sources. So there was a symbiotic event of two different organisms, and their fusion and merger basically originated eukaryotes. So these ancestors were an archaean on one side, and then on the other, some bacterium somehow related to an alpha proteobacterium that originated mitochondria. But very little was known, particularly about the archaeal ancestor of eukaryotes. And the discovery of the low-key archaea, which was published in 2015 for the first time, was the first glimpse into an identity, single lineage of archaea that was more closely related to this archaeal ancestor of eukaryotes, and can therefore tell us something about their biology and their origin. So again, this was 2015. This is a very recent discovery, and the following years were tremendously informative in showing that Asgard archaea collectively, so this is low-key archaea and close relatives, were a pretty diverse group. And indeed, there were two publications from 2016 and 2017 that found at least three major groups related to low-key archaea, and that were called Thor archaea, Odin archaea, and Heimdall archaea, of course keeping with a very nice theme, and collectively called the Asgard archaea. And yeah, so phylogenetic studies showed that these Asgard archaea collectively seem to be the closest relative to eukaryotes, although the exact positioning of eukaryotes with respect to these was still very undefined. And of course, their genomes contained a lot of fascinating information. And indeed, they seem to contain many genes that were thought to be typically eukaryotic. These were related to several different functions. There were some genes related to N-glycosylation in eukaryotes that were present in Asgard archaea, as well as Escort machinery, for example, that has to do with membrane remodeling, ubiquitin system, cytoskeleton, or trafficking machinery. So these were somehow building blocks for typically eukaryotic functions that were already present in the Asgard archaea, although of course the discovery of these was only done bioinformatically, so it's not very clear what kind of functions these genes perform in Asgard archaea. Although in the last couple of years or so, there's been some very interesting experimental studies showing that at least for some of them, such as some cytoskeletal proteins, they do indeed seem to have similar characteristics as the eukaryotic homologs. But indeed, very little is known about what these genes do in Asgard archaea and what role they may have had into their origins. So yeah, not much is yet known about Asgard archaea, and one effort that we are currently undergoing to characterize them a bit better is a research project that we are carrying at Atacama's lab that I'm doing in collaboration. Well, it was originally led actually by Laura M. and Eva Cáceres, and we're working on this together currently, where we expand by quite a lot the set of genomes that was available for Asgard archaea from the original 11 from the 2017 paper, although there's been additional publications with more genomes in the last years. And then in total, we have now about 80 genomes that we are describing and characterizing with an evolutionary biology framework. So these 80 genomes, they are very diverse, and there's a lot of information that we are trying to extract from them. And several of them come from our lab, but there's been amazing work coming from some collaborators that were able not only to reconstruct a lot of genomes, but also find very interestingly diverse ones. So together, this forms a very nice data set to understand a bit better the genomic diversity existing in Asgard archaea. And indeed, we find a lot of genomes related to the groups that I presented previously. So here in the darker shade, you have the proportion of genomes that was known by 2017. And then this study expands by quite a lot, each one of those. But additionally, we have found several major groups within the Asgard archaea that we didn't really know about before. And we have called them Freya, Iden, Hel, and Balder archaea. Indeed, the Hel archaea were published last year in a Nature Communications paper. But indeed, together, these set of genomes has a lot of very interesting information that, it's taken a bit of time to analyze. And of course, part of this effort has to do with trying to reconstruct a trustworthy tree of archaea and eukaryotes and try to understand what the archaeal ancestor of eukaryotes look like. And something that we consistently see in our phylogenetic analysis is that most Asgard archaea seem to form a single monophyletic group, while there's a couple of groups, the Iden and the Heimdall archaea, that are closer related to eukaryotes. And in this group, we don't really know exactly what the actual topology is, whether eukaryotes are more related to one of these two groups or perhaps to both equally. But this topology of Asgard archaea into two major groups seems to be quite consistent in our studies using different gene sets and different phylogenetic methods. Yes, so there's a lot that has to be still uncovered about the Asgard archaea, several additional very interesting genes that we are analyzing currently, and I urge you to follow this this topic, and hopefully we will be able to wrap it up soon for publication. We'll see. I also wanted to mention that this growth of the tree of life is coming from many different places, and indeed at the bottom of the ocean, Loki, archaea, and the rest of the Asgards are far from being the only novel lineage. And other studies performed at a similar time in our lab from other samples from marine sediment. They also uncovered very unusual, very interesting lineages that raised a few eyebrows. And these had to do, these were related to the chlamydia. So chlamydia are well-known bacteria that infect mostly animals. Those are the ones that we know most about. There's also a lot of chlamydia that infect protists. And indeed, here you have a tree of chlamydia that we reconstructed recently. And in color, you see the chlamydia that were known before the study, and in orange, chlamydia that were newly described. So this was work that was mostly performed by Jenna Duramshi. She's a great PhD student in Theis's lab. And this was published earlier this year. So yes, indeed, Loki's castle also contained a huge diversity of chlamydia that didn't really seem to be related to the ones, they didn't seem to be the ones that we know about, but they expand by a lot the diversity of chlamydia as we understand it now. And indeed, for many of these, there's still a lot that we don't know. And there's indications that some may not even be host associated, which would really be a fascinating discovery and help us understand the evolution of pathogenicity and host association. And indeed, one of these groups, we have called it the Anoxychlamydiales, and they are extremely interesting. And in a follow-up study that was led by Jenna and also Courtney Stairs in our lab, so they have analyzed a group of genes from this Anoxychlamydiales that are homologous to some genes found in several microbial eukaryotes for anaerobic metabolism. So these chlamydia seem to be capable of anaerobic metabolism, something that was not really seen before. And here in this figure, you have pathways related to anaerobic metabolism and microbial eukaryotes, and the colors represent the identity of their ancestors, like what kind of organism they originate from in eukaryotes. So purple proteins here, the purple circles, and so yes, this protein seems to originate from alpha proteobacteria, so indicating that probably they come from the original mitochondrial ancestor. And then additionally, there's several genes that were found in this anoxyclamidiales, potentially pointing to a scenario where a chlamydial ancestor exported these genes, transferred them either to a eukaryote, bona fide eukaryote, and then transferred it to additional microbial eukaryotes, or potentially to the lineage present before the common ancestor of all eukaryotes, and then was inherited vertically, perhaps with some additional horizontal transfers. So this really comes to show that there's a lot of diversity that we don't know yet about, and they hold very interesting potential information for evolutionary scenarios. And in this case, this even raises the possibility that the origin of eukaryotes was more complex than previously thought, perhaps involving even more lineages than the two that we already know about. There's more work that we are doing to try to uncover potential origins from chlamydial genes into eukaryotic, into the eukaryotic domain. And hopefully there will be more information about this coming out soon. But indeed, yeah, there's a lot that we don't know about the tree of life. And it is mostly sequencing studies and follow-up experimental studies that are showing that the diversity that exists in our planet is not only tremendous and holds a lot of information for processes that we thought we more or less understood, and it really happens to be that there's a lot that we can still say about them. Right, so this was everything I wanted to present, and I really would like to thank everybody for your attention. Thanks.
Okay, very, very interesting talk. That was pre-recorded, so any questions, I will contact Daniel and try to ask him to answer them on social media. But for now, it is my pleasure to introduce our next live guest. And we're just going to do a quick tech test before we get into his presentation. But I'm here now with Fred Passman, and he's from the International Biodeterioration and Biodegradation Society. So how are we doing here, Fred? I think you're on mute. Can I unmute you here?
Myself. There we go. You can hear me
Great.
now?
I can hear you now.
And I just want to check, you see a full screen and you see a disorienting laser pointer.
Yes, yeah, I can see it in the center, yeah.
Excellent. Yeah. Okay, then I guess we're set to go.
Fantastic. Well, I'll hand straight over to you, Fred. Joe told me to say hello, and he said that I'll really enjoy your talk. So I'm really looking forward to it. The show is yours.
And to those who are listening and watching in, I'll wish you good morning, good afternoon, and good evening, wherever you may be. I thought this would be a great opportunity to talk about the dark side of industrial microbiology, when microbes damage fluids and the systems in which they are contained. And I've got it as the 16th of September because where I'm sitting in New Jersey, it's still the 16th. Now let's see if I can get my screen to advance. There we go. So over the course of the next few minutes, I want to share a few definitions. Although I confess I've been listening to the previous several presentations, and we're sharing a fairly common theme, so that's very helpful. Provide two examples of what I mean about biodeterioration, and then wrap up with several invitations to listeners. I think by this part of the evening or morning or afternoon, everybody understands that when we talk about microbiology as a discipline, we're talking about the study of organisms that are too small to be seen by the naked eye. This includes the single cell algae, the archaea, the bacteria, the yeasts and molds within the world of fungi, and protozoans. Within microbiology, we have the subdiscipline of microbial ecology, and this is the study of relationships and dynamics among microbes living within a particular environment, an ecosystem. Collectively, we refer to those microbes as the microbiome. Many of you may have heard of the Human Microbiome Project, recently completed, and giving us an insight to the number of tissues which previously we thought were sterile, and now we understand have their own microbiomes. Industrial microbiology is that branch of microbial ecology where we study the types of organisms either used for production. We use them to create antibiotics, various types of foods, specialty chemicals, and it includes the study of microorganisms that degrade materials. So we have within industrial microbiology both what we call biotechnology, using microbes as tools for manufacturing things, and the study of microorganisms that degrade materials. Of course, even biodegradation has two sides of a coin type story. When we want the microbes to do what they do naturally, we call it bioremediation. This is when we intentionally use microbes to do things such as composting, oil spill cleanup, other types of hazardous waste cleanup, basically helping to return tainted environments to their natural healthful state. When they're doing the same things, but we don't want them to be doing it, we call it biodeterioration. And a couple of examples of this are oil field souring. That's when we produce a lot of hydrogen sulfide in the petroleum formations, and that sulfide is both a health risk, but also causes tremendous damage to oil field equipment. Microbiologically influenced corrosion and other types of damage I will mention later in this presentation. So that's biodeterioration. The little photo on the right, this is, oops, now it's moving. Sorry. Okay, so my laser pointer went amok. This is an MIC hole where essentially the acids produced by microbes have eaten clean through the wall of a steel pipe. So biodegradation, it can be, if we want it to happen, good news, we call it bioremediation. If we don't want it to happen, bad news, we call it biodeterioration. And biodeterioration damage globally is estimated to cost several $100 billion US each year. A lot of that is in the petroleum sector, but also damage to buildings, artworks, and library archives. The list is as long as our imaginations will allow. So what we have here is what I call atherosclerosis of a transfer pipe in a metalworking fluid system. These are tiny metal particles that have been glued together by biofilm and have essentially blocked off this pipe. This pipe is about 20 centimeters in diameter. And as you can see, only about a third of it is still open for fluid to flow. So in the first case study, we'll talk about damage to emergency diesel generator systems. Diesel generators, well, they run remarkably on diesel fuel. Diesel fuel comes from bulk storage to day tanks that are close to the engines. Typically, the day tanks will have equipment to help keep that fuel clean and in good shape. Filter units, recirculating units, sometimes also water separators. That fuel is then supposed to go to a diesel engine. The diesel engine, when it operates, powers a generator. If the fuel doesn't get to the engine, the generator cannot be powered, and the consequences can be quite disastrous. And one of the most common causes of diesel system failure is plugged filters. And if your filter is plugged, the fuel doesn't make its way to the engine, and the effects can be quite dramatic, as we'll show in just another slide or two. Inside the tank, microbes grow and form biofilms. And here we have the inside of a fuel storage tank, a close-up of this mass of slime, and a photomicrograph of what a biofilm community looks like within that slime. Left unattended, you turn your tank into a colander. This tank was pulled out of the ground because it had just a few holes that had been drilled by microbes. Microbes, this mass that's growing on the walls, can also come free, floating in the fluid. And when that happens, you can then quickly slime over filters, and that's what prevents fuel from making it through the filters. The impact, as I mentioned, fuel starvation stops engines. In facilities like data centers, hospitals, hotels, it's estimated $2 million US per minute of emergency generator engines not operating. In nuclear power facilities, if you don't have an emergency generator operating when needed, you can have a Fukushima type event. Now, the Fukushima event occurred not because fuel wasn't making it to the generators. In that case, the generators were flooded, but the impact was the same. The failure of the emergency generator caused the meltdown in the reactor. Similarly, in turbine oils, people say there's no water in turbine oils, but these systems can quickly accumulate traces of water. And I think, as you've heard in other presentations today, it only takes a milliliter of water to provide what we would consider an ocean of fluid in which microbes can thrive. And so what happens is microbes will grow in discrete points along the recirculation system. And you see a schematic on the right of a recirculating turbine oil system. And when they grow, they cause damage to the oil. So here's a close-up of a system. Here's the oil reservoir. Reservoirs often will have a bottom drain. They call it a bottom drain, but my friends, the engineers, refuse to actually recognize what a true bottom is. And so they think, oh, 2-3 centimeters of water, that's a dry tank. And the microbes will give them many sleepless nights because they should have put these drains in the true bottom of the tank. When things are working well, you have nice, clear oil. This is what turbine oil looks like as it's in use. As water becomes dispersed in the oil, it gets hazy. You can't see through it anymore. And in the worst case scenario, we create, now don't try this at home and certainly don't eat it, something that's very similar to mayonnaise. It's an oil and water emulsion that really mucks up the works. Turbine oil meets a variety of specifications, most obvious of which, it's got to be clear. There's got to be no visible water or sediment. Its acidity, its acid number is got to be negligible. There's got to be a negligible measurable water concentration. It stays stable. We say that it resists oxidation. It prevents corrosion. So there are additives in this oil that help coat the metal surfaces of the system so that the oil will not allow those parts to become corroded. And its ability to flow. is within very tight parameters at both 40 degrees Celsius and 100 degrees Celsius. The combination of the two we call viscosity index. Biodeterioration affects all these properties. The acid number goes up. Clearly, we're getting visible water accumulation. Were we to measure this water, it would be increasingly turbid, and its ability to resist oxidation degrades very quickly, and we start seeing corrosion in the system. When our turbine oil conditioning system is working well, the oil that's coming back to the reservoir first goes through a preliminary filter, or in this case, a screen, and then a polishing filter, and sometimes even a chill unit with a water stripping filter. And you wind up with nice, clean turbine oil. If those filters become plugged, or if these filters no longer, especially the water removing filter we call coalescer, can no longer do its job, instead of this, we get this or this. Microbes produce acid, and that contributes to the loss of oxidative stability. It also contributes to the corrosiveness and the acid number of the fluid. When we talk about corrosiveness, this is the inside of a transfer line that's been cut open, and you see what looks like a mountain range inside. These are corrosion tubercles. Microbes are growing under these, and we have alternate layers of various iron and sulfur molecules, iron sulfide, and microbes, biofilms, produce acids, and they will eat right through this pipe. The other thing microbes do is they produce surfactants. You're all familiar with surfactants. Anytime you mix a detergent with water, you get a water, you get an oil in water droplet we call a micelle. And in oils, we have the opposite phenomenon where these little red dots here, little blue dots here, have a charge. They're polar. The little squiggly tails are hydrocarbons. They're nonpolar. They do not have a charge. And so the hydrocarbons are soluble in the oil, and the polar heads provide a capsule that traps little droplets of water. And as you see, the more droplets we get, the more it looks like mayonnaise. Finally, we produce slime, biomass. This mat, this is all biofilm, can be as tough as a canvas sheet. It really is well held together. I don't know if you'd want to use it to make a tent. The odor can be a little bit interesting. Not only does it face off filters, but as you can see in the inside here, it's also corroding the core of the filter, causing that to become damaged as well. When filters plug, no oil gets to the bearings. When there's no oil to the bearings fail. If the bearings fail, the power generating turbine fails, and that's disastrous. So microbes in these systems produce acids, surfactants, slime. They do other things. But again, if the lights go out because a power generation system has failed due to turbine oil problems, this will cost billions per minute in a large metropolitan city. Not only that, but it will increase health and safety risks. We don't understand really what's going on yet. We know vaguely that if we have high bioburdens, lots of microbes present, chances of damage occurring increase. But we've seen, we being the microbiologists, the very few microbiologists that investigate these types of systems, have found that if we test, we'll sometimes see very high bioburdens, but no evidence that they're doing any harm. Other times we'll get much lower bioburdens, but see considerable evidence of their doing damage. So we're just beginning to get a sense for how little we know and developing the tools for learning more. I've given two examples in the sectors where I'm personally most active. Aviation industry. Microbial contamination of fuels is a major concern. You certainly don't want to have the fuel line plugged up when you're up at 50,000 feet. It can be embarrassing to everybody aboard the aircraft. Similarly, if you're out in the open ocean and your fuel filters plug up and you can't get fuel to drive your ship, that can be an expensive proposition. If fuel or crude oil is your cargo, microbes typically cause about 10% of the total value of that cargo is lost due to the impacts of microbes just growing in those fuel tanks during transport. Ditto for surface transportation. And I mentioned earlier in the presentation, metalworking fluids are typically about 5% of a chemical mixture diluted in water, perfect environment for microbes, lots of aeration, and not only can it damage the production process, microbes can present a significant health risk to machinists exposed to this environment. I focused on the industrial side, but I'd be remiss if I didn't mention there's a lot of biodeterioration going on buildings, in books and documents, and a whole range of art objects. All these are areas where we still have tools to develop and need a lot more work done to develop better strategies for reducing the risk of and for remediating when biodeterioration does occur. So I am a member of a society, International Biodeterioration and Biodegradation Society, and our members cover the gamut across all areas of biodeterioration and also bioremediation or biodegradation. And I invite those listening to this presentation to check out our website and consider becoming contributing members of our society. We are a member society of Fems, of course, and we try to do our best to support Fems activity. Our next international symposium will be held at Montana State University, home of the Biofilm Center for Biofilm Engineering. A year from now, almost exactly a year from now, and you can get more information about that conference at ibbsonline.org/meetings. Finally, for those of you listening who might be undergraduates or graduate students, we offer bursaries of up to £1,000 for undergraduates and graduate students to help them fund their research. More information can be found at this link. So I'm inviting you to join, I'm inviting you to attend, and I'm inviting those of you who are undergraduate or graduate students to apply, take advantages of what we have to offer. And with that, I'll just quickly introduce myself. I've been at this for, as my British colleagues say, donkey's years. I first started mucking around underground cave streams in 1969. I was a budding marine microbiologist when some oil field folks asked me to help them out, and I've been looking at biodeterioration control since 1979. And with that, if there are any questions, I'll be glad to entertain them.
Thank you very much, Fred. That was really interesting. Some of the examples and the photos you shared, I mean, I have a microbiology background as well, but, you know, these filters that were completely caked and covered in this slime, I mean, you just had so many examples of it. I mean, how serious of a problem is it? Because it's something that I haven't really considered so much before, especially in fuel.
And I will say, Ben, this is one of the things that inspired me to give this evening's presentation. So few people are even aware that microbes can live in these nominally water-free systems. And one of the slides I've probably gotten more use of than any single slide in my fairly large library, I have a person standing next to Mount Kilimanjaro. And I say, if you turn Mount Kilimanjaro upside down, hollowed it out and filled it with water, that would be like a bacterium on a surface underneath a 2 millimeters of water. And so immeasurably small traces of water in systems create habitats where microbes can thrive. And that also makes it a challenge to collect samples. I advocate, the people I talk to are mostly engineers, and they're used to collecting representative samples. And I say, no, when we're doing microbiology, we need to get diagnostic samples. And the samples that make you what your turbine oil quality is, or your fuel quality is, or your water quality is, are not adequate for determining whether or not your system is at risk. And we're really looking for, young researchers to get involved in this less apparent and perhaps less glamorous side of microbial ecology than those who are doing the wonderful work in industrial microbiology and molecular biology in terms of using microbes to produce things.
Yeah, I mean, it's fascinating. And I'm wondering about some of the, because you talked about solutions that there might be in the future, and kind of early on in the talk, you kind of alluded to the fact that possibly just having some kind of microbiology input at some of the design phases, like you're talking about this, the designs that didn't allow all of the water to kind of clear from a system. What do you see as some of the solutions, maybe in the very near term, trying to fix some of the big problems they have now, and hopefully, a few years in the future with some more technological advances?
To a certain extent, as you already hinted, it's engineering. And if every engineer could have a semester course in microbiology, especially microbial ecology, that would create a sensitivity. Just the word seems to have more syllables than many folks can handle. And so I tend to speak in terms of bugs rather than using the full word microbiology. And of course, you can see our association, well, we just call it IBBS because international biodeterioration and biodegradation is quite a mouthful. The other thing is on the regulatory side, the chemicals that have been traditionally used to control microbial contamination in cooling tower systems, in personal care products, in paints and coatings, in fuels and lubricants, they're coming under regulatory pressure, some for good reasons, some for non-technical reasons. And so we have to develop strategies that perhaps we haven't really given much consideration. There are days when I wish I was 50 years younger so that I could be where I was back in the late 1960s, early 1970s, with the tools we now have available that we didn't have then. And the last presentation, I don't know how many were online for it, but it was wonderful discussion just the past 2-3 years, what we've learned about the archaea. And there's still very few reports of archaea in these systems, but I believe with a perfect faith that essentially we haven't learned how to find them. We haven't learned how to detect them. I recall when we were still calling them archaebacteria and thought that they only existed under extreme environments, 200 atmospheres pressure, 120 degrees Celsius, temperatures. And over the course of the first decade and a half after the recovery, the more we looked, the more we found. And we understand now that they're pretty much ubiquitous.
Yeah, indeed. Yeah. And do you think microbiologists have enough access? So I think sometimes the problem is the kind of the connection and the dialogue that can happen between two different disciplines, such as engineering and microbiology, and they're of course very closely linked in this case. But do you think there's enough access? Because when you talk about some of the experiments that are going on in other fields, you know that there are teams of microbiologists working on lots and lots of different cases and lots of different samples. Is this happening in this field? Do you think the microbiologists are getting enough access to the systems and the samples?
Not enough. There was perhaps a golden era in the 40s and 50s where many of the petroleum companies had microbiology teams. By the 70s, the microbiologists were fewer and farther between, and these days you'll very rarely find a microbiologist at a petroleum company, as an example. And so the important multidisciplinary communications that are needed just aren't happening. And even within microbiology, we're such a large discipline, and you go to an American Society of Microbiology conference, and the microbial ecologists maybe represent 5% of the attendees. And within that group, many of them are not really focused on industrial types of problems. They're doing wonderful work at marine microbiology, aquatic microbiology, things like that. So we really need to get more people engaged in just wash my tongue and I can't do a thing with it. Interdisciplinary studies that look at biodeterioration of buildings, artworks. There's a lot of materials science, there's a lot of engineering, there's a lot of chemistry, and there's a lot of microbiology. I like to quote Donald Rumsfeld, a former Secretary of State, and one thing that he said that I agreed with: we have our known knowns, the things we know, the known unknowns, the things we know that we don't know. But in microbiology, I think we are really have a large backlog of unknown unknowns. We don't even know how to ask the right questions yet. And it's wonderful being a member of a young science like microbiology.
Yeah, right. I mean, it's exciting, isn't it? I mean, there's so much to discover. I'm just looking through social media now to see if we have any questions. I've got one from Facebook. Mohit is asking, what kind of microbes most commonly cause the biofilm formation in water tanks?
So, a lot of work has been done with Pseudomonas aeruginosa. However, as we get smarter at using genomic tests, I think the answer to that question will change. If you're looking for a particular organism, if you have traditionally been focused on culture methods, historically, Pseudomonas has been one of the easiest to recover from surface swab type samples. I would be reticent to say that's the most common microbe in biofilms. There's a tremendous amount of complexity. And again, I think we're still developing effective methods for parsing out what's present. It's a terrific question. I hope you'll follow up and do some research to provide us with some answers.
Yeah, I hope so too. I was wondering in your presentation, the previous presentation from Benjamin, how much kind of media coverage affects kind of research and what's going on in the field. I mean, the two examples that spring to mind. So when you talk about Fukushima and these kind of huge disasters, and I also think about a few years ago, I think there was this some kind of biofilm on one of the monuments. It might have been the Washington Monument or something like that. And that suddenly somehow got a lot of press coverage, the fact that there was this thing growing on one of these treasured monuments in the US. Do you see any impact of that kind of discussion or media coverage on research and investment?
in most of the areas where I am directly involved, people would prefer there not be press coverage. My poster child for that is a certain petroleum company who 1 year had a pipeline failure that set them back about $5 billion. And that was because they had, quote, saved money on manual testing. They were using a very smart device that went through pipelines and could detect everything, except it couldn't detect MIC. And so they had this one failure event, spent a tremendous amount of money on public relations about how it would never happen again. And then 2 years later, they had a much smaller event that only set them back $2 billion. Now, you know, as a poor microbiologist, when I think of $7 billion and what I could do with that in terms of condition monitoring, research, and improving preventive processes, it boggles my mind. But over and over again, you see corporations with that kind of funding and with those sorts of problems pretend that an event is a black swan, it'll never happen again.
Wow.
And it's very frustrating.
Yeah, it kind of it's kind of mind-boggling that I mean, because those sums of money, I mean, they're not sums that I'm used to kind of dealing with. You'd think it would be worth looking into.
Yeah, we had a senator, late great Strom Thurmond, used to say $1 billion here, $1 billion there, and before you know it, you're talking about real money. And it really, I find that the return on investment can be in the millions of dollars per year for even relatively small operators. And because they aren't educated in microbiology, they are disinclined to invest, or I say in the petroleum retail side, where, again, the simple filter plugging issue can save a individual forecourt operator as much as $1 million a year per dispenser on their site. So you go up to a modern forecourt, and I think even in you're in the Netherlands, so I think even there you have forecourts that have anywhere from 12 to 16 dispensers. And if each dispenser is experiencing opportunity costs, I'm not talking about damage, I'm just talking about because you're restricting the flow of fuel through the dispenser, $500,000. And you've got, let's say you've got 10 dispensers. So that's $5 million per year that you don't need to be spending if you would only do a better job of condition monitoring and contamination control. And for 40 years, I've been trying to convince operators to do that. And very few have actually even tested my model, let alone taken my recommendations.
Wow.
But hope springs eternal.
Yeah.
If you were going to spend, $50,000 in order to save $1 million, how much thought would you give to that?
Right.
And I've found it, and I think part of the issue is there are fewer than a dozen people seriously studying fuel microbiology in the world. We need more people involved, and as we get more people involved, then it'll be easier to communicate to the non-microbiologists.
Sounds like a good call to action, call to arms. We need more research.
Certainly.
Do you think? Is there any problem with the kind of the fuel that's going into the cars? Then you saw about the forecourts. Does it affect the cars at all? The consumers
When
have
the
any?
fuel is degraded, yes. Most often, some years ago, I tried to talk to the manufacturer, but they wouldn't talk to me. But a local repair shop told me that their supplier of aftermarket passenger vehicle fuel tanks, their sales were up 200% over the previous decade. So as, again, fuel becomes more severely refined, when we go to the biofuels tend to be more readily biodegraded and more susceptible. They hold more water, therefore they provide more easily provide a suitable environment for microbes to grow in the tanks and cause fuel tank deterioration.
Right.
So it's the sort of stuff that happens all the time, but it's the analogy I use is I've worn glasses most of my life, but if I hadn't, I wouldn't know that I had a vision problem. It's only after you get the corrective lenses you begin to realize what you've been missing, and I feel that's the situation we are when it comes to biodeterioration. People are used to spending sums of money because of the damage caused by microbes without even realizing that's why they're spending that money. And until we get a critical threshold of people that recognize the issue and take preventive measures, it's a Gibbs free energy constant we have to overcome, and that's why our outreach for IBBS is to help make more people aware of the dark side of microbial ecology.
Yeah. Great. Well, I think coming back to that is the perfect place to leave it. Thank you so much for your presentation, the discussion. See, there's some contact details on this slide here. Is that the best place for people
Absolutely.
to
Just
get in
go to
touch
the
with
website, and my email address is at the bottom of the slide. It's also available from the website. And you know, shameful promotion, self-promotion, I do have a fairly long blog series, mini articles about various aspects of microbial problems in a variety of industrial systems.
Oh, great. Great. Then, yeah, everyone go and check those out. And thank you very much, Fred, again. Yeah, we're going to take a short 20-minute break now, but we'll be back in 20 minutes with another live presentation. So stay tuned.
Good. And thank you, Ben. Great questions.
No problem.
Bye-bye.
Thank you very much. We'll speak soon. Bye now. Okay, so I'm going to put the title slide on here because we're only taking a very very short break, and I will be back in 20 minutes. Okay. Hello there, Jidong. Can you hear me okay? And can you unmute your microphone? I can't hear you just yet. Is there a unmute button on your side?
Let's see. How about now?
Yes, there we go. I can hear you perfectly now.
Good. Yes.
Fantastic. How are you?
I'm fine.
Great. Well, we're back from the break on the live stream for International Microorganism Day. It's a pleasure to be here with you just now.
Thank you.
Yes, we've got a couple of minutes before the hour when we start. So I just tell people a little bit who may have joined after the break about the live stream and kind of what we're doing for International Microorganism Day. So we're streaming this presentation and lots of other presentations live on social media for the entire day of the 17th of September, because it's International Microorganism Day. It's a day when we can celebrate and geek out and try and tell people about the importance of microorganisms. So we have some pre-recorded talks and we have some live talks, and we are definitely here in person live. So if anyone has any questions, I'd encourage them to ask away on social media wherever they are watching this presentation, and I will try and get the questions to you if we have time at the end of the talk, but if not, we'll try and answer them on social media itself.
Great.
And so without further ado, I would like to hand over to you, Professor Jidong Gu, Professor at Guangdong. I'm sorry if I'm going to murder this pronunciation here, Guangdong Technion Israel Institute of Technology.
Yeah.
And you're going to talk to us about microorganisms and world cultural heritage. It's a pleasure to have you here. Please take it away. The floor is yours.
Thank you very much.
No problem.
My name is Jidong Gu. I'm working at Guangdong Technion, Israel Institute of Technology in mainland China. And today I'm going to present a talk on microorganism in line of International Microorganism Day, and also the relation to World Cultural Heritage. And this work has been a long time accumulation of research evidence with Japanese colleague Yoko Katayama, and that's why I put her picture over here, because we have collaborated more than 15 years on this project. So to start with, I'm going to give you an outline what I'm going to talk about in this 15-20 minutes. I'm going to introduce briefly about the subject of cultural heritage and biodeterioration. And I'm going to focus more on stone material as the material of cultural heritage here. And I then going to look at the microbial processes which are affecting the integrity of the stone material, which is cultural heritage. And then several specific processes which are damaging the sandstone. So basically, that's a combination of science and also tune down of some of the information to a better understanding, I hope. So to start with, when we talk about cultural heritage, especially in Southeast Asia, Angkor Wat is probably one of the most famous cultural heritage. And it's so famous. And over here, you can see it's Angkor Wat. And in front of this small pond with reflection, magnificent. And it's also being shown on the national flag of Cambodia. This cultural heritage, if you get closer to it, is will be able to show to you that there is a constant maintenance, repairing of the cultural heritage because it's old, about 1,000 years
history, and it has some maintenance work all the time. And this particular cultural heritage has been listed as World Cultural Heritage. Has about 18 years of history. It's not that long. 2 years ago, we had a celebration of 25th years of cultural heritage for this Cambodia site, which was held in Angkor Wat in Cambodia. So if we look into the importance, historical importance of Angkor Wat, it's really the culture, humanity, the art. And this is one part of the Angkor Wat. If you go there, look at the bas relief carving. The carving is on sandstone, and the carving on this stretch is 400 meters. It is an enclosure of 4 sides. So basically, if you walk around 400 meters in the east, 400 meters in the west, and if you go around it, it's 1,600 meters of bas relief. And on this carving is recorded history of Cambodia, which is a Khmer in the past. So it has a great importance of humanity, of artistic presentation, and also the humanity of that particular culture. But now I'm shown to you at the early part was about Angkor Wat. Angkor Wat has been the international image for Cambodia, but actually closer to the Angkor Wat, there is an Angkor Thom. What does this Thom mean is this is an Angkor city, because Angkor Wat is a temple, but Angkor Thom is a city. The city was a real live city. The king was living in it. There were also people living in it. But Angkor Thom has not get so much importance and attraction as Angkor Wat. So I want to show you some information about Angkor Thom. And this is going into Angkor Thom, the city in the south side, south gate. If you look at on the left-hand side and also similarly on the right-hand side, which you can't see, they are statue of guard, and they are the warrior guard. But below it is canal, water. So the water was and actually is still surrounded the city, but on top of the river, the canal, there is a gate. So we can walk and we can go into it. And within the city, it is where the life and the king would be. So if we go into one of the temple in Angkor Thom, this is called the Biofilm temple. And for the Biofilm temple, you can see a little bit, but actually this is the central tower. And if you look at the appearance of the sandstone architect, with all different kind of microbe, that's the color about. But if you look at this particular stretch of section, it's a gallery. The wall are all being carved over here. And you can also see over here, this particular section of the stone column, the top part was missing. So immediately, this is in the 1960s, when the French discovered Angkor Wat again. They use concrete to make an emergency protection and conservation of this part of the gallery. But if you look at a little bit to the left here, you can see here, this section of the tower, the top part, was missing. Missing in this part was missing. So this particular gallery, this section is completely exposed. So the carving are exposed. So similarly over here, this part of the tower is also missing. So what I'm trying to show here is the temple has a long history and is also in a damaging state, being destructed. And when we look at the magnificent part of the Biofilm temple, it is on top of the tower. On every tower, there are 4 large faces of the are each to the cardinal direction, and each of them are unique. There is no duplicate. Every one of them are unique. This gives a major attraction of the biome temple to tourists. So what we do, when we started working on protection of Angkor Wat and Angkor Thom and also other temple, we can go to the temple and do sampling. This is something I'm going to talk about in terms of technically how we did it. After we get our sample, then we bring back the sample to the lab to analyze what are the microorganisms associated with this brick red color, this dark color, and here is a brownish color. By doing that analysis, we would be able to know what are the microbes are living here on the surface to cause this discoloration of the bas-relief carving on the temple. Something I want to briefly mention here is I'm not going to be able to go to great detail is what in the protection of the temple. So in another way, if we can limit the amount of natural water, rainwater to the temple, then Angkor Wat would be able to be protected for a much, much longer time. The second factor in this whole process, this is something I'm going to be into more detail, is the biochemical reaction of the microbe, which are destroying the temple. They contribute to the damage of the temple we are seeing today. And this is the second part. And the third part is a summary, which has just been come out this week, actually. This is a publication we have wrote, and this is summary of what is happening on Angkor Wat, on the sandstone. If you are interested in, you can get the article, and then you can read some of the information in more detail. So the first step is to collecting the sample, which would not destroy or damaging the sandstone of the temple. So the technique of sampling. This is a technique which has been invented, and which has an adhesive material inside, but the whole sampling device is sterile without any microbe. So what we can do is we can open this device, let the adhesive part to adhere on the surface of the sandstone, so any sandstone. And then if you look at the illustration on the right, which would be a cross-section of this sampling device, push onto the biofilm, the microorganism, then we can peel it off. This is the first sample. Afterward, we can take a fresh new device, push it onto the biofilm again, we take the second sample. So in theory, we can take the third one, we can also take the fourth one. And basically, on this particular site, you can take as many as you want for the sample. And after taking the sample, each of them going to be in this device, we take back into the laboratory, we can extract the DNA of the microorganism, amplify them by PCR technique. Afterward, we can manipulate basically to reveal this particular sample. This is over the depth of the sample. So initially, this is the sample we take, and then we analyze. And here is look at one particular site over here. We take 36 samples, and over time, we will be able to analyze the DNA in the sample. We see dynamic change of different microbes over the depth, because the first one is the outer surface, and when we go into higher and higher number, that means into the deeper of the biofilm, you can see that cyanobacteria was high in the outer surface. When we get deeper, their number are get lower. But the facultative microorganisms, which do not like oxygen, they become higher and higher in the lower layer. So we are able to get the spatial information of the microorganism distribution in this particular site. and on the sandstone. This is one of the techniques. So similarly, we can also get the whole community composition of the different microorganisms. This is at one particular site of biofilm 42. And by doing problem in the community, but there is also some problem. Which one are actually damaging the sandstone? This next generation sequencing does not tell us. We need other technique. So let's look at one of the example is sulfur. This is at Angkor Wat. You can see on the back part is the bas relief, and here is one of the column in the gallery, and the lower part has been damaged. So what we did was, this is the Yoko Katayama. We took a sample on the top without any damage. We took a sample at the intersection between damage and intact. And then we take sample at the serious damaged part. So there is A, B, and C. So for the 3 samples we took, we incubate them in the culture media for sulfur oxidizing microorganism. What we see is within 5 days, C, which is the very, very lower part, they would have a pH drop of 1.2. This means production of high acidity, a lot of acid being produced. But for the B and A, even after incubated for more than 2 months, the pH drop was minimal, very little, particularly for the top part, there is no pH drop. So this indicating sulfur transformation are involved in the damaging of the sandstone, producing acidity. So similarly, if we cultivating the microorganism, this is the bacteria we isolated. You can see over here, if we put sulfur in, the microorganisms grow very well. If we do not put sulfur in, they do not grow that well. But when they grow very well, they cause a pH drop. This is the pH drop. And here is the control. The pH doesn't really change that much. And similarly, if we monitor another group of microorganisms, which are fungi, Angkor Wat, Angkor Thom, and also non-chrome, which are 3 different sites, you can see that the fungal, which can oxidize sulfur, their population are between 100 to 200 per gram of material. But this is a culturable fungi, which are much, much less than the total number of fungi, but we found them on the temple. And here is also an illustration in the laboratory condition. We can see that the microorganism can utilize the sulfur in the agar plate, causing a clearing zone. If we analyze what is over here, the C section, we found they are thiosulfate and sulfate. So those are the products from transformation of the sulfur put onto the agar plate. This is a complicated process, but I'm not going further because of the time. I'm only going to touch onto another process. What we have found recently is nitrogen transformation is also evident. What we found was when we monitoring what is in the moisture or the water of the sandstone, we found there are higher concentration of nitrate, but not ammonia or nitrite. This leads us to monitoring what is happening here. So this is related to the nitrogen cycle. I'm going to jump quicker a little bit to save my time. So what we did was we take sample at the ground level, in the gallery level, and also the higher level of the biome temple. We analyze what are the microorganisms in it. So on the ground level, gallery level, and the central tower, we see much, much higher concentration of a microorganism called ammonia oxidizing archaea, much, much higher than bacteria. What does that mean? Both of them are contribute to nitrate and also acidity, but archaea concentration are much, much higher. That means archaea are the driver of this reaction. We know what are the microorganism name, bacteria name, and we repeat this particular experiment in the following year at 3 different temples. We found similar result. That means archaea are much, much higher than bacteria. And we also carry out a simulation experiment, and we carry 36 liters of deionized water onto the top part of this gallery. Sorry. And then we simulating rain water. So let the water to drip down and collecting the dripping down water and then analyze. We found there are ammonia, nitrate, there are also a lot of sulfate in it. So those are the iron which has been leached out from the sandstone to confirm what we have observed previously. And the last piece I want to show to you is we use another technique, which is called isotope technique. This allows us to differentiate what are the two processes contribute to the removal of nitrate. By doing it this way, I'm not going into the technical detail, but I want to show to you is there is a very active ammonia oxidation to produce the nitrate removal by denitrification and the animal process was very, very weak because I use a small arrow here. And because of that, the nitrates are being accumulated because they are not being removed very quickly by the two processes. This is what we found on Angkor Wat, which result in accumulation of nitrate on the sandstone. So basically, if I conclude, I would say that colonization of microorganisms on the surface of a sandstone monument is a common phenomenon. We cannot prevent this to occur as long as the sandstone monument is in the open environment. When water is involved, particularly in tropical environment, microorganisms going to grow very actively, and the damage to the sandstone by microorganisms would be higher. The damaging process in this particular case we presented involve sulfur oxidation, ammonia oxidation, and in the ammonia oxidation process, it is the ammonia oxidizing archaea play a much, much more important role than ammonia oxidation bacteria. So to understand the damaging process causing biodeterioration of the sandstone, both biology are important, which providing the basis for our understanding of the process involved and also providing the information for management of the Angkor Wat in the long run. Because my time is running up, I would conclude that this project has been supported by Japanese trust fund for a long time, more than 15 years, and recently has also been supported by the Research Grant Council of Hong Kong government. So with that, I would just stop here, and if there are any questions, I would try to entertain any question, and that's the end of my talk. Thank you very much.
Thank you so much, Professor Ji Donggu. Unfortunately, we don't have time for questions just now, but if there's any, is there any way for someone to reach you to ask questions later on?
Yeah, sure. Because on my first slide, I show my email, and anyone, if they have questions, they can contact me through the email. That would be one way. To my email or through any means, because for the Zoom, I would also afterward monitoring if there are any questions sent to this particularly event.
Perfect. Well, thank you very, very much. And yeah, we'll be in touch.
Great. Thank you. Yeah.
Okay.
Okay.
So
yeah.
Bye now.
Yeah,
it's my pleasure to introduce the next talk, which is a pre-recorded talk, and this will be from Portugal, from the PCR podcast, talking about human pathogens and future challenges. So let me just find it there.
We are from the Portuguese Science Communication podcast, PCR, and we are here to celebrate the International Microorganism Day with a special edition of our program.
We would like to thank Fems for the opportunity to participate in this event, and to do so, we brought you a very special guest.
It is only natural to celebrate this day with a conversation with a researcher that studies microorganisms, so we are very happy to talk with Raquel Salião, group leader of the Molecular Microbiology of Human Pathogens Lab at ITQB Nova here in Portugal. Thank you, Raquel, for accepting our invitation to celebrate the International Microorganism Day and talk with us for a bit.
So thank you very much, José, Felipe, and Ricardo. It's really my pleasure, and it's an honor to be able to participate in this interview.
Okay, let's get started with the first question, and I will get right to the focus of this talk. And the first question is, what is the focus of your research at ITQB?
Okay, so at ITQB, my group is very much focused on a particular microorganism, a bacterium, that is named Staphylococcus pneumonia, or pneumococcus for short. And as the name suggests, it's a main cause of pneumonia, but it can also cause otitis media or meningitis. So a different spectrum of disease, from less severe to very, very severe and even fatal. And worldwide, the morbidity and mortality associated with this bacterium is very important. Having said this, what we focus in our group is not on the disease, but on the asymptomatic colonization. And so this bacterium lives with all of us in the upper respiratory tract, so more or less here, and most of us will be colonized several times over our life without having any symptoms. And we focus on this step on colonization because colonization is essential for three things. For transmission between hosts. If you are not colonized, you will not transmit. And the things I'm telling you can just think about SARS-CoV-2 because it will be the same. And nowadays people are very familiar with that. So if you are not colonized, you don't transmit. If you are not colonized, you don't get disease. And if you are not colonized, the bacterium in our case, the pneumococcus, will not evolve, right? So we divide our research in two main things. One is very related to public health policy. There's a vaccine nowadays that targets a subset of these pneumococcus, not all of them, but a few of them, okay? And what we do in our lab is to do field work. We go into the community, we get swabs from people, exactly like you do for SARS-CoV-2 when we are trying to test people for the presence of COVID-19. And then we look at those swabs and we characterize the bacterium that's in there. And based on that, coupled with epidemiological data, vaccine use and demographic data, we can understand what the vaccine is doing on the bug and ultimately on disease. And you can also make the parallel with COVID-19. We all know by now that what happens in Portugal is not necessarily identical to what happens in Asia, or in the United States, or in Africa. So local epidemiology is very important. And we get data from Portugal, and we share it among the Portuguese society. So that's one of the main focus. The other focus is a bit more basic research. Nowadays, we know that we have not only pneumococcus in the upper respiratory tract, but several other microorganisms which are part of the microbiota. And if we understand how the pneumococcus interacts with other microbiota, we can use that information to find alternative strategies to prevent colonization. And so if we prevent colonization, we will prevent transmission, disease, and evolution, right? And so that's the other second line of research that we carry out in the group.
Okay, so picking your second line of research, I would like to ask you why it's important to study microorganisms as pathogens.
Well, I think that touches all of us, right? So when we think about pathogens, I think most of us will think about human pathogens, but it could be plant pathogens, animal pathogens, you name it, right? But pathogens, as the word suggests, it's going to affect our health state. And it's the main goal all societies, I mean, to live, we would like to live as much as possible, but in a healthy state. So by studying pathogens, of course, we try to understand how can we tackle those pathogens, how can we treat the infections. But here I'd like really to make a focus. To treat is good if you can, to prevent is even better, right? So if you can prevent disease, that's very important. So I think that's why me and many of us study pathogens.
So you've mentioned SARS-CoV-2, right? And that's the main reason why we can't do this interview like face-to-face. We have to use this new way of doing conversation. But since you mentioned it, I think we all agree that people now are more aware of the effects that the pathogens can have to us as a society. And by pathogens, I mean either bacteria or virus. But do you think now is a good time for us to, as a society, discuss changes in public health policies? Do you think people in governments are more open to it?
Well, definitely. And definitely, I think so. And when we think, I mean, and here the focus, when you think about public health policies, it really means, in a very broad sense, anything that will affect health as a whole. So it could be obesity, depending on society, right? But now we are very much focused on infectious diseases. And I think this is what we are going through. It's definitely something very unique and extraordinary that will remain with us for our entire life. I mean, the experience that we are going through, not the pandemic. So I think this pandemic, as probably many of you are aware of, had been predicted for many times, because we had pandemics in the past, and they come from time to time, and we'll have pandemics in the future. And actually, in the WHO website, for example, for a long time, you would go there and you'd have the major threats, and you'd have the agent X. Nobody knew what was going to be Agent X. It was going probably to be a virus, but we didn't know which virus or which microorganism at all, and when it would come, and from where it would come. But it was predicted it would come, and so I think definitely what we are going through is having a tremendous impact in our lives, not only in public health, but in all our society. And I hope that a few things regarding infectious diseases and infection control will remain in our society, because these are some of those things we knew them for a long time, and they are very important. And one thing that maybe two key messages I'd like to convey when we talk about infection control is that washing hands, for example, this has been known for decades. That is the simplest, the cheapest, and the most effective way of preventing infectious diseases in general. And we know that, and that is true not only for hospitals, but also for infections going on in hospitals, but also in society. And although we know that for many, many decades, we still have a long way to go before population as a whole washes hands as frequently as needed. And the other thing that I think it's also very interesting is that the use of masks that we now all use, social masks or surgical masks, we know that, for instance, in Asian countries, this is a custom for many years. When we travel, we see very often Asian people with masks, and we don't know if they are protecting ourselves from potential infections they have, or if it's the other way around. But they used to use it. And this has been known also for decades, for over a century, that surgical masks or masks in face masks will help us protect transmission of infectious diseases, particularly respiratory infections in general. It's not only about SARS-CoV, it can be a common flu, it can be influenza, anything. And so this is also a habit that probably we should keep for the future, and would also help us prevent infections. So one thing that I think it's an opportunity here, and that became very clear at the beginning, is that we really need to invest a lot in education. Education about microbiology, I think, in general, for the population, for our students, for our politicians and so on, has increased a lot very recently. but we should definitely invest on that. I think that's very important.
Yeah, worst case scenario, we'll learn from this and move forward with many other cases that may come in the future. I guess that's the main thing.
Mm-hmm.
Okay, so last year your lab participated in a simulation of an hospital outbreak, and you managed to identify the threat in record time. And this was last November. Am I correct? So fast forward to the present day, we have the coronavirus pandemic, and it's a global outbreak. Although it is a virus, and your simulation was from bacteria, but can you tell us what kind of differences did you find in when comparing the simulation, the controlled simulation of the hospital outbreak to this global outbreak of coronavirus?
So what we did was very specific, because we are not the infection control team, right? We were assisting them in our simulation. We were assisting them in identifying the source of the outbreak and tell them in record time what was it, so that they can do their work. And so in the simulated outbreak, what we had were several samples. Some had been collected from patients, and suddenly a particular pathogen which was multi-drug resistant, was being frequently isolated from different patients in different rooms. And this was unusual, and immediately this rang the bell that maybe something was going on and this pathogen was being transmitted from patient to patient. And if that was true, what was the source of it? Who was transmitting it, or what was transmitting it? And so what we did as well was to get, in this case, environmental samples. So we got samples from the knobs of the doors, from sinks, and so on. And what we had to do was to extract total DNA, total genetic material, and compare it in record time and see how close was this DNA from the different samples. And we could identify in this simulated outbreak that environment, in this case, it was the environment, was playing a role, in the sense that, for instance, a patient would be admitted to a particular room, and the room was contaminated in the sink. So when people were washing hands, aerosols would form, and in the end of washing hands, you might end up with your hands contaminated, and then auto-inoculate yourself and get the infection. And so what we did was, in record time, to obtain this information and give a call to our infection control specialist and tell, this is not a coincidence. All these bacteria have a common ancestor and they are being transmitted. And that's the key information they need to decide how they are going to tackle the problem. Because if it's a single source, now they will have to control that source and prevent dissemination. And that's very different from imagine that and every single patient is coming with a bacteria that is close to another, but they are all different. Then you'd have to tackle in a different way with this problem. How does this compare to the global pandemic? Well, when you look at the global pandemic, this, which is a huge problem in Portuguese hospitals and hospitals worldwide, the control of nosocomial infections, seems simple, I would say, in a way, in the sense that it's confined to a hospital, and you have a physical confinement, and so it's very difficult to deal with. But compared to the SARS-CoV-2, I think it's much simpler, of course.
So now we have one question that usually we do in our podcast, where you have to choose one option. And you have already talked about the main lines of the research from your group. But now with all these pandemic problem, we know that you are also doing some job related with COVID diagnostics. So our question is, it is harder to deal with COVID samples or with PhD students that did not have their coffee?
I don't know. I can deal with both of them.
This is bad for us.
I cannot complain about my PhD students.
There's time for one last question. And let's put it this way. I think we can consider pathogens as the bad guys of microorganisms. Why would you say that in science, as in movies, the bad guys are always the most interesting characters?
Well, to be honest, that's a tricky question because you are already implying something that I might disagree, right?
Yeah, I'm happy that you disagree with me. Don't worry.
I think bad guys are always the most interesting ones. I'm not sure. I'd have to think about it. I think it will depend.
The bad guys, yeah, I did this. Yeah, it's like they're always the ones we're searching for. More in science, but you can actually see the same thing in movies.
Yes, yeah, because the bad guys do us harm, and nobody likes that, right? But actually, you will find some scientists, some scientists that will focus on the good guys, and from where you can learn a lot. And if you think about, for example, the model organisms for bacteria, they are good guys. It's Bacillus subtilis and Escherichia coli. They are harmful. They don't do any harm. And in fact, that's one of the reasons, not the only reason, but one of the reasons why they are model organisms, because they are safe.
Yeah, so we basically need the two of them to move the story forwards, either in science or in the movie. Basically, you need to look for the bad guy and for the good guy.
Yes, and now in changing just slightly a little bit the focus, but that question is important, particularly for a day in a day like the International Microorganism Day, is that most people know that bacteria exist and microorganisms exist, but many people will associate them to bad things. And we know that we need bacteria to be healthy, and we need bacteria for many things in our lives. And it's not only about disease. So although the bad guys catch most of the interest, I think we have really to promote the good guys in this case.
I would like to finish this conversation, let's say. I would like again to thank you for accepting this invitation, and of course thank you for this interesting conversation. You guys can know a little bit more of Raquel's work in a link that we're going to leave here, but it's at ITQB's website. You can also check our social media to know a little bit more about our podcast. And I think that's it. We'll wrap this up. See you soon, and have a nice Microorganism Days, guys.
Thank you. Bye.
Bye.
So thank you very much to PCR Podcast for a very interesting topic and a really cool and interesting format and a really inventive way of presenting interviews. Like they said, in a time of COVID where it's difficult to meet up with people. Without further ado, I would like to continue to the next pre-recorded talk by Tony Gutierrez from Heriot-Watt University, UK, to talk about oil-degrading bacteria and their importance in cleaning up our oceans. So without further ado, here is Tony Gutierrez.
Hello, my name is Tony Gutierrez, and I work at Heriot-Watt University in Edinburgh in Scotland. I'm going to talk to you for the next 20 minutes on oil-degrading bacteria and their importance in degrading oil in the oceans. So I hope you enjoy this talk. I'm just going to start sharing my screen. Hopefully it'll come up. Let's see. Right, here we go. Okay, so I'm going to start off now to begin with showing you how significant the volumes of oil are that enter our oceans and seas each year. This slide shows the major transportation routes of oil worldwide. You can see by the multitude of shipping routes, as represented by the network of lines going around the world from one country or nation to another, millions of tons of crude oil and its refined products are carried across all the major seas and oceans every year. This results in oil entering the sea from a ship's exhaust fumes and spills that may occur along the way. Now, in this next slide, it shows all the major spills. This is of more than 34 tons that have spilled oil into the sea from any source, but which is non-military. A major take-home message here is that oil spills in the sea occur frequently in one shape or form every year. Now, there can be up to more than 100 spills in a single year around the world, and the volumes of oil that enter the marine environment from these can be from hundreds to thousands of tons to over millions of tons. So you get the idea. From human activities, we release a lot of oil accidentally into the sea every single year. Now, you'll notice the volume of oil released was huge in 1979, right there, as I'm pointing on that graph there. And this was from the Ixtoc 1 oil spill, which was a massive disaster that occurred in shallow water at about 50 meters depth only in the Bay of Campeche off the coast of Mexico in the Gulf of Mexico. It resulted in the release of nearly 500,000 tons of oil. Now, the more recent Deepwater Horizon spill, it's not shown on this graph because that occurred in 2010. So this graph is a little outdated. But that Deepwater Horizon spill does bear in comparison, and both spills today are recognized as the worst oil spill accidents in recorded history. The other one was the Exxon Valdez, but the volume of oil released in that spill was way less, but it did cause a lot of havoc because it occurred in a very pristine region in Alaska. Now, if we were to include on this same graph the volume of oil released from one military-related spillage, in this case, it was from the Gulf War, as you'll see from this next slide, there it is there, the Gulf War spill. So this spill, basically, if you put it on this graph, it dwarfs all the others in terms of the volume of oil released into the Persian Gulf. And that was not an accidental spill. That was an intentional, deliberate spill. Now, the Gulf, fortunately, the frequency of oil spills has decreased over the years, as you can see from this graph. And proportionally, the volume of total oil released into the oceans has also decreased. This is shown by the oil droplets. As you can see, the oil droplets represent the total volume of oil released each year, sorry, not each year, over a period of decades. So the first one in the 70s, the next one in the 80s, and so forth. You can see the volume of oil entering the ocean over the decades has significantly decreased as well. Now, this decrease is attributed mainly to the implementation of stricter regulations with respect to health and safety, the prevention of spills, such as by constructing oil tankers with double hulls. So if they were to run aground, they are less likely to rupture and release their cargo of oil. Now, what I want to show you here with this slide is that in addition to large volumes of oil entering the sea from human activities, it is estimated that as much as 49% of oil entering the seas from natural oil seeps. And this is represented by this huge chunk of the pie, the purple section here. That is the volume of oil that represents from natural seeps that inflows into our seas and oceans every year. When you factor the volumes of oil that enter our oceans from both human activities and natural seeps, so this other half is from human activities, the total is enormous. So as you see, hundreds of millions of gallons of oil enter the oceans every year from different sources. Now, it is calculated that this would be enough to cover the surface of all the oceans and seas around the world in a thin blanket of oil. When I'm talking about a thin blanket, I'm talking about thickness of a few molecules of oil. But our oceans and seas aren't covered in a black goo. And this is fortunately due mainly to oil degrading bacteria. Now, these bacteria, these are the ocean's gas guzzling machines that help to mop up the oil as it enters the sea. These types of organisms do the same job on land, but their there's something unique here about the ocean in terms of these bacteria. As far as we know, only the oceans are what contains oil-degrading bacteria that use oil as their preferred food. So these types of bacteria are referred to as obligate oil degraders. In other words, if you give them conventional food sources such as sugars, they won't touch it. But give them oil and they'll be very content and thrive. Now, since the first description of an oil-degrading bacteria back in 1913, we've come a very long way to understanding the diversity, function, and ecology of these organisms, especially at oil-contaminated sites. Now, in this slide represents a phylogenetic tree. I'm not expecting you to understand what a phylogenetic tree if you're not familiar with these things, but it shows just the evolutionary relatedness of the various obligate oil degraders that we know of today. And these I show you here on this tree in bold. And these are species with names: Alkanoclasticum at the very top. Going down, we have Oleophilus, Oleospira, Thalassolitis, Alkanovorax, Neptunomonas, which apparently is not shown there in bold, but it is an obligate degrader, and Cycloclasticus further down. Now, all the other species shown on this tree are also oil degraders, but the difference is that they are not obligate in this sense of degrading oil, because they are also able to utilize other conventional sources of carbon for their growth and energy, such as sugars like glucose and fructose and so forth. So there's something quite unique about the ocean, that it contains these types of bacteria which just thrive and love and only prefer as a food oil. So where do we find these obligate oil degrading bacteria? Well, with some exceptions, essentially everywhere. So this map is a bit outdated, but it shows you where to find these organisms, and they're essentially all over the world. They are what we call ubiquitous. There are some large areas, mainly open ocean sites, where you don't see colored dots, like out here in the Indian Ocean, bottom Atlantic, only one dot there in the North Atlantic, and out here in the Pacific. Now, there's a reason for that. Of course, these large areas, mainly the open ocean sites, where you don't see these colored dots, it's because no one has looked for them there. And also you have to consider that research cruises don't often go far away from a coast. It's very expensive to take a research cruise out so far out. It takes time, and thus research cruises out to the very large expanses of the ocean are rare. So, but if we were to sample these open ocean sites, we would definitely be finding oil degraders there. Now, in recent years, some novel obligate oil degrading bacteria have been discovered that I haven't presented in the previous slide showing that phylogenetic tree. And these new ones have been found living associated with marine microalgae or phytoplankton. If you know what phytoplankton are, like the plants or the rainforests of the oceans. They're the ones that produce in the oceans about 50% of the oxygen in the atmosphere. So these discoveries has expanded our knowledge on the diversity of these types of bacteria in the oceans. And we wonder how many more are left yet to be discovered. So it's probably quite likely, I would imagine, many new species of obligate oil degrading bacteria still are waiting to be discovered in the oceans. Now, to exemplify the importance of oil degrading bacteria when oil spills occur at sea, I thought what better example than to show this with the historic Deepwater Horizon spill. Now, this spill made headlines all over the world, and it became the focus of many research groups in many countries, not just in the USA where it all happened. It had to be a big one if a movie was made about it, which it was. It starred Mark Wahlberg and Kate Hudson. Now, someone that I met with over 30 years experience in working on oil rigs in several countries said to me that the film was a pretty accurate description of the oil rig blowout, including how Mark Wahlberg and other actors portrayed the character of workers on an oil rig. But the movie was nothing to do with the microbiology, where in the eyes of a microbiologist, the real drama occurred on the sea surface and below, where the oil eventually impacted large expanses of the Gulf of Mexico. So the disaster occurred on April the 20th of 2010, when oil started gushing out from the riser pipe at the seabed here, at the very bottom of the ocean, which was about 1.5 kilometers below the sea surface. And it was capped about 3 months later on July the 15th of that year. Now, estimates vary, but about 4.1 million barrels, which is a bit more than 500,000 tons of oil, entered the Gulf of Mexico over a period of 84 days. It's almost about 3 months. To try and picture this, that's about 300 Olympic-sized swimming pools full of crude oil. Now, that's quite something. The rate of oil gushing into the Gulf varied over the duration of the spill, but on average, this equates to about 3.5 Olympic swimming pool volumes every single day. Now, as depicted on this slide, the spill resulted in the unprecedented formation of an enormous oil plume right here. At a depth of around 1,000 to 1,300 meters below the sea surface. The plume stretched about 30 kilometers on a horizontal axis, a few kilometers in the other horizontal direction, and about 300 meters thick vertically. Now, like a cloud in the sky, this massive oil plume that was suspended in the water column also moved gradually and in a southwesterly direction in the Gulf of Mexico. A lot of the oil that gushed out from the leaky well had also reached the sea surface to form large expanses of oil slicks that could be seen from space. And what was also unprecedented about this spill was the large quantities of chemical dispersants that were used to treat the spill, both at depth here, where the oil was gushing out at 1.5 K below the sea surface, and also on the sea surface by spraying, for example, from airplanes. But the main arsenal to combat an oil spill comes naturally in the form of those oil-degrading bacteria, which we found responded strongly in both the plume and sea surface oil slicks. Now, bacteria and other types of microorganisms are excellent indicators for the status on the health of local ecosystems, as well as indicators to perturbations in an environment. They are like taking your temperature, if you like, to assess whether you might have a COVID-19 symptom. Now, this slide shows a depiction of the water column in the Gulf of Mexico, where the Deepwater Horizon spill occurred. Here, showing the oil gushing out from the leaky well head at the bottom here, and the formation of an oil plume at depth right here. Now, and as you can see, oil also rising up to form these oil slicks at the very top of the sea surface. The scientists analyzing the microbial communities showed that there was a strong response by oil-degrading bacteria in the oil plume compared to above and below it. So there was a very strong response in this section here compared to above and below that plume. And scientists have shown that there was an enrichment in a particular taxonomic order of bacteria called the Oceanospiralalis within that plume. And it accounted for about 90% of the total bacteria in the plume region. And it has been shown by sequencing the genome of those types of organisms that they had the capability of breaking down oil hydrocarbons. So they were strongly enriched, and we know that they were involved predominantly in breaking down the oil hydrocarbons within that plume region. Similarly, oil-degrading bacteria on sea surface oil slicks up here were also strongly selected for, in particular species belonging to Cycloclasticus, which is an organism that's very good at breaking down some of the most toxic hydrocarbons in oil, which are called the aromatic hydrocarbons, especially the polycyclic aromatic hydrocarbons. Other organisms on the sea surface that became enriched were called Halomonas, Pseudoalteromonas, and Alteromonas, which are also recognized as oil-degrading bacteria. Now, work that led to isolating some of these microbes that became enriched by the oil, and then analyzing of their metabolic capabilities showed that they were actively involved in degrading the various hydrocarbon components that make up the oil that gushed out from that leaky well during this massive spill. Now, so I'm coming now to the end of my talk, and so just a recap here. I hope this talk has come across to convincing you of the importance of a particular type of bacteria in our world, and believe me, the diversity is enormous. But this type they're very important, the oil degraders. They keep our seas and oceans in check effectively from pollution, mainly from crude oil. Of course, there are many other types of bacteria in the oceans that are important in cleaning up and keeping our oceans in check from other different types of pollutants. But these organisms are essential. They're key in breaking down the oil, and is reason why oil does not accumulate in our oceans. So in summary, there are enormous volumes of oil in the hundreds of thousands of tons that enter the oceans and seas every year. The fact that the oil does not form a black slick on the sea and ocean surface is largely due to the presence of and activities of these oil degrading bacteria. These microorganisms perform a fundamental role in the oxidation and mineralization of the hydrocarbons that make up the oil, and they are at the heart of bioremediation processes. Now, in the sea, some of the most important organisms doing this job are the so-called obligate oil degraders, as I've explained in my talk. And we know quite well who these species are. And based on new species having been discovered recently, such as some that have been found living associated with microalgae or as they can be termed phytoplankton, it seems likely that many more may await discovery. Large oil spills like the historic Deepwater Horizon disaster are horrible incidents, but represent an excellent example of the importance of oil degrading bacteria cleaning up our oily mess. So I'm just going to now stop sharing my screen because this has come to the end of my talk. And right, there's me. I hope you've enjoyed this talk. I look forward to answering any questions you might have. Just thread them through the program organizers. And I also thank the hosts for organizing this wonderful program on Microorganisms Day. And I thank you all. Keep well and keep safe. Bye-bye.
Okay. That's a fantastic presentation from Tony Gutierrez, and now I'm here with Associate Professor Kimberly Klein from CELSI and Nanyang in Singapore. Hello there, Kimberly. Can you hear me okay?
Hi, yes, I can hear you. Can you hear me?
I can indeed, I can indeed. And that's a lovely background you have there. It looks very, very sunny and tropical.
That's how it looks here.
Fantastic. And so I gather you're going to talk to us today about your work on Enterococci and also some words about the new Fems journal that you're editor-in-chief of. Fantastic. Well, without further ado, I'd love to hear about it and hand over the floor to you. Thank you very much.
Great, thank you. Can you see my slides?
Yes, yes, I can.
Okay, great, thanks. Thanks for inviting me to be part of this, Ben. This is really a neat initiative. I'm going to start by introducing a journal that Fems has just launched that I'm heavily involved with, and this is called Fems Microbes. And our tagline for this journal is Open Science Toward a Sustainable World. And the reason for this tagline is because we are really interested in publishing manuscripts covering all of the microbial sciences. And so our scope is to publish both basic and applied microbiology papers across the areas, so including industrial microbiology, environmental microbiology, clinical and veterinary microbiology. And we're dividing our journal up into a couple of different areas. And so we hope that this will really cover the spectrum of microbial sciences, and you can see where your submissions could fit into the journal. And so these include microbes and metabolism and multicellular life, disease, environment, and microbes and society. In addition to these sections, we publish a number of different kinds of manuscripts. So we have research articles, of course. We're also publishing short review articles as well as commentaries. And so we invite pre-submission inquiries, particularly for those reviews and commentaries. This journal launched a few months ago, and our first published articles should be going online next week. So we're really excited about that. And we think that this journal has a couple of interesting properties that we want to point out to you. And so first, these are the folks that are involved in the journal. And so you can see on the right, the editors-in-chief are myself, Jenna Jass, and Kathleen Scott. So it doesn't take a lot of attention to realize that we have an all-woman editorial board, and this is really exciting to us. This wasn't intentional, it just happened this way, but many editorial boards do not have this gender makeup. So we're kind of excited about this. More interesting is the diversity in terms of geography that our editorial board represents. And so we have covered Europe, Southeast Asia with me, as well as North America. And our handling editors, the rest of our editorial board that you can see on the left, also is a very diverse group of people covering a range of microbial sciences, a lot of different geographical sites, different career stages as well. And so this is something that's also a really important aspect of the journal for us. So some details about our journal that might be interesting for you to know about. So we have what we consider progressive publishing policies. So we are very open to and actually encouraging of people to publish or post their work on preprint servers prior to publication. So we encourage this. We have a very transparent peer review process as well. So authors have the option to publish all of their peer reviews as well as the author's responses to those reviews along with their manuscript at the time of publication. In addition to that, the peer reviewers themselves have the option to have their names included with their reviews. And so there's the option of full transparency if everyone is interested as well. We also, in the interest of transparency, recognize co-reviewers. We appreciate that many reviewers invite trainees, postdocs, PhD students to help with the reviews, and we think this is very valuable. But we recognize that those co-reviewers don't always get credit for their contributions. And so we will be recognizing those co-reviewers, and they can get credit via Publons for their contributions to the reviews. We offer scooping protection. So if a publication comes out showing what you've demonstrated while your paper's in the pipeline with our journal, that has no negative consequences for you at all. We offer discounts for Fems affiliated societies, for publication fees, and our publisher, Oxford University Press, as well as Fems, the society, do a great deal of work to help promote our papers. And so what that means for you is that if you publish your paper in Fems Microbes, it will experience increased visibility because of this behind the scenes work from everyone involved. And then finally, one of the, I think, most important things from my perspective about this journal is our strong dedication to incorporating early career researchers at all stages of the journal. So we are actively building a pool of early career peer reviewers. This has been sprinkled around social media as well, and Fems will be formally inviting people to submit their names to our growing database of early career researchers for peer review. We are calling for submissions for thematic issues, and we'll particularly pay attention to those submissions coming from folks that include early career researchers as part of the guest editorial team for that thematic issue. Fems hosts a number of webinars where we highlight papers that we're publishing, and so this is a great opportunity for early career researchers to get their work and their faces out there to the public and share their research with the world. And then Fems is also really active on social media, and so there's a number of ways to get involved with that as well, as you can see here on this slide. So if you have more questions about Fems, there's a lot of information online, and so you can check that out at these different links. You can drop me a question if you're listening live into the chat here. And we look forward to hearing from you and seeing your submissions down the road. And so with that, then I'm going to switch and tell you a little bit about the research we've been doing here in my lab in Singapore, where we focus primarily on the bacterium called Enterococcus faecalis, and we are most interested in its ability to cause biofilm-associated infections. And so we're motivated to study this organism because it is always on the serious threat lists that are put out by, for instance, the CDC or the WHO for its high prevalence of antimicrobial resistance. And so you can see the numbers here on this slide. It's often associated with resistance to antibiotics of last resort. So this is a major clinical and public health problem. So there's two species of Enterococcus that most frequently cause infection in humans, and those are E. faecalis and E. faecium. We study primarily E. faecalis, and this is a commensal in the GI tract of probably everybody listening to this talk. It's just part of the healthy human gut microbiome. However, when things go awry in susceptible hosts, it can get out of the GI tract into other sites and cause opportunistic infections. And so it's most frequently associated with catheter-associated urinary tract infections. In people that are exposed to broad-spectrum antibiotics, it can grow out of control in the GI tract, and this can lead to infections in other sites, such as the bloodstream. And it's also associated with a wide variety of wound infections, including diabetic foot ulcers. And so in my lab overall, we're then interested in understanding how E. faecalis perceives these different host environments it finds itself in, and how it responds accordingly by, for instance, the elaboration of surface structures that allow it to thrive in these environments and to also form the biofilms that we know are really important for all of these different infections. And so today I'm going to tell you just a little bit about the work that we've been doing in the context of wound infections. So I just mentioned that biofilms are important for these wound infections, but it's not just E. faecalis biofilms. And most of these infections, and actually most of the infections it causes across the body, these are not monomicrobial biofilms. These are often polymicrobial interactions that E. faecalis finds itself participating in. And so this is something else that we're really interested in understanding. How does E. faecalis interact with other organisms at these infection sites? And so I'll tell you again a little bit about the work we've been doing in the context of wound infections. So we use a mouse model of wound infection in the lab. And what we see is that if we infect these mice with about 10 to the 6th bacteria, within a few hours after infection, we see this very acute and rapid replication of the bacteria within the wound. However, as time progresses, what we see is that the bacteria sort of level off at this chronic persistent infection level. And we think this is important because this reflects what happens in humans experiencing chronic wound infections, where the bacteria just are not cleared well by the human immune system. They just hang out for long periods of time. There's some other features of this wound infection model that also reflect what are seen in human wound infections. So these are biofilm-associated infections, and we can see readily these little microcolonies of E. faecalis scattered across the wound bed. And in addition, in humans, wound infections are associated with delayed healing, and we also see this in the animals. So here on the bottom, this is a cross-section of a wound that was not infected. 7 days later, what you're looking at here is a completely healed wound. Whereas on the top, this is an infected, E. faecalis infected wound 7 days later. This is not healed. You can still see a very clear scab. And in the underlying tissue, there's no clear signs of healing yet. So this is clearly delayed wound healing. So we wanted to understand then how E. faecalis was causing these outcomes within the wound infection. And so to start this out, we looked at the immune response at these different stages of infection. And so during this acute replication period by E. faecalis, we looked at the cytokines and the chemokines that we could observe at the site of infection. And so what you're looking at here in this spider plot is animals that are mock infected in black, and then the animals that are infected with E. faecalis in blue. And what you can capture here is that there's a strong pro-inflammatory pattern of cytokines and chemokines being produced during this acute infection. However, if we look at these later infection stages during this persistent infection, the picture is quite different. So you can see that the blue and black lines overlap predominantly, so there's not a strong immune response. And if anything, some of these blue lines, these infected animals, dip down below the mock-infected animals. And so this started to hint at us that there is some degree of immune suppression happening during this persistent infection. And so we can also see that in the immune cells that respond to the infection site. We can look at neutrophils, and again, during acute infection, we see a strong neutrophil response, which is what we would expect and is consistent with the cytokines and chemokines we observed. And this dissipates over time. And if we look at macrophages, it looks the opposite. We see very few macrophages early in infection, and this starts to go up over time. And so this presented us with a conundrum. So if these immune cells keep coming in during the chronic infection, why aren't they clearing it? And so to address that, we turned to some in vitro models to look at this. So here you're looking at macrophages in vitro. We use an NF-kappa B reporter assay to look at the degree of macrophage activation. And we asked the question of whether Evolutionary could actually suppress immune responses in this experiment. So we activate our macrophages with LPS, which will maximally activate them. And then we add increasing numbers of Evolutionary to the cells. And what we see is a really strong dose-dependent pattern of immune suppression of LPS, activation with Evolutionary. And this is in the absence of any cell cytotoxicity. And so LPS is kind of an artificial way to do the experiment. So we also asked if we could see the same thing with a co-infecting organism. And so here we infect our macrophages instead with E. coli. This also by itself can activate the macrophages. And again, Evolutionary can actively suppress that E. coli-driven macrophage activation. And so this then became pretty interesting to us because I told you Evolutionary often participates in these polymicrobial infections. And so we wondered if this might be important for these mixed species infections. And this interaction between E. coli and Enterococcus is relevant to real infections. This is metadata from bacteria that are most frequently cultured from diabetic wound infections. And so here you can see Enterococci are in the top 3, along with Pseudomonas and Staph, but E. coli and Klebsiella round out the top 5. So we wanted to then understand even better this interaction between E. coli and Enterococcus within wounds. And so now we go back to that wound infection model I just showed you. If we infect E. coli by itself, we see that the animals have relatively low titer E. coli wound infections. Each of these dots is the number of bacteria in one mouse. And so we see 100 to 1,000 bacteria per animal. But if we co-infect with Evolutionary, this changes dramatically. We see many of the animals have extremely high titer E. coli wound infections. And so we wanted to understand this better. What is driving this high titer wound infection? And so we can model this, what we see in animals, also in the laboratory, in vitro. We can grow biofilms in different assays and look at these interactions. This is a macrocolony biofilm assay. And these macrocolonies are 5 days old. So this is E. coli by itself, Evolutionary by itself. And if we start to mix bacteria together at a 1-to-1 ratio, we don't see very much. But at the time of inoculation, if we increase the ratios of Evolutionary to E. coli, so there's a little more E. coli or Evolutionary in the mix, we start to see these really dramatic changes in colony morphologies over time. And even though there's more Evolutionary at the start of this experiment, this outgrowth is actually due to E. coli. And we know that we can see that a little more clearly here in these proximity assays, where on the right, we spot Evolutionary along this diagonal, and on the left, we spot E. coli. And what we can see is that the E. coli grows into much larger colonies the closer it is to Evolutionary, and we can quantify that as well. So these E. coli are really proliferating as they get closer to Evolutionary. And so then to understand how that was working, we did some transcriptional expression profiling within E. coli close or far away from Evolutionary. And we find that the genes that are most highly upregulated in E. coli are those involved in iron acquisition, and specifically in the production of siderophores. And so what siderophores are these small molecules that are made by E. coli and exported into the extracellular space. They bind iron with very, very high affinity, and then the E. coli can take up those iron-bound siderophores, and this serves as a source of iron. This is important because iron is an essential nutrient for E. coli growth, and iron is often very limited in the environment, particularly during infection. And so we have mutants then in each of these siderophore biosynthetic pathways in E. coli. There's 3 of them, Enterobactin, Salmonellin, and Yersinia bactin. And so we then asked, do those mutants grow to bigger colonies just like the wild type E. coli does? And I think you can see in this picture, the answer is no. In panels A and D, these are Enterobactin mutants. So if E. coli doesn't make Enterobactin, it cannot grow better in proximity to E. faecalis. So this is what we see in vitro, but we're really interested in what's happening in that wound infection model. So we now step back to the animals and ask if the same thing is true. And it turns out the answer is yes. So I told you that if we co-infect wild type E. coli with wild type E. faecalis, we see that most of the mice have very high titer E. coli wound infections. But if we instead co-infect with that Enterobactin E. coli mutant, this is not able to get the benefit from E. faecalis and grow to higher numbers. So Enterobactin is important for that enhanced E. coli growth in the proximity of E. faecalis. So how does E. faecalis drive this in E. coli? And so through some genetic screens that I don't have time to tell you about, we discovered that one particular gene, ARCD, which encodes an antiporter called ARCD, seems to be important for driving that interaction. So ARCD simultaneously imports arginine and exports ornithine out of E. faecalis. And so we hypothesized that that exported ornithine might be serving as a cue for E. coli to grow better in proximity to feces. So to test that, we add ornithine directly to E. coli in the absence of E. faecalis, and we see indeed ornithine can drive E. coli biofilm growth by itself. So it's sufficient for that enhanced biofilm growth. And it grows biofilms then as well as E. faecalis, the E. faecalis mix does. And really importantly, this is dependent on Enterobactin, because if E. coli doesn't make Enterobactin, ornithine has no effect on it. So then together, all of this says, in vitro, E. faecalis exports ornithine. This serves as a cue for E. coli to grow better in an Enterobactin-dependent manner. And so lastly, then we go back to our animals and ask, is ARCD and ornithine also important in vivo? And here the answer is a little bit mixed. And so again, E. coli by itself it has really low titer wound infections. The mixed species wound infection, we have lots of E. coli in the wounds. If we co-infect with wild type E. coli and the ARCD mutant that doesn't make ornithine, we get an intermediate phenotype. We have fewer animals with high titer wound infections, but we still have these animals, a residual subset of animals with very high titer E. coli wound infections. And so this then led us to hypothesize that the immune suppression that I told you about at the beginning of my talk might be responsible for this residual growth of E. coli. And that turns out to be the case. We now have mutants in E. faecalis that are unable to suppress macrophage activation. They can't suppress the immune response. And so if we instead co-infect E. coli with those E. faecalis, so there's a stronger immune response, essentially, we lose those residual high titer E. coli wound infected animals. So what this says together is that E. faecalis has a couple of mechanisms that are ultimately beneficial to a co-infecting organism, in this case, E. coli. For one, it helps E. coli acquire iron by this constitutive export of ornithine, leading to Enterobactin production and iron acquisition. And simultaneously, by a completely different mechanism, E. faecalis is suppressing the host immune response. And we know that E. coli by itself is very pro-inflammatory. So this, we think, has an added benefit for E. coli as well. So taken together, we have started to think of E. faecalis as kind of a Jekyll and Hyde organism. So in normal healthy hosts, if it stays in the GI tract, it's a completely innocuous and often helpful organism. But due to its ability to both modulate the immune system and participate in these interesting polymicrobial interactions, it can turn into a bad guy. And so we call that Mr. Hyde. And so, of course, we're really interested in understanding then how E. faecalis interacts with other organisms that it finds itself with within different infection sites. And so that's some of our ongoing work. And so with that, I will just thank very much the people whose work I showed you today. This is Kelvin, this is Brenda, Patrick, Wei Hong, and Damien. And I will stop there and thank Ben again for the invitation to join you today.
Thank you very much, Kimberly. Fascinating stuff, both with the journal and this really cool and complete story. Unfortunately, we don't have time for questions here, but is there anywhere people can kind of ask you stuff and get a hold of you?
Yes. Let me just go back to my first slide. I have my, hold on. Let me do it this way. Here we go. Kimberly at Kim in Singapore is how you can find me on Twitter, or you can go to my lab website and you can find my email address there too.
Perfect. Thank you. I'm sure there'll be lots of questions. And I look forward to hearing more about your research and more about the journal. Thank you very much for your time and your talk.
Thanks a lot.
Okay. Bye.
Bye-bye.
With that, then, it's my pleasure to introduce the next pre-recorded talk. And this is a presentation from the University of Valencia, from Manuel Poisson, and he is talking about, in his presentation, the planet of microbes. So I'm going to go straight ahead and roll that video now.
So good morning or good afternoon. I'm going to give a talk, the title of which is Planet of Microbes. My name is Manuel Porcar. I'm a researcher from the University of Valencia, Spain, and I have a research group there in the Institute for Systems Biology, but I am also the co-founder of the company Darwin. So both of them, the University of Valencia and Darwin, are here. You can see it in the map, in the scientific park of the University of Valencia in the east coast of Spain. So we're going to talk about microbiology, and what I would like to start with is with the fact that microbes are everywhere. They are in the forest, on the leaves, on the roots. They are on the water, on the food. They are even on the sky, in the clouds. So everywhere where you can look at in our planet is virtually full of microbes. So normally microbes live in biofilms. A biofilm is what you can see here in this amazing electron microscopy picture. And this kind of tiny bugs that you see placed nicely one after the other, like if they were wagons in a train, those are bacteria. So one of the things that we made a few years ago was to study several particular environments in order to see whether we were able to identify bacteria or other microorganisms there. One of the maybe most famous articles or works that we did is this one, the coffee machine bacterium, or some people call it an espresso bacterium because it's the kind of machine that we studied. So the idea is that, as you know, these machines work with capsules, and these capsules are stored inside the machine. And there's always a bit of liquid dropping off from the capsules. And there we studied them and saw that it was quite an impressive amount of bacteria and some fungi as well. In this graph here, these colors represent the specific bacteria that we were able to identify. And what I want to highlight here is that first you start with quite a big diversity of bacteria. Then it comes some other specific species, Pantoea, Bacillus, then in time Sphingodium. And after you are operating 2 months, this machine, this coffee machine, you end up having a pretty specialized community of bacteria, for example, Pseudomonas, which live happily in this caffeine-rich environment. In other words, you have a machine, you have bacteria in it, this is normal, but the kind of bacteria that you are having in a coffee machine is those who are caffeine lovers. This is another kind of research that we did. As you can see here, I am with one of my students, Christy Tanner, sampling a solar panel. So the idea was crazy again, is there any kind of bacteria able to live on the surface of a photovoltaic panel, which is a surface that is oriented towards the south, in the northern hemisphere at least, and is super heat in summer up to 60 degrees, very dry, etc. So in the picture on the right, you can see how many bacterial isolates we were able to identify. There's full of bacteria there. These are the species that we were able to identify through a technique called next generation sequencing. Basically, you can know very well today which specific bacterial species are on a given environment. So these bacteria you can see here, Deinococcus, Phingomonas, Novosphingolium, Hymenobacter, etc. They are typical from deserts. They are radiation resistant, they are heat resistant, they have a lot of carotenoids, which is this fruity compound that they give them these fruity colors. And they have sphingolipids that very likely help them to adhere, to stick to the surface of the solar panel. I'm not going to go into the details, but when we analyze these communities in order to see to which other environment on Earth they were close to, we identify that the closest Earth environment compared to a solar panel is the poles. So the north and south poles, in which you get no heat, of course, but a lot of sun irradiation. And bacteria there, they're also reddish, as you can see here, because they have to cope with some of the same stresses that bacteria of solar panels have to cope with. And those stresses are lack of water sometimes, extreme irradiation, oxidative stress, etc. So after seeing this, we decided to go to the north and the south poles. So we didn't do it personally. It was unfortunate because it would be a wonderful trip. But we collaborate with researchers who were working on northern Norway, on Tromsø, and in the Livingston Island in Antarctica. So what we did there was to analyze the bacterial communities in the solar panels in the North Pole and to compare them with the bacterial communities of the solar panels of the South Pole. And to our surprise, they were strikingly similar. So they are very, very far away, thousands of kilometers away. The climate is not that similar. It's cold, of course, but it's much wetter in one place than the other. It's not that similar. But the bacterial community is very similar because it's adapted to solar panels. Then, after analyzing the north and south distribution, we decided to do a similar thing with the east and west distribution. We are in Valencia, in the Mediterranean coast of Spain, as I said in the beginning of my talk. Then I moved to California, which is more or less in the same latitude, but of course very far away, on the western coast of the United States of America. And there we sample, again, solar panels. You can see some of the bacteria there at the left with these wonderful pink and reddish and orange colors. And they are there attaching to glass that we used to study whether they were able to attach to the glass. And they attach very well to the glass. When we compare the microbial community of the solar panels in Berkeley, California, with the bacterial community on the solar panels in Valencia, Spain, we found that they were almost identical, very, very similar, despite the distance. So the conclusion here is that wherever you look at on our planet, very likely, solar panels tend to host a very similar bacterial pool. And this is not because the climate is the same, because the North Pole and California or Valencia have very different climates, but because the ecological niche is very similar. So living in a solar panel is a very difficult thing in Valencia, in California, in the Northern Pole, or in Antarctica, because you have no water retention ability, the glass is always dry, you have a lot of irradiation, oxidative stress, etc. So this is just like a basic research in order to see what is the biodiversity of a previously unexplored environment, such as was the case with the coffee machine. But is it useful? Can we use this bacteria for something? So yes, we tried this organism as a model. This is C. elegans. This is a worm, a nematode. And we fed this worm with our bacteria. And we saw that when this worm was fed with our antioxidant producing bacteria, it was able to much better survive oxidative stress in the form of UV light or other regions that make oxidative stress occur. So these bacteria are producing an antioxidant effect on a living organism. That's a different thing. So I don't know if you like I do, these Sergio Leone films, with some of them Clint Eastwood, that are supposed to take place in southern United States, these very dry places. So this is not United States. All these Sergio Leone and Clint Eastwood films were shot in Spain, in Almeria, in southeast Spain. You have this picture there, 4 Bravo, 4 kilometers. This is Spain, in fact. And they chose that location because it's extremely dry and it's similar to southern United States of America. In fact, it is supposed to be the only European desert. What we did like a couple of years ago was traveling to that wonderful place. It's a very nice place, super dry, but very, very aesthetically appealing, and took a lot of samples that we analyzed in order to see if we were able to identify drought-resistant microorganisms, bacteria that are able to resist without much water. And to our surprise, we were able to identify a huge diversity of microorganisms, several of which have never been described before. So we are still now in the lab, which is, by the way, there. We are in the lab now trying to identify new species that we were collecting from these places. But not only the coffee machine, not only the solar panels, not only deserts. So the sea, what about the sea? This is 3 locations in the Valencian land that we sampled about 1 year ago. And we took samples from these specific places that are on the rocky shore. And they are sometimes covered by water, sometimes they are not. So it's extremely salty places with a lot of heat, sun irradiation, a very bad place to live. And yet, we identified bacteria. And some of those bacteria had wonderful antioxidant effects when tested on these organisms I mentioned before, C. elegans. This graph here shows the percentage of survival of the worms when you stress them with, for example, UV radiation. So only 40% of them survive normally. But if you give them vitamin C, which is an antioxidant, 60% survive. So this has a clearly positive effect on their survival. But if instead of giving vitamin C, what you are giving them, what you are feeding them with is our bacteria, so they survive better as well. In other words, our bacteria have a very good antioxidant effect on C. elegans. But only on worm could our bacteria be useful as a kind of therapy in a human disease. So there's an amazing example of that, which is what I call the ultimate motivation. Could our bacteria help sick children? So there is a disease, a human disease called Leigh syndrome, which is a mitochondrial disease. And this disease affects kids that normally die at the age of 2. So it's a terrible, very bad disease. The idea is there's only few treatments that work a little bit and not very well against this disease. And some of them are based on antioxidants. So I had a meeting with Albert Quintana, who's a researcher in Barcelona working on that. And then I had the idea to use our bacteria. It was a crazy idea to see whether our antioxidant bacteria were able to make something on this disease. Of course, we were not allowed, and we would never do that, to work with children, but Albert Quintana Lab has a wonderful facility with mice that have this disease. And these are the amazing results that we have. The black line here shows the percentage of survival of the mice that have this disease. So as you can see here, they die more or less at 40 days of age, which corresponds more or less to 2 years in a human, in a kid. But if you supply these mice, these diseased mice, with bacteria, they live much longer. They live up to 70 days. So this is a lot, like 50% more. Of course, this is just the first step. Of course, we are far from saying something sure for human treatment, but as a first step, it looks really, really hopeful and wonderful. And the basic idea is that when you supply bacteria, antioxidant bacteria, to these disease mice, they live longer and better. And I say better because this is another image of the brain of these mice. And when they are not treated, this means when they do not eat our bacteria, they have a lot of inflammation on their brain. Is the two pictures on the left. So you see a lot of red and green colors because the brain is really diseased. The same mice, if they are eating our bacteria, you get the pictures from the right. So there are less colors, there's more black and less red and less green because the brain is clearly better. And people working with this mice told us, listen, it's not only about the results we are getting, it's not only about these pictures or the mortality. You should come and see the mice. They look much better. So this is a very nice thing that we want to explore in the future, of course. So in summary, microorganisms are useful and it can be used for many, many things. Just to give you one example, this is one bacteria of the genus Pseudomonas that we isolated from a forest. And what you can see in this amazing electron microscopy is a piece of rubber. So this is a latex globe that we explore under the microscope. And our bacteria were able to dig holes on the surface of this latex. You can see the bacteria there, which are these long structures. And if you see the one on the right is like placed in a hole because the bacterium, has excavated that hole by dissolving and eating the latex. So the idea here, of course, is that you can use bacteria in a process called bioremediation. So you can use bacteria to remove environmental pollutants because they eat them, basically. This is something that we did in our academic lab in the University of Valencia. And in our company, just to give you one example of things that you can do with microorganisms, you can combine artificially microorganisms to do something useful. So in this case, there is a brand that we have just created called Masamatter. Masamatter.com is a website in which we are making a new product. And this product is a sourdough. So it's for making bread, for baking. And it's a combination of wild yeast with lactic bacteria. And in the same way that use black or red colors to make the color that they want, microbiologists can use bacteria to make the bread that they want, with the sourness that you want, with the flavors that you want. And we have made dozens and dozens of breads in order to, until we end up with the perfect formula. And I think that the bread is really, really good like that. So this is one example of things you can do with microorganisms, of course. Just to end this presentation, this is a picture of my team. I am extremely grateful to them. This is the company, Darwin, and this is my academic lab in the University of Valencia. So I thank you very much for your attention, and I'm very sorry that I cannot answer questions, but you have my email there and my Twitter, so in case you want to contact me, I will be delighted to have your feedback and to exchange any kind of comments or information. So thank you very much.
Okay, fascinating presentation from Manuel Porcar there in Valencia. And from there we go to Montana State University in the USA to Roland Hatzapincha, and he is going to be talking about the microbial ecology of extremophiles. This is another pre-recorded talk, but again, if you want to leave any comments or questions on social media, on Twitter, on Facebook, on YouTube, we will get back to you, or we'll get the presenters to get back to you. So without further ado, here is Roland.
This is Roland Hatzapincha. Thank you for joining my talk on the microbial ecology of extremophiles, of extreme-loving microorganisms on International Microorganism Day. I'm an assistant professor at Montana State University's Department of Chemistry and Biochemistry, and I'm also affiliated professor at the Thermal Biology Institute and the Center for Biofilm Engineering here in beautiful Bozeman, Montana. For those of you who do not know where Bozeman is located on, the southern side of Montana. And according to Star Trek lore, at least according to the movie Star Trek 8, it will be the site of the first contact between humans and the Vulcans in the year 2063. It's a small city of about 50,000 inhabitants with a big university with about 15-ish, 20,000 students, undergraduates and graduate students. And we actually, in fact, the closest university to Yellowstone National Park, which is also what I want to talk to you about today, microorganisms in the extreme features of Yellowstone National Park. Before we go to Yellowstone National Park together, I first want to give you a quick introduction of what environmental microbiologists like myself are doing. So the questions an environmental microbiologist has whenever we see a new microorganism, the same questions that you would ask a person when you meet them for the first time. Who are you? What are you doing? When are you doing that? Where are you located? And where do you do your job? With whom do you typically hang out with? Who are your friends? Who are your enemies? Who are your frenemies? And how many of you are there in the environmental sample or in the environment? And so traditionally, how environmental microbiologists have approached these kind of questions was to using cultivation approaches. So we went into the environment of interest. We took a sample that could be, a soil sample or a water sample. And we tried to grow microorganisms from that sample on a petri dish or in an Erlenmeyer flask in the lab. So we provided some artificial growth medium in a petri dish. We provided them with energy sources, with carbon sources, and so on. We made life as cozy as possible for these organisms in the lab, and then just waited a few days, and then waited up which microorganisms might be growing up in these colonies of bacteria that were growing up on the agar plate, and then try to identify who they are, and then what are they doing, and so on. There's a problem, however, and that is that the majority of organisms that we know out there cannot be grown in the lab. So if I would plot all the microorganisms that we know exist out there and only show the fraction of the ones that we can actually grow in the lab under artificial conditions, we would see that about only 1% on average of any microorganisms we know out there can be grown in the lab. But the majority of them is what is called the microbial dark matter, in a term that we borrowed from astrophysics, or the uncultured majority that makes up the majority of microorganisms in the human microbiome, in seawater, in sediments, in pretty much any habitat you can think of. The majority of organisms, we know that out there, but we cannot grow them in the lab. And to show you a little bit what big of a problem this really is I want to give you the example of soil. If you go outside and scoop up some soil, you will have about 5 grams of soil worth on your little spoon. In 1 gram of this soil, there live about 1 billion different microorganisms that are affiliated with bacteria, with archaea, or are members of the eukaryotes. To put this number into perspective, our galaxy has about 100 billion stars. In the soil, this 1 gram of soil that we just scooped up has about 10,000 to 100,000 different species of microorganisms. To put this into perspective, on the entire planet Earth, we have about 6,000 different mammal species. So we're talking about gigantic numbers here. And the Earth is obviously not only composed of soil, there's many different habitat types, in every one of which, as we just showed before, the majority of microorganisms cannot be grown in the lab. I first want to talk a little bit about why that is. Why is it so hard for us microbial ecologists and environmental microbiologists to replicate the conditions in the lab, and why some of the lessons we can learn by studying microorganisms directly in the lab are ultimately limited. And I want to give you this example of a lion. What food is a lion eating? How is a lion interacting with its environment? What you will do is you will go into the Serengeti and you will observe a lion. What are they doing? What you will not do is you will not try to capture a lion in the wild, bring it back into a zoo, put it into captivity, and then feed it a chicken. Because that condition is obviously not how a lion is living in the wild. But this is exactly what we're doing if we try to go to an environment and try to isolate an organism from its environment and grow it in the lab under artificial conditions. What you learn is still important. You still learn something about the biology of the organism, right? In this case, you learn that a lion eats a chicken. Of course, the lion will eat the chicken. Does that mean that the chicken is the natural prey for the lion? No, of course it's not, right? And so it is very, very important to keep that in mind for microbiology. What you read in a textbook about microbiology, right, has been done by studying organisms in captivity in a glass beaker that is shaking at 200 rounds per minute in a lab somewhere. This is not how the organism is growing in its natural environment. And this is what we as environmental microbiologists want to do. We want to understand what is the organism doing in the real world, in the real conditions where it's hanging out with, you know, thousands and thousands of other species. The other problem is that organisms, microorganisms, just as animals, have activity patterns. Some of them are dormant during the day, some of them are active during the day. Some of them are active in the night, and some of them, you know, can be dormant for quite a long time. There have been some microorganisms found in deep sea sediments that apparently have been dormant for millions of years. But if the conditions switch to the right, the environment suddenly, you know, has a burst of nutrients that, you know, flowed into the environment, suddenly the microorganisms become active because, you know, the condition changed for the better. The same concept obviously applies in zoology, right? We would never assume that all organisms are only active during a particular time of the day. Of course not. Everyone has their particular environmental niche, and the same is true for microorganisms. By growing them in isolation, in a beaker, you ignore all of that. You take this out of its natural conditions. What are we studying, or which kind of microorganisms are we studying in Yellowstone National Park? Why do we study hot springs to begin with? The first one is we know that hot springs resemble life's earliest habitat. At the start, after the formation of the Earth, after the Earth cooled down to allow liquid water on its surface, Earth was pretty hot still, right? We are talking about 80, 90, 100 boiling temperatures of water temperature. And this is the habitat where the early life forms, the first microorganisms formed. So by studying extant life in hot springs, we learn something about the origin of life, the origin of cells in the ancient habitat where Earth evolved 3.5 to 4 billion years ago. And we learn what the implications of that are for astrobiology. So for the study of life in the universe. Are there any other planets, any other celestial bodies out there that could also be home to alien life forms? And by studying the extremes of life on the planet Earth, we can learn what are the extremes that we can expect on other planets. Like, for example, planet Mars, or when we talk about hot environments, we typically talk about, the Jupiter moons, like Europa, where we think there is, deep sea hydrothermal vents on these moons. Another reason is that, as I just mentioned before, the majority of microorganisms on our planet cannot be grown in the lab. And 99% of the life on our planet is microbial. So 99% of the 99% of life on our planet are microorganisms that cannot be grown in the lab. So if we go to the environments that provide the most extreme conditions, we can explore the entire scale of diversity of life that exists on our planet. We want to look at the deepest habitats on the planet in the deep sea. We want to look at the coldest places in glaciers in Antarctica. And we want to learn about life in geothermal hot springs that are on the other side of the temperature spectrum. And the last one, hot springs are a great reservoir for potential biotechnology and biofuel industry. The most prominent example for this is DNA polymerase, the TAC polymerase that a scientist called Thomas Brock found in the organism Thermus aquaticus that he isolated from mushroom hot spring here in Yellowstone National Park, and in which a few years later isolated the DNA polymerase from on which the PCR, the polymerase chain reaction that we today use in labs worldwide to amplify DNA is coming from. So not only did this discovery and the development of PCR was awarded the Nobel Prize in 1993, the production of this one enzyme, this one thermostable enzyme from this 70 degrees hot spring alone is a multi-billion-dollar industry today, which shows that the potential for discovering new organisms, and their enzymes and how they can be used for biotechnological purposes with tremendous potential for health and for the economy. As well, the best place to study microorganisms that are living in hot springs is just, a 1 hour, 20 minutes drive south of Bozeman, where I live, in Yellowstone National Park, that is just located at the intersection of the US states of Wyoming, Idaho, and Montana. The park is about 3,500 square miles, or about 9,000 square kilometers large. To give you an idea, this is larger than the states of Rhode Island and Delaware combined, and is larger than several of the states in Germany or France, for example. So we're talking about a pretty big territory. Yellowstone National Park features about 14,000 geothermal features and is estimated to hold about 55% of all the world's geothermal features. And you see this map here that shows you the basic outline of Yellowstone National Park with a big lake in the center, and then all these red and yellow and orange colors indicate the biggest geothermal fields, in the park. So where you have clusters of hot springs and geysers, basically. And these green circles that are numbered here are the main locations where my lab is currently performing research. We said there's about 14,000 features, about 7,700 of them, so give or take about half of them, have been logged, meaning we know where they are, and we know what the temperature and pH is. What you see in this plot is the gray dots are 7,694 plots of every geothermal feature in Yellowstone National Park that has been logged. And on the x-axis, you have the temperature, and on the y-axis, you have the pH. And so what we see, there's a huge diversity in terms of the temperature and the pH of the geothermal features. But one problem is that historically, researchers have focused on a few very particular sites of hot springs. One of them being very, very hot, so in the range of 80 to 90 degrees of boiling temperature with a very, very low pH, so very acidic features. In the other ones, circumneutral, around pH 6, 7-ish, in a temperature of about 50 to 70 degrees Celsius. These features here have been mainly studied for the reason that they're so extreme that they can only host a very low diversity of microorganisms. In some of these features that are extremely hot and extremely acidic, you literally have like only 3 or 4 different species of microorganisms. Compare this to the soil I just talked about, where you have 100,000 species in 1 gram, right? This is a very simple community, which makes your experiments easier to conduct because you're only dealing with 3 or 4 species rather than 100,000. And these features in the center here have been mainly looked at because these are some of the most gorgeous features in the park. So this is a big problem I'll talk about in a minute. So after my lab was founded about 3 years ago, my lab has been conducting a very large survey of Yellowstone features, Yellowstone hot springs, to basically circumvent this problem that historically we only have looked at a very small portion of features in the park. And so all the red dots here are indicating hot springs in the park that my lab has sampled. And you see that, we try to get a very broader representation basically of all the features in the park to understand their geochemistry and their microbiological inhabitants. And so one of the things we found is that researchers are also humans, and we are just drawn to beautiful, quote unquote, aesthetically pleasing sites. So many of the sites in this area that I pointed out before that are, about circumneutral 50, 60, 70 degrees Celsius happen to be very colorful. And the reason for that is that at 70 degrees, just at the end of where this blue plot is here that I just showed you, pigments break down. So most of the colors that we typically see in many of those hot springs disappear. And so you're in this niche where you have brilliant, vibrant colors just before the pigments break down, which makes these hot springs beautiful to look at. And so historically, a lot of researchers have focused on these gorgeous sites simply because they're nice to look at. And the problem is just a few meters away, there might be a hole in the ground, like shown here, that is basically a pit that is, a meter in size, that is filled with muddy water, that also has about 60, 70 degrees Celsius. You see some gas bubbles forming here. And if you actually look into the microorganisms that live in one and the other hot spring, you learn that the ones in the left, in these gorgeous, vibrant colors, are already known to science for a long time. Because researchers have been studying these for 50, 60, 70 years. since the 1960s when modern microbiology in Yellowstone National Park started. So from my very high standard as an environmental microbiologist, right, the organisms in the left are pretty boring. They are still pretty awesome, don't get me wrong, right? But in terms of their novelty, they are not really that interesting. We know them for a long time. In turn, when we look at that hole in the ground that is just dirty mud water at 70 degrees Celsius, we actually found so much new microorganisms in this small little pool here that the diversity of new phyla, of new lineages of microorganisms in this pond is larger than the entirety diversity of mammals on our planet combined in this one little mud pond. And so this shows you the tremendous potential that we as microbiologists, as microbial ecologists in particular, have when studying these diverse features. If you're interested, you can actually go online and watch Yellowstone live. The National Park Service of the United States has installed a webcam in the Geyser Valley at Old Faithful Inn, which is one of the most prominent geysers in the world. And if you go to this address, or alternatively, you can just use your phone and scan this QR code, you can start the webcam and it will give you a live view, 24 hours, 365 through the entire year, that shows you a live view, and you can actually watch the geysers in the park erupting. The National Park Service is nice enough to even provide you an update when the next time the eruption occurs. The next eruption of Old Faithful will happen at 2:56 p.m. plus minus 10 minutes. So I encourage you, if you're interested, go to this website, check when the next time Old Faithful is breaking out, be there a few minutes early, and then see Old Faithful go off in Yellowstone National Park. It's really tremendous. It's really a tremendous sight. The last thing I want to mention is, how do we study microorganisms? So one of the things we do is we go to a hot spring and we basically, take DNA from this sample, we obtain biomass and sequence the DNA of the microorganisms that are contained within there. Using this DNA, we then determine what is the genomic potential, what kind of reactions can the organism run, what are potential energy sources, what are carbon sources, and so on. And we then use different assays to test, are these processes actually happening in the hot spring? So for example, we observe whether certain gases that we predict can be produced by these microorganisms are in fact produced in this hot spring. We use sophisticated fluorescence microscopy techniques to visualize where the microorganisms are in the environment. If and how do they hang out with other cells? So this goes back to these friends and frenemies questions we had before. We run incubations directly in the hot springs to determine the biochemistry and the physiology of these organisms using some sophisticated spectroscopic techniques. We sample multiple sites over Yellowstone National Park, not only in one feature, but many features across the park, hundreds and hundreds of them, to get an idea of the biogeography of the organisms. So where are the organisms? And last, we then take all this information that we've gained from all these different experiments, right? And we try to grow them in the lab. Because there are certain things, right, that you can only do if you can actually grow the organisms in the lab, and maybe if you can genetically modify them, right, to learn what these organisms is actually doing on a genetic level. Well, and for that, we need to bring this into culture. And so with that, we think we combine the best of both worlds, right? We try to understand microorganisms exactly how they are in the environment using tests that do not depend on cultivation, that use microscopy techniques, field work, and so on. And then we also try to culture them once we think we've gained enough information in order to bring them into cultivation, into culture. So with that, I thank you for your attention. If you're interested to learn more about our research, feel free to check out our lab website, environmental-microbiology.com. If you want to save time, you can also scan the QR code with your phone. You will land directly on our website to learn more about the people, the graduate students, the postdoc, and the staff scientists in my lab who are actually doing the research, our funding agencies, the specific questions we work on, and some other research that have nothing to do with Yellowstone National Park, but where we also study some extremophiles in deep sea sediments and other organisms in salt marsh sediments. With that, thanks for your attention and have a great rest of International Microorganism Day. Thanks.
It's always so inspiring to see such passionate people presenting their research. So it was a fantastic talk. I'd like to go swiftly into the next one because we don't have so much time for these now. I want to go to Cristina Dorado, who is from the University of Antofagasta in Chile. And this is another pre-recorded talk, but again, please feel free to write comments on social media and we will get back to you and answer if we can, or we'll even get the presenters, the authors to actually come back and answer your questions, have a discussion, debate with you. So without further ado, here is Cristina.
Hello, everybody. Thank you very much for the invitation to give this talk today at the International Microorganism Day. I'm very happy to present a little bit about our research in this very special ecosystem located in northern Chile. The title of my talk is Microbial Diversity and Conservation of Polyextreme Environments of Atacama Desert. My name is Cristina Dorador. I'm working at the University of Antofagasta in northern Chile. This is a satellite image from the northern Chile and also southern Peru, Bolivia, and northern Argentina. All this area is very interesting because it's just the southern part of the Andes, and between the Andes is located this high plateau of the Andes called Altiplano, where some of this area are occupied by large evaporitic basins called salares. In the past, these salares were part of large paleolakes that occupies all this area, and through the time, because of different processes, especially evaporation and also some tectonic movements, produce the isolation of these saline lakes. So now in Chile have been described more than 50 of these ecosystems, and today I will talk about some of them, especially about the microbial diversity that we can find on these systems. And there is my city, Antofagasta, just in the middle of the Atacama Desert. Well, northern Chile is also very well known because of the large capacity of astronomical observations. So from Atacama have been described many important astronomical discoveries, and also it's important not only the astronomical universe, let's say that, also the microbial universe. That is something that we'll try to mention today. The microbes of Atacama have been described a long time ago. This is one of the earliest works that is from 1966. It's a first description of some bacteria that have been isolated from soils of Atacama by the Jet Propulsion Laboratory in the US, showing at the time a very important diversity, especially regarding taxa like actinobacteria and also some firmicutes that are very common especially in the hyperarid area of the Atacama. So since then, many different investigations have been done in the area, as I say, with some astrobiological interest, but also asking especially how this community can live here, where are under, for example, a very high UV radiation and also nutrients limitation. All this information or this research have been, as you can see here, focused on topics about microbial diversity especially in the last years, because of the appearance and also massive use of sequencing without culturing. But also, as I said before, there are a lot of interest about the microbial community composition of soils or Mars-like soils. Also, how it could be the colonization of endolithic or apolitic community. Also, some topics about ecology, environment, and biotechnology and biogeochemistry. So it's a place that is a lot of interest, especially about the microbial communities here. But Chile is a very special country. We can say it's a land of extremes, from the north to the south, to the Patagonia and Antarctica. But especially in the north, it's dominated by arid ecosystem. So here it's possible to find thermal systems, saline ecosystems, also fresh water, and of course, desertic environments. All of them, as you can imagine, are very interesting microbial ecosystems. If you wanted to know more about it, I invite you to read the special issue that we published last year in Anton van Leeuwenhoek about the microbiology of the Atacama Desert. So the microbial ecology of northern Chile have been focused especially in topics about diversity, adaptation, functions, bioprospection, especially regarding the searching for new bioactive compounds, and also astrobiology and extreme environments. So this is a picture of a typical salar here in northern Chile. This is one of my favorites, Salar de Tara. It's located at more than 4,000 meters above the sea level. All the systems are characterized by the high evaporation compared to the rain. So always these places are evaporating water, so that's why they have a negative water balance. Also, the solar radiation is very high. This is one of the places that receive the largest doses of UV radiation on Earth. They present a very high variability, either in the space and also through the time. They also are a system with a high biodiversity, and therefore they present a high endemism at different species level, or I mean at different taxa. And all of these characteristics configured very fragile ecosystems. So where little changes can also produce big effects. So salares, or high altitude saline wetlands, as we usually mention in our publications, are dominated by microbial life. Here is another picture of the same system, Salar de Tara, where you can see this nice red microbial mats that in this case also coincide with a high concentration of iron. So extreme environments are almost by definition microbial habitats, where here in Atacama and in the Altiplano are modulated these communities by high or low salinity, high or low temperature, high pressures, but especially high concentration of heavy metals, high solar radiation, high aridity. Could be system with low and also some with a high pH, different concentration of oxygens, and also low activity in terms of the water activity. Just to give you one example of the different interaction that is possible to describe here on this ecosystem. This is a very nice example I really like. It's from one hypersaline lake of Salar de Atacama. What you can see here, this is snails. And on top of these snails are microbial mats that live next to the animal. And interestingly, also the microbial mats, because of the high production of exopolysaccharides, allows that the eggs of the snail can attach to this matrix and avoiding the release of the eggs to the water. So microbial communities here plays a very important role, not only in terms of the high biodiversity, also functional role that are beyond the biogeochemical cycles. So this spatial heterogeneity I already mentioned, you can see here in this picture. This is a picture of Salar de Huasco at the Tarapacá region in northern Chile. Plus these polyextreme conditions, as already mentioned, produce multiple niches and, of course, different opportunities of diversification. So that's one of the reasons why we usually found a high levels of taxa or microorganisms living here. For example, this is a typical picture of the surface of a freshwater stream in one of these salares, where we know now that these pink mats, for example, are dominated by members of Rhodobacteriaceae or alpha protobacteria. Here, Bacteroidetes. Here, other Hydrogenophaga, and these large colonies, black colonies, are nostoc, macro colonies of the cyanobacteria. Compare with the same ecosystem, just different distance, the landscape looks completely opposite. It's very dry, and these hypersaline pools are dominated by, as you can imagine, halophilic archaea, but also the different bacteria living here, like Rhodobacteriaceae again, cyanobacteria, Bacteroidetes, actinobacteria as well, and many others groups. These also are a typical habitat for flamingos and other charismatic species of these ecosystems. In terms of specific microbial diversity, we found, as I already mentioned, a large bacterial diversity, especially. This system are dominated by bacteria, comparing with archaea and eukaryotes. We describe in one sample, or it is possible to find more than 40 or 30 different bacterial phyla. And in the whole system, we are counting more than 55 different phyla. So it's a very large number. And when we see this composition in terms of phylotypes, of course, the diversity is increasing quite a lot. The most dominant groups are Protobacteria, followed by Bacteroidetes, Cyanobacteria, and others. And also here is a big role and a very important role of the red biosphere in different activities or biogeochemical processes. So in terms of the network analysis, we can see here that every site that we have sampled, and also every sample, have an important number of unique types, and some of them also are shared between them, either with DNA or with cDNA, showing this very important kind of fingerprinting of different salaries. So each salar is different in terms of the microbial composition. Just to give you an example of this, this is a study we have done this many years ago, but I really like because it's just in a very small microbial mat. It's not more than 5 millimeters. So it's basically a complex biofilm, stratified phototrophic biofilm, where we found different layers regarding the color: pink layer, green, gray, and black. And in this microbial mat, we detected high diversity and also very nicely different functional groups dominated by phototrophic bacteria, especially in the surface layers. And then because of the anoxic condition at the 6 millimeter depth, it's possible also to see other taxa as typically is found in microbial mats. But again, that's happening in a very small mat. So you can already maybe imagine how important are bacteria and microorganisms in this ecosystem for different function. Well, we also have works in description of new bacteria. in some of the salaries. Our group have participated in the description of 2 streptomyces, Streptomyces actinoplanensis from Salar de Huasco and Streptomyces huascoense, also from Salar de Huasco, both of them producing very important, or having the potential to produce very important bioactive compounds. And also this another bacteria is another actinomycetes, subtercolabile, from Lago Yuyayaco. That's one of the highest lakes described yet. It's more than 5,000 meters, 6,000 meters, sorry, altitude at almost the summit of volcano Yuyayaco. Another topic that I want to talk today is about environmental issues, because as already mentioned, these are very fragile ecosystems. And salaries now are very very usually found talk about them in the press, because especially in South America, because they're also a very important source of lithium. As you may know, lithium now is one of the most important salts to produce lithium-ion batteries that are everywhere in our computers, in our cell phone, and of course in electric cars. So there's a very high demand to obtain lithium. And in Chile and also in the other salares, they're very rich in lithium, especially Salar de Atacama. So in Salar de Atacama, we see these very nice microbial mats and flamingos that are, of course, part of this trophic web, where the base are these photosynthetic communities. But at the same time, in the same ecosystem, we have these pumps that pump brines from the salar every day and night, thousands of liters per second of brine, filling these big evaporation pools, and that produce at the end of the process brines that are enriched of lithium. This is the greenish color. So here in this GIF image, you can see how this evaporation pools have been increasing at the southern part of the Salar de Atacama through the time, actually in the last 20 years. It's very impressive to see the coinciding, the high demand of lithium and also the growing of these evaporation ponds. So the question here is, this human activity is really affecting the ecosystem. There's some evidence showing that it is. And what we're trying to also to work currently is to figure out if how that could affect actually microbial communities. It's a very complicated question because, as I already mentioned, these are terminal ecosystem salaries. So it's not easy to see here environmental damage of bacteria. It's a very complicated question. So we're working on it. Our first approach was to understand or to know if these lithium brines are actually a microbial ecosystem or not, because it's a human-made environment. So what we did was compare the microbial communities that are present at the natural brine that are actually pumping, compared with the concentrated brine that is a final product of this process. So natural brine are dominated by sodium chloride and the concentrated brine by lithium chloride. And what also has found that these concentrated lithium brines of Atacama are one of the highest saline places on Earth, with more than 56% of total salinity. So we actually found differential microbial communities in the natural brines compared with the concentrated ones. The natural ones are dominated by archaea, and the concentrated are dominated by bacteria. So that was one very important result for us, because we we cannot really explain, not explain, but we were very surprised to find these differences, and also to find that a lithium brand called harbor actually a very diverse community of bacteria, dominated by Firmicutes, especially Bacillus. So here we report the presence of bacterial archaea in these brines, demonstrating that the hypersaline lithium brines of Atacama support viable microorganisms and represent the upper saline limit of life on Earth by date. Well, another example of environmental damage of this ecosystem have been the continuous extraction of water of these salares, especially the ones are located at the Altiplano, for the copper industry. So here, this is an example of Salar de Lagunillas, near Salar de Huasco, that during 20 years suffer water extraction. And you can see here from the images of Google Earth, how the surface of water have decreased dramatically. So in 20 years, thousands of years of evolutionary and biological history disappear. So that's also I wanted to say that the damage that produce the extraction either of brine and waters is beyond the physical state. I mean, it's beyond to take just water or brines. It's also affecting the whole ecosystem and a whole evolutionary and natural history present in these ecosystems. So at the end, I wanted to say that the microbial diversity of these salares is not only characterized by the presence of multiple bacterial phyla, that also we can say that are microbial diversity hotspots, but also the frequent presence of unknown rare taxa. This high diversity reported here and elsewhere will be related with a broad range of tolerance or adaptation to extreme conditions that are really poly-extreme conditions, and the presence also of multiple microniches, and probably having different functions, of course. So this diversity and their variability has important consequences in the function of the ecosystem, and they're key to sustain other species that are present in this area. So extreme environments in northern Chile are unique and need to be preserved. And it is necessary, of course, a national and probably an international strategy of conservation for extreme environments and also extraterrestrial analogs. And here, again, is our role as a researcher to maybe focus on some of this question. What we really need to conserve? The environment, the taxa, or all together. So it's a very complicated issue for microbes. So again, I think Atacama gives us a lot of examples to work on this direction, especially when we are not only dealing with this human intervention, but also probably with dramatic changes in the future because of climate change. So thank you very much for your attention, and enjoy this International Microorganism Day.
Okay, thank you very much to Christine there. Now it's time to go to Felipe de Carmo from the French National Institute for Agriculture, Food and Environment in France, and he's going to talk about probiotics' role in gastrointestinal disease. This is the penultimate talk before we're going to take a little break before we move into our breakfast show, but please feel free to comment on social media, ask questions, and yeah, get in touch with us, we'll respond. Okay, without further ado, here is Felipe.
Hi everyone, I'm Felipe Ducam. I'm here to talk about probiotics' role in the gastrointestinal disease. Okay, the suture here was developed through a collaboration between the Federal University of Minas Gerais and the National Research Institute for agriculture, food, and environment. The collaboration between Brazil and France form International Associate Laboratory, or LIA. The aim of this LIA is to identify bacterial determinants involved in pro or anti-inflammatory process to characterize inflammatory process in disease and develop new treatments. If we do that, we use beneficial bacteria or compound. Okay, first introduction. Gut microbiota is diverse and complex. It is colonized by up of trillions of bacteria with a high diversity of species. The microbial colonization of human gut begins at gestation and birth, and immediately thereafter, the microbial changes. And several studies show the most of part of these bacteria are bacteroids and firmicutes. And this complex microbiota has immunological equilibrium, but when we have a disequilibrium of dysbiosis, we have an increase of pathogens, bacteria, and consequently we have inflammation in the gut tract. You have intestinal inflammation. Gut microbes are key to many aspects of human health, including immune response and the metabolic process. We know that gut microbiome can influence a disease, but it's obvious that we can influence too. Diet and the medication have a strong influence on gut microbiota composition. And we can do the same thing using probiotics, because probiotic supplementation has several beneficial effects on human health. Okay, but how does probiotics do that? Probiotics can act through different mechanisms of action, like enhancement of epithelial barrier, increased adhesion to intestinal mucosa, inhibition of pathogen adhesion, competitive exclusion of pathogen microorganism, production of antimicroorganism substance, and also modulate of the immune system. This is very interesting because a specific strain has one or more mechanisms of action to modulate the growth response. And we have a specific strain for a specific condition. In addition to realize these metabolites can act to regulate and mitigate inflammatory process, probiotic bacteria have structures that are implicated in response to the growth, like sulfurous proteins, flagellum, pil, and capsular polysaccharides. These structures, which can regulate signalization pathway to produce cytokines, inhibit apoptose, and attenuation inflammation, and enhance the function of gut epithelium. Okay, the presence and the identification of bacteria is compounds such as surface layer proteins can help us to select a bacteria or best candidate to a probiotic. And these surface proteins form a paracrystalline bidimensional array in external cell wall. And the surface protein may be involved in key functions in probiotic role, like resistance to extreme environment conditions like acid and biliary salts, bacterial persistence in the gut, adhesion to host cells, and immunomodulation. We know that S-layer proteins are recognized by dendritic cells and can stop inflammation in the host. Moreover, we have several criteria that help us to screen probiotic candidates, like assays like a simulated gastrointestinal condition, MEF and zeta potential that we study bacterial surface, antimicrobial activity, adhesion into intestinal epithelial cells, and also in vitro challenges in inflammatory conditions. These informations can help us into in vivo models, like models mucocytes and colites. These are characterized by chronic inflammation in the gastrointestinal tract, in the main present in two forms, ulcerative colitis and Crohn's disease. And ulcerative colitis is characterized by continuous inflammation limited to the colon and affects only the mucous layer, whereas Crohn's disease can occur in several segments of the gut. It can affect all layers of the intestinal wall. Another inflammatory condition is mucositis that affect a large portion of patients who undergo constant treatment with antineoplastic like 5-fluorouracil. Mucositis is a severe inflammation that affects the interdigestive tract. In the same time, the treatment for mucositis and colitis are ineffective and also cause a serious side effect. Some studies propose to use probiotic bacteria to help the treatment of mucositis and colitis. And one of these bacteria is a candidate. It's a Propionibacterium frenderichi, a gram-positive bacteria, safe to consumption, used as cheese or heparin starters, and they have a great probiotic potential in gastrointestinal disorders. And also, these bacteria have a surface layer protein. The surface layer protein is SLPB. This protein is we think it's involved in anti-inflammatory process. And we investigated the probiotic potential of Propionibacterium frenderichi. We selected 7 different Propionibacterium frenderichi strains, and we saw that different profile the adhesion to intestinal epithelial cells, 1829. The strain 129 is the strain of most of high potential to adhesion to epithelial cells in vitro. And this strain possessed a SLPB protein. And after knockout of this gene, we saw that significant diminution of adhesion to epithelial cells. That implied that protein that's a knockout gene, it's involved in decrease of adhesion to epithelial cells. Moreover, the impact of SLPB gene mutation in Propionibacterium frenderichi 129 decreasing complete and change complete the surface extractable proteins, surface charge like a potential zeta, and the surface hydrophobicity. And moreover, stress tolerance for acid and bile salt and health stress are completely altered. This implied that protein, SLB proteins involved in several processes and several criteria that we use to select a probiotic candidates. And to explore the effect of SLPB gene mutation on immunomodulatory properties, we subject both strains, wild type strains and the mutant strain, to in vitro challenge in 1829 cells stimulated by LPS. The relative expression of anti-inflammatory and pro-inflammatory genes are verified, and we saw that 129 strain, the wild type strains, induced the expression of EL10 and the other inflammatory cytokines, but mutant strain lose the ability to induce EL10, and moreover, the mutant strains increase expression of pro-inflammatory cytokine like EL8. Moreover, we challenged both strains in vivo mucocyte model induced by 5-FU, and we saw that ability of wild type strain to mitigate the inflammatory process caused by chemotherapy in mice, but wild type strains lose this ability. And moreover, we identified that 129 strain it's able to enhance epithelial barrier. And in this way, it's evident that the SLPB protein has an important role in the anti-inflammatory effects of Propionibacterium friedreichii 129. So a protective matrix like cheese, it's essential to prevent the proteolysis. In this work, we see that the cheese prevents these proteolysis, and moreover, we see that SLPB protein it's able to induction only the SLPB protein is able to induction production of EL10. Okay, finally, we produce in industrial conditions an MHL cheese use the same strain, Propionibacterium friedreichii 129, in combination with other two strains, Lactobacillus and the Streptococcus, and we investigate the probiotic potential of the emmental cheese in the SS coli models. We did pre-treatment of cheese, and after the induction of colitis, we saw that a consumption of emmental cheese reduced severity of subsequent DSS-induced colitis inflammatory process, and we have mitigate histological score and the other parameters like disease activity index and weight loss. These are very important parameters for colitis disease. But the main question is, does this always work? No, unfortunately, no. We try to develop a functional food with other strain, Lactobacillus casei 01, and unfortunately, we don't have the same results we saw with emmental cheese. For conclusion, the probiotic strains, one or more, needs ability to modulate the immune response and control the pro-inflammatory process, and moreover, improve the epithelial barrier or modulate the microbiota as a specific strain for a specific condition. Thanks to everyone who took the time to watch this presentation.
Thank you to Felipe, and we're going to move on to our last speaker of this session before the break, and the speaker will be Omololu Fagunwa from the University of Huddersfield. and he'll be talking about thank guts, food fiber, and gut microbiota. Please feel free to get in touch with us and take part in the online activities where you can see these talks, comment, interact with us, interact with other people behind International Microorganism Day, and also the presenters and the speakers. So feel free to reach out, and without further ado, here is Omololu.
Hello, my name is Omololu Fagua. I am a PhD student at the University of Huddersfield. I'll be talking today about food fiber and gut microbiota. The title before me is what I term tying gut. I'll be talking about the importance of fiber and how it helps our gut microbiota. For introduction, let me point to you that our gut microbiota determine the state of health which we find ourselves in. So we have many factors that help us progress from disease state to healthy state. And what the diagram I've showed here is just to let us know how our gut microbiota affects our health. A reference man, average man of 70 kg, has about 3.8 times 10 raised to power 13 bacteria in the body. That is trillions of millions of bacteria in the body. And this figure is very similar to the one found in human cells. So if you look at the bacteria we've got in our body as an average human being or man, with about 70 kg, is just about the same number of bacteria we've got in our body as the same number of human cells we have. If you're talking about red blood cell, any kind of cells at all, if you look at the number, it's very similar to the number of bacteria which we've got in our body. The gut microbiota is influenced by a number of things, such as genetic makeup, genetics, also the host physiology. What I mean by this is maybe the age of the host. An infant, for instance, has very low diversity in terms of microbiota for an infant compared to an adult. The same thing as we age, the gut microbiota get depleted in terms of diversity. So age of the host determines, likewise state of health, maybe there's a presence of a particular disease, condition, or the absence, as well as stress and some other physiological components that affect the microbiota. Apart from genetics, physiology, there is another aspect, which is environmental factor. This includes living condition. If you live in a clean environment, for instance, the amount of and the diversity of microbiota, the bacteria in your body, will be a little bit different from someone that lives in maybe an unkept environment. For instance, people living in urban and rural area, the bacteria they've got inside them are quite different. Also, medication influence the diversity of bacteria in the body. Someone that is being placed, for instance, on antibiotics, we have reduced number in terms of diversity of bacteria in the body, because the antibiotics, though it's actually being used for treatment of a particular condition, will be affecting the bacteria at the same time, and some other medications. What I've got here is just for the sake of differentiating between, we all hear about probiotics. There's difference between probiotics, prebiotics, and symbiotics. Probiotics are live bacteria and yeast that, when it's administered in a viable form, has adequate amount that is beneficial to human beings. In most cases, well, what we see in our food supplements, yogurt, and all that are probiotics. What about prebiotics? Prebiotics are kind of substrates. They are kind of what microorganisms can add on so that they can confer a benefit to us. What do I mean by this? There are things like fiber. There are things like carbohydrates that the microbiota can, this bacteria and yeast can easily access so that they will confer benefit to us. What is symbiotics? It's kind of mixture between prebiotics, the substrates such as fiber, the probiotics such as what you've got in your yogurt, in other food supplements, lactobacillus, and the rest. So fiber on its own, prebiotics now, fiber on its own is pro-health because there are various studies that have been done. There is a systematic review. There are systematic review to see all the studies that have been done on fiber to see, are they really pro-health or not? So I think it's established that fiber is pro-health. It reduces the risk of developing heart disease, diabetes, arthritis, and even all-cause mortality. So what are the fibers we're talking about? You can get them from fruits and vegetables, from nuts, from legumes. These food substances contain lots of fiber. So we need to understand that fiber don't feed us directly. They directly feed our bacteria, our gut bacteria. When we eat fiber, we don't derive that direct benefit from them. We derive indirect benefit from fiber. So what do I mean by this? What I mean by this is that when we eat fiber, any kind of, especially indigestible fiber, because they are not easily digestible by our own gut system. So what it does is these fibers have been acted on by bacteria. They have been digested by bacteria, and when they have been digested by bacteria, they produces a lot of chemicals and other biomolecules that we benefit from. For instance, short-chain fatty acid, which has a lot of health benefit, butyric acid, propionic acid, and some of all these short-chain fatty acids. So in other words, when we eat fiber, indirectly, they give us the profound health benefit to us, but directly, bacteria feed on them, and the products from the bacteria is what benefits us. So what kind of fiber help most? We don't fully understand this, but there is hints that fibers that contain fructans and galactosaccharides, they correlate with abundance of bifidobacteria, lactobacillus in fecal. Another question would be, how much fiber do I need to consume? I think there is a study that said less than 5 grams fiber is still enough for fermentation to take place. That means if you consume less than 5 grams of fiber, they will still be useful for the bacteria in the gut. You can consume more than that. And the question we now need to ask ourselves, which is the basis of this presentation and my work at Huddersfield, is what class of bacteria should we consider? We should look at fiber-loving class of microbes. What do I mean by this? We know of our lactobacillus, we know of our bivid, but beyond the popular lactobacillus you get in your yogurt, beyond the bifidobacterium you get in your yogurt and some other food supplements, there are some other class or genera of bacteria which are very useful. Example of them for the bacteria is the bacterials. Bacterials are very, very useful. They are probiotics on their own, though you can't readily get them in store. And that is where fiber, eating fiber, that's where it comes in. Because you can't readily get them as probiotics in stores, but if you feed your gut well, they can replicate these bacteria in large amounts to confer health. Also, I would like to point out that apart from bacterials, another fiber-loving class is Lactobacillus. Is a kind of order, which a family rather, is a family which I'm going to talk about in a minute. There are lots of fiber-loving class or genera, but I'll only focus on these two. So if we look at this figure here, this is actually my PhD result, one of my results. I put this up here for just to show us example of the importance of fiber or the benefits of fiber. A Western diet will look somewhat like this. If Western diet would look something like this, with high sugar, high fat, high meat protein, with low fiber and all that. A prudent diet, which has mixed of, sometimes it has sugars and some fat, but it's just being prudent in that there is fiber, there's balance of everything. Will look somewhere like this. And what I did in my study is that I had two class or two groups of people or two groups of samples, one from the Western diet and the other from vegetarian, which the presumption here is that Western diet contains high fat, high sugar, and vegetarian diet contains high fiber. Actually, in my studies, what I did is the participant filled out food diary for 7 days. So from the food diary, I have been able to analyze what people are taking and see the amount of fiber in it. So when I classify based on Western diet and vegetarian diet, what I mean in other words is vegetarian diet for real, high fiber, Western diet are low fiber from the participant that participated in my studies. And we get to know this from the food diary they fill. So from this result, I mentioned that Lactobacillus family, fiber-loving family are the kind of bacteria which we want in our gut, really. They are present there, but at the same time, in Western diet, it appears from my result here that it's really low. So the good bacteria, if you look at the green lines, that's for the vegetarian diet, you can see lots of them falls on the green, and very minimum falls on the Western diet. So when you looking at from 0 to 100% being the highest, you see the majority falls above for vegetarian, they fall above 50%, majority of them. While Western diet is just, in most cases, is just about 20-30% for the Lactobacillus family of bacteria. And from this family, things like bacterial, things like Eubacterium rectale, Rosburia, which we get from some yogurt, Lactospira are the kind of bacteria which we get from them. So these are kind of family of good bacteria or probiotics. Another result which I would like to show you is that apart from the Lactospiraceae, which have shown that high fiber tends to give more of that family of bacteria, is another probiotic called the Bacterials. So I looked at the general really, and what we can see here is that, for instance, the Dietary, Cyanomonas, Alistives, and some of these good classified probiotics. I put it as classified because they are not things that you can easily get in stores in form of yogurts or bio drinks. But what we can do is to have high fiber or get good fiber in our body. The bacteria hat on the fiber, it produces this bacteria in good quantity that will be beneficial to our health. Again here, it shows that vegetarian of high fiber contains lots of these classified probiotics. But beyond eating fiber, another thing which I found out from my study is that we need to watch salt intake as well. So it's not just getting the good fiber in there, expecting that there'll be good bacteria or probiotics in right quantity or large quantity. Salt can affect the amount and the type of bacteria as well. This is one of those things we've been able to get from our studies. For instance, what I did here is I classify, still using food diary from the Western and vegetarian. Because people consume salt, from the food diary, we were able to know the amount of salt people consume. So we classify them based on low salt, average salt, high salt. 6,000 milligrams per day is average salt, recommended salt intake or dietary salt intake for an average human. And people that falls far above the 6,000 milligram or 6 gram, like 10,000, 12,000 are classified under high salt. So when you see high salt, are people that have consumed salt per day up to 10 grams, 12 grams above. And average is around 5,000, 6,000 milligram. Low salt is about 2,000 to 3,000 milligram per day. So what we was able to get here is that people that consume low salt it correlates, high dietary salt correlates with reduction of these probiotics. When the salt intake is high, probiotics reduces, especially for this class of this family of Lactospiraceae. An example of Lactospiraceae is Blautia. And we're able to get the same thing from this Blautia, the same thing with other different bacteria from this very family. So salt influence the amount and the type of probiotics you get, despite the fact that you are eating high fiber or you are eating fiber. So that's another thing. Don't just look at the fiber you're taking or trying to take in fiber. At the same time, look at the salt intake as well. So that's for my little presentation on this occasion of International Microorganism Day. I have been able to tell you that we have fiber helping our gut microbiota, and we should take notice of fiber at the same time of our salt intake. Thank you very much. My name is Samuel Olu, University of Huddersfield.
Fantastic. Thank you very, very much to you and to all of our speakers for this last session. It's been fantastic. It's been very, very educational. What you may have noticed from the last session is lots of science, lots of scientific talks talking about research and relating those back to real world problems and solving them using microbes. But also some of the problems that microbes cause. In the next session, we're going to have a break now. It's going to be about 50 minutes long. We're going to have some more science talks, which are going to be very, very interesting. But then we're going to have the breakfast show. We're going to have education, the education morning, followed up with loads and loads of talks over the afternoon. So I really encourage you to stay tuned, keep up to date with what's going on, and wherever you're watching this, there will be a link to the program for International Microorganism Day, so you can see what's going on throughout the entire day, and you can take part whenever you like. So thank you very, very much for watching now, and get in touch, talk to us on social media. We'll be back in about 50 minutes. Thank you.